Hallo, guten Morgen. The video link is done as well, I think. We have, I don't know actually how many we have. We have quite a few. We have, I think, 70 or 80 in here when they're all here. But we have, do you know the number on the video link? More than 100 on the video link. So good morning and welcome to the Technical University of Munich, to our 12th symposium, HEF Symposium on the limits of food production, vertical farming. My name is Zentholt Assink. I'm the chair of digital agriculture. I'm also the director of the Hans-Eisenmann Forum. The Hans-Eisenmann Forum is a central institute of the Technical University which brings together all the shares of agriculture. Before I continue, I'd like to introduce Claudia Luksch. She is a managing director of the Hans-Eisenmann Forum, and she will take you through some logistic aspects.
Good morning, very welcome from my side. Warm welcome to everyone in here. It's an interesting view because everyone's sitting so far apart, but it's nice to have you all here on site again. It's our first time since 1.5 years to have people really in our lecture hall again. And it's our first time we do it as a hybrid event. So please excuse us if there are sometimes a little bit of technical problems or if something is not perfect, but we are learning. Before we start with the content and the talks, I would like to give some organizational issues here some space. And can I have the first slide? Oh, I have to do it myself, okay. Okay. So all of you have on site have gotten a flyer with the COVID rules. So keep your distance, wash your hands, wear your masks when you're walking around. You can take them off when you drink your coffee, of course, and I think you can also take them off when you sit here. The building is currently under reconstruction or remodeling, so please watch your step when you use the stairs because they're not light and illuminated. The good side of it, you can spill your coffee because the carpet is going to be exchanged next week. So don't worry about that. Then technical issues. We have a detailed program with all abstracts and the CVs of all the speakers on our website. And we have a Q&A session also here, but you can also use the tool Slido, as well as people online can do it. All the QR codes you need for that are on the flyer you just received. And everyone taking part online, you received all the necessary links with an email the last couple of days. If not so, please contact us via email. We can react on that. Please note that all presentations and the whole event is going to be streamed live via YouTube and will also be available afterwards. So if anyone wants to see something again, that's possible. So that's for me all this boring stuff. Now, I give it back to you.
Thanks, Claudia. Let me introduce our Senior Vice President, Professor Kramer. He will give an opening address to the audience for the symposium. Professor Kramer.
Also ich höre gerade nichts. Ich weiß nicht, ob Sie mich hören.
Wir können Sie hören.
Ah, jetzt höre ich Sie. Sehr gut. Soll ich einfach anfangen?
Ja,
oder
ja, ich habe Sie schon vorgestellt. Also nochmal, der Senior Vice President, Professor Kramer, gives the opening address to the symposium. Professor Kramer.
Thank you. My apologies. I didn't hear you at the beginning, so I didn't know whether I should wait or not. But let me then begin. I see I'm on a very big screen. I'm actually calling in from the Saantal, which is in South Tyrol, somewhat south of Munich in the north part of Italy. So let me begin. Yes, dear colleagues, dear students, dear friends, welcome to today's symposium on the limits of food production and the opportunities of vertical farming. I would especially like to thank the organizers of this symposium, Claudia Luksch and Sentholt Asseng, who both direct the activities of the Technical University of Munich's Hans-Eisenmann Forum. This forum serves as a network for our agricultural science researchers. Your participation today shows the importance of vertical farming and of cooperation across scientific areas. The topics of climate change and sustainability are currently receiving a great deal of public attention. In this respect, there are particularly strong challenges in the area of agriculture. Agriculture is a 7 trillion euro global industry and occupies about 40% of the livable land on Earth. Food production generates a large part of all greenhouse gases. Moreover, by 2050, the global population will be 9.7 billion persons. And if the prosperity of densely populated areas continues to grow and our diets do not adapt, then we will even need to further increase food production. A key building block for food security is vertical farming, which promises several improvements in crop production. For instance, one can decouple production from seasons and climate, ensure consistent quality, harvest several times per year, reduce land use, improve nutrient efficiency, reduce adverse environmental effects, and enable regional production with lower transport costs and losses. Vertical farming can mean higher yields, fresher produce, and a smaller carbon footprint. Vertical farming is an integrated system with many variables. One must, of course, address lighting, but also humidity, airflow, irrigation, and many other factors. To manage the system, the skills from many sectors must be brought together, including mechanical and electrical engineering, software development, crop science, and biology, to just name a few. This diversity creates complexity, and there is generally a lack of understanding of the complexities of vertical farming as a business. For instance, it costs hundreds of thousands of euros to erect a mid-sized vertical farm, and energy use might be prohibitively high. Advances in technology can help by connecting automation, machine learning, and cloud-connected software to vertical farms. Firms can increase capacity and optimize a tremendous range of cultivation variables. Vertical farms can thereby become profitable, and the market is expected to be worth over 10 billion euros in the next 5 to 10 years. At the Technical University of Munich, our Entrepreneurship Research Institute works with our affiliated institute Unternehmertum to help establish growth-focused and technology-based startups from the idea phase to the founding of a company and to a rapid growth phase. In doing so, we have become Germany's leading higher education institution for startups. We bring about 70 to 80 tech startups to market each year, and these companies attracted more than 1 billion euros in investments in 2019, and it's similar in 2020. Together with Unternehmertum, we aim to become a European leader in technology-based startups, and we support these teams with a network of incubators. Known as the TUM Venture Labs, these centers develop deep tech startup families in selected areas, such as food, agro, biotech, aerospace, healthcare, robotics, and artificial intelligence and quantum technologies. The Venture Labs provide technical and social infrastructure, entrepreneurship coaching, and support from businesses and investors. At TUM, we respond to societal problems on many levels through our research, teaching, and entrepreneurial activities. We take a transdisciplinary approach that incorporates the natural sciences, life sciences, engineering, social sciences, economics, and medicine. We are now developing strategies and new approaches to sustainable agricultural and food production, and researching methods to counter the impacts of climate change. In particular, the TUM campus in Weihenstephan offers a unique environment to establish an internationally visible center for vertical indoor farming. For this purpose, pilot facility is being planned where new approaches to vertical indoor farming will be implemented and which will let us present our ideas to the public. In closing, vertical farming, research, and industry are just starting to take off. Thank you for your attention. I hope and wish that you have inspiring discussions throughout the symposium.
Thank you, Professor Kramer. Thank you for the nice opening words. To get into the conference, I want to show you a couple of slides to sort of, as a bit of a teaser, where we actually want to get to today. Can I get the next slide, please? I do this here, okay. Many of you know the law of minimum, which says that the scarce resource is a limiting factor for growth and yield, which was actually developed by Karl Sprenger about 200 years ago, but it was popularized by Justus von Liebig, and it became actually named after him as Liebig's law, or the law of minimum. And since then, it actually has been guiding crop production in the field of how to manage crops. And what it actually says, whatever is a limiting factor keeps your production or your yield back. For instance, if you don't have enough water, you can't increase the yield anymore by putting more fertilizer on. It's water, in this case, is a limiting factor, as other factors can be limiting as well. The question is now, what are the limits of growth in a vertical farm, where all the factors, everything which sort of drives growth can be controlled, including light, including humidity, including the CO2 concentration, and of course, water, nutrients, and so on as well. So what will happen when we then apply the law of minimum? How do we actually know what are the limits? And I think we come together today to actually discuss this, to find out how far can we actually push the system? What are the next limits of production when we control everything? With that, I'd like to hand over to Eliza Stein. She is the moderator for today and will take you through the program. Eliza.
Thank you very much, and good morning to everyone, or whenever you are joining from the world, good evening or good night already, or good afternoon. So I'm really happy that we are discussing this very important topic today. We've heard it already, the importance, because we have a growing population, and of course we need food, we need space to live, so it's important that we discuss this topic today. And therefore we have invited different experts from all over the world coming together to discuss the topic, but also sharing their research results. So we've already mentioned it's a hybrid event, which is really the most complicated form of event, and we hope that everything works out well today. One important issue, because a hybrid event always is complicated, so that we can combine the digital and the live audience. And to do that, we also integrated a poll later this day, where we also want your participation, and we also want your questions. So your questions are highly appreciated. Please always ask your questions. Therefore, we have integrated Slido, and you will find the link below this streaming window. So if you go into the descriptions at the very bottom of the text, there's a link to Slido, or here you have also the flyers, and there you can scan the QR code coming to the Slido poll and Q&A. Please make use of it. After each speech, we have the chance to have like 5 minutes to discuss your questions. And then at the end of this event, we also have a panel discussions with almost every speaker. And there we can go further into details. So make sure you won't miss it and make sure to really participate asking your questions. They are highly appreciated. But now we would like to start with our first speaker. And he gives us an introduction about vertical farming, and we will also learn more about the crucial role of LEDs. And the presentation is held by Professor Dr. Leo Marcelis, and he is head of the chair group Horticulture and Product Physiology at the Wageningen University, which is in the Netherlands. And his research focuses on sustainable production of high-quality products in vertical farms and greenhouses. Please welcome Professor Dr. Leo Marcelis.
Thanks a lot. So good morning, and I just realized good evening or good afternoon wherever you are. So I'm very excited to be here on this symposium. Also for me, the first symposium since long where I could really be in person. And yeah, I'm very happy to discuss a bit about limits, but in this talk I thought, well, I have prepared a bit of a general introduction on vertical farming. Well, we've just heard the talk of Professor Hoffmann, and well, he addressed a lot of these things already, so we'll do it just very quickly. Just one second. Yeah, that's better, eh? Yeah. Okay, good. Well, I still hear myself, but so there is climate change. I don't need to say that many places of the world are water shortage. So drought is a very big issue. In agriculture, we're still using a lot of pesticides. That's something that we want to get rid of. Natural resources are being depleted. As also indicated by Professor Hoffmann, the world population is rapidly growing, more and more living in an urban area, in the big cities or mega cities. And this all together requires, in fact, that we develop a new vision on our food production system. How are we producing the food? And I would now talk in particular about vegetables. What do we want with the vegetables? First of all, they need to be safe for the consumer. No contaminants, no pesticides on it. They want, so that relates already to the health, but in fact we want to have them promoting our health. So we want products with higher nutritional value, for instance, extra vitamins. Of course, they need to be tasty. We want to be produced sustainable, but also local production. And in particular, the whole COVID crisis has made us even more aware of the consequences or the difficulties in transporting products, and in particular food, over the whole world. So what I see is more and more need of local production. Vertical farming, vertical agriculture, and if I talk about vertical farming, I'm referring to a production inside buildings where we do not use solar light, so the light is usually coming from LEDs, and where we can have multi-layers above each other, but it can also be a single layer or maybe vertical walls where we grow the crop. So other terms could be indoor farming, or in Asia it's often called plant factories with artificial lighting. What I think is the most important of vertical farming is that we have a full control of the whole production process. So we can control everything. We can have a very high yield, so we don't need much land for production. And it means we can grow the crop anywhere. We don't need a fertile soil. It can be in a very hot area, so it can be in the tropics. It can be in Arctic, so where it is very cold. And it can be nearby the cities. What I think is important is really that we can say, well, we can have a strict guarantee that we produce so many kilograms of a defined quality on a specific day of the year. Vertical farming, it is all still very new. or at least in the commercial sector. But what we see at this moment, there's a lot happening. There are huge investments, and I did not try to have here a complete overview, but just a few examples of investments. And those investments in vertical farming, they usually go not with a few million euros at a time, but often with hundreds of millions at a time. So it's really huge things are ongoing. And of course, we can then discuss how economic is it. It is all new. We do not know all the economic figures, but definitely there is a development ongoing at this moment. To understand that, it might be good to look a little bit back in history. So what you see here are some pictures of about 100 years ago of some first forms of controlled environment agriculture in the Netherlands. You see, well, just only a very low technology level. This is probably somewhere in one of the years that I was born, so probably in the 1960s. Greenhouse was already a little bit different. This is how a modern greenhouse looks like, or could look like. I have to be fair, well, I took this picture already 20 years ago, but this is still how what you can find nowadays is as greenhouses, where you see some lights are being used. Here there's still HPS lamps. They are still used a lot, but what we see now, if new greenhouses, they more and more are using LEDs. Why do I show those pictures? That's showing that the technology level increased, the level of control increased. And if you realize that, then maybe it's not so surprising that the next step in technology level and in control of the production process is the production of crops indoor without solar light. And I also think those two sectors, maybe they are partly separated, but it will become a gradual change between them and learning from each other. There are also many different types of vertical farms or indoor farms. It can go from a home appliance to a large-scale production facility. Well, we can debate whether we could call this a vertical farm, but at least some of the ideas of growing a crop indoor, what consumers could do. And that, of course, well, the most important thing is that then people get connected to what they eat and that they grow their own. We see a number of supermarkets having cabinets where they grow the crops. Like here an example of a supermarket. We see container farms, and it can be a large-scale production systems. So there are a lot of advantages of vertical farms, but I think it should also be that we should look at what are the bottlenecks. And I think two of the major bottlenecks is the use of energy, and it is the costs. Well, the energy is mainly because we use lights. We need, and we use LEDs. LEDs are very efficient, but still, we need a lot of light. Therefore, we need a lot of electricity. From a sustainability point of view, there's definitely a need to reduce it. Even if we use renewable energy, I think from a sustainability point of view, we need to reduce it. But also it means a lot of costs. And in particular now, at this moment, energy prices are rising very rapidly. That makes us even more aware of the costs for, well, overall costs, but energy costs are a substantial part of the cost. So what actions are needed? First of all, we should reduce the energy use. That starts with developing more efficient LEDs. LEDs, they have become much more efficient in the last 10 years. And with, I should call it efficacy, so how many micromoles per joule electricity can be produced. I assume in the coming years this can be improved further, but there will be limits to it because they are already very efficient. So the biggest step should be made, in fact, that the plant can make better use of the light, so improving the light use efficiency. And I think even though we do already a good job in these indoor farms, there's still a lot of room for improvement. So that's also what in our research and many research groups all over the world are working on improving the light use efficiency in order to reduce energy costs, to reduce energy and related costs. Of course, it is about the plant, because that's a primary thing, but it's also looking at the whole technology, the climatization, and maybe also looking on whether we can integrate it in the energy system of an urban area. Altogether, I expect for the coming years a lot of improvements here. I think also the cost price will go down. However, I don't expect that it will become very cheap. Is it then a problem? Well, then it depends on the value what we produce. So if we have a better product, which is possible, a product with higher added value, well, even if it is then not that cheap, it can still be economically viable. As I said, we're going to more and more control of the crop. Light is one of the major factors where we can control the growth of the crop. And if you're talking about light, there are many factors. It is first, of course, the light intensity, how much light is there directly affecting the photosynthesis. It is about the photoperiod, or day length. It can have a lot of effects on the development of the crop, on the flowering. It is about the color of the light. What color is ideal? Also what you see in research where it used to be all those conditions like spectrum and intensity, that initially a lot of research is on constant conditions. I think the next step will also be on thinking about how to adapt it during the development of a crop, how to change it during the time of the day. Okay, so 3 factors: light intensity, photoperiod, light quality, but then there is also the direction of the light. Solar light typically comes from the top. So that's, of course, the most logic place now to have the lamps on top. But for a number of crops, we can also have the lights in between the canopy, or maybe even from below. That also has a lot of physiological consequences for the crop. Still not yet fully explored. And we should never forget that light also represents heat. So it affects the air temperature, but even more the temperature of the plant, and that's in the end the most important one, and it affects the transpiration of the plant. If we then look at light, in particular the spectrum, there are a number of photoreceptors in the plant. So the most well-known are the UVR8 for the UV light, although at present in vertical farms, at least in the commercial farms, you will not see much of UV light. The cryptochromes, photoreceptors, in particular sensitive for the blue light, and the other most well-known are the phytochromes, sensitive in particular to red and far red light. Now I would like to show a few examples of far red. So far red, when I talk about, let's say, all the light between 700 and 800 nanometer. Classically considered not part of the photosynthetic active radiation. But there is also quite some evidence now in literature that it also has a role in photosynthesis. Nevertheless, I think apart from photosynthesis, this far red light play a role in many processes in the plant. The most important, what I think, is the effect on the elongation of the stem and expansion of the leaf area. So what do we see here on the picture? We see young tomato plants grown under red and blue LED, so probably the color of these this cord. And then we added far red at different intensities. So you can see the intensities of the micromoles of far red. Going from left to right, you see taller plants, so more elongated plants on the far red, more expansion of the leaf area. And this is what we see in many crops, strong effect of this expansion. And in particular in young plants, this expansion of the leaf area is very important, because that makes that there is more of the light intercepted. And that is important because only the light is intercepted, or if I'm more precise, the light that is absorbed by the leaves contributes to growth. So if we want to optimize the vertical farming, we need to ensure that all the photons are being absorbed by the plant. Therefore, in most cases in young plants, when leaf area is limited, far-red can give quite a boost to the growth. However, we do not want always those elongated plants. So there we also have to balance on what type of plant form do we want. There's one more interesting thing to see in this graph, that's at the completely right-hand side of the picture, where it says end of the day. Well, in fact, a better term should have been start of the night. That is the moment that plants are very sensitive to far-red. It's not surprising that they are sensitive at that moment, because that's in fact what happens in nature. If you have the sunset, sun is going down, think of it, sun's going down, it's getting darker and darker, so you see a shift towards higher wavelengths. So then you get relatively a lot of far-red. That's probably why plants are at that moment in particular sensitive to it. So if you see here the end of the day, where it's only for 15 minutes, well, often 10 micromole is already more than sufficient to have an effect. So short duration of low intensity far-red gives already quite an elongation of the plant if you compare the one completely right-hand side towards the completely left of the plant. So this also indicates more and more that we have to make use, or that we could make use of the sensitivity which changes throughout the 24-hour cycle. This is the same experiment, but then a little bit later, same type of treatments. Again, what you can see is with far-red, quite a big stimulation of growth. When you are a grower, well, I'm not sure what would be preferred, because then not always the very elongated plants are desired by growers. This is about young plants, and despite there is a lot of genetic variation, I think these effects in young plants are relatively general, and also relatively, well, I would say classical knowledge. If we go now to tomato plants in the fruiting stage, we can also see here that with far-red added to red-blue LEDs increased the fruit dry mass. And then the question is, where does this growth increase come from? Is it the photosynthesis? Is it the leaf area? And we did do here a complete yield component analysis, and I will not go here into all the details, but what we did do here, an experiment, red-blue light versus red-blue with additional far-red. We got an increase in the fruit dry mass of 45%. Well, then it can be because the plant grew more, or because the plant invests more in the fruits. And yes, we got a bit increase in total growth of the plant by 6%, but the most important one is the dry matter partitioning, so the fraction to the fruits. So what we see now with far-red, we can manipulate or control the balance between vegetative growth and generative growth. So in this case, the major effect is on that. And then we also have to realize this is something that growers are continuously trying to control, this balance. And now, in fact, with far-red light, we give them an extra tool. And it is not that always you need to maximize it, because it needs to be balanced. You do not need, of course, you would like to have only leaves, only fruits, but to some extent you also need leaves. So use it as an extra button to control the balance between vegetative and generative growth. Then we did do some further studies. Why is then this assimilate partitioning affected by the far-red light? And it is really in the fruit itself, the fruit sink strength, so the capacity of the fruit to attract assimilates. So it affects the partitioning of assimilates in the plant between vegetative, generative, well, and it also affects the shoot-root ratio. That's already known for a bit longer while. Here you see an example of lettuce plants grown without far-red light, so that's the blue line, or with far-red, and then with far-red you get a higher shoot-root ratio, so relatively more shoot compared to the root. So far-red light, well, I could probably continue for at least an hour with all examples, so these are just a few general examples. Blue light, of course, also very important. This is an experiment indoor where we used as a background a solar spectrum, a plasma lamp which more or less mimicked the solar spectrum. And then we replaced part of the white light by blue LEDs. So where it says here, with the plant's percentage is blue, 0 to 50%, that is the percentage of the blue LEDs. So where it says 0% blue, I have to correct it. It is a 0% blue LED, but please realize there is white light. White light with a spectrum mimicking solar radiation means it contains already 25 to 30% blue. If it's in the picture where it is 50% blue, that means 50% of the white light replaced by blue. Well, then what we see here, the general response to blue, or what we often find in the experiments, with more blue, you get a more compact plant. Often photosynthesis at the leaf level is high, but the total growth might be reduced. This is important where to find where is the optimum, because if there is no blue light, that's usually not the best. So in many cases, there is an optimum somewhere. Well, if it is completely indoor, 10%, sometimes 20% of blue light, optimal for growth in many species. But on a number of crops, we want to grow them in a very compact way. So blue also opens opportunities to create more compact plants. So I said, well, it is about light, intensity, it is spectrum, it's also the photoperiod. And here we have an example of tomato. Tomato, usually considered as a crop which is day neutral. And ideally in a vertical farm, when we have made all those investments in the whole system and in the lamps, ideally we would like to use them 24 hours a day. So can we use the lamps for 24 hours a day? That would be ideal if the plant could make optimal use of it. What we see here, tomato, I must say this is an experiment in a greenhouse. We grew plants at 12, 15 or 18 hours of light, always the same intensity, so the longer the day length, the larger the light sum or the cumulative amount of light in the greenhouse. So the lower point, I'm not sure if people can see, so this one, that's with 12 hours of light. If we then increase it to 15 hours, it means the total amount of light increased, the yield increased, the fruit yield increased by 20%. If we then do a similar step, but now from 15 to 18 hours, the yield also increased. So what we can learn from it, going from 15 hours to 18 hours and therefore more light, gives a yield improvement. However, that yield improvement is definitely less than going from 12 to 15 hours. So if you want to make maximum use of the light, in fact, we better go to a shorter day length. What is the economic optimal? That's a different question, but from a sustainability perspective, it would be better to have a bit shorter day. Then there is one other treatment that's so the 15 hours at constant light intensity, and then we had another treatment, 18 hours with a little bit lower light intensity, such that the total cumulative amount of light, so the daily light integral, in fact, is the same. So this confirms it further. The same amount of light, if you distribute it over 18 hours, that's less optimal for a tomato. If you would go to 24 hours of light in tomato, growth is completely disturbed. In fact, it is also known which gene is involved in it. But this gene is not in the commercial varieties at this moment. So this would be nice if this gene would be introduced in commercial varieties, and then that we could make use of the light for a longer period of the day. This is for tomato. And this also holds for a number of other species, but definitely not for all species. And here we see another example of different day lengths. So what was done here? 16 hours, 20 hours, 24 hours of light. In the article there are a few more species, but what I show here is the rocket. So on the left-hand side, or left is for you, that side. So the rocket, and what we see there, on the x-axis, the DLI, daily light interval, so the total amount of light per day, but the 3 data points of daily light interval, so the differences are created by the different photo periods. And what we see here, going from 16 to 20 to 24-hour light, nicely resulted in an increase in both the fresh weight and the dry weight in rocket. So obviously that grows quite well under 24 hours of light. Let's now look at the other graph, the right-hand side, the lattice. And then we see something very remarkable. The dry weight increased. So if you look, and that's what we usually, as scientists, we often look at the dry weight. We see a nice increase with an increase in daily light integral. But look at the fresh weight. That is dropping. So the dry matter content of the plants, in fact, increased. Well, then we could also think about what would that mean for the quality. And yeah, for a grower, in particular, he sells a fresh weight. So the kilograms is being reduced with the long day. So far, I gave a few examples of growth. In the end, the quality is at least as important, and probably even more important than the kilograms. Here an example of tomato fruits. Tomato, if there is, then the fruits are growing on a plant, and we increase the light intensity at the level of the fruits, we can increase the vitamin C concentration. So this could be due in fruits, but also in a number of other crops where we see that the vitamin C concentration can be affected by the light. I will show later one more example. It's also about the spectrum of the light. Here there are a lot of data in this scheme, so I did not intend that you realize all the details, but just as an example, what we see, you can do experiments with many different colors, and then you can measure many different compounds, which might all be relevant for quality or for nutritional value. Often there are effects of the spectrum. It's not always that one color is the best for all the metabolites. Often we see that blue light does do an upregulation of many metabolites. So we have seen that now in many experiments, many species. However, I should also make a warning. We also did do quite a number of experiments changing the ratio of blue to other colors where we did not find an effect. And to be honest, I don't know why sometimes in some species and some compounds it has an effect and others not. And I think it has to do with the species, but I also think it has to do with interaction with other factors. Another example of quality is here with lettuce. They were grown at different light intensities, but interestingly, we only did do that during the last 6 days of the growth. And that's also an idea in general that we think, well, initially, choose conditions which are maximum for growth, and then the last 5 to 7 days, focus on the quality at harvest. So that's why we only manipulated here during the last 6 days. So the last 6 days, more light, not surprisingly, a bigger plant, looks nicer. But then it is also after harvest, and this is a picture of how it looked like 13 days after harvest, stored in darkness, and I hope it is visible. At the highest light intensity, the leaves still looked very nice, but those which were grown at only 50 micromoles started already browning. Well, then we can also look 3 weeks after harvest. I would not say that any of them look good, but at least you can see very clearly that at the higher, those grown at a higher light intensity look much better. So the shelf life was definitely prolonged by growing the crop at a higher light intensity. These are data from the same experiment. So we had 3 different light levels. The higher the light level, the higher the vitamin C concentration. Also, the higher the carbohydrate concentration. And these correlated with the increase in shelf life. So I cannot say it is a proof for a causal relationship, because strictly speaking it is only a correlation, but if you think about the roles of vitamin C and of carbohydrates, then it is quite likely that those are the reason for the increase in shelf life. So that's relevant and important for the plant itself. And apart from that, well, vitamin C, at least, well, that's a compound which is where we can officially state that it is good for human health. So also for the consumers, it is interesting to have a product with an increase in vitamin C. So far about lights, we do a lot of work on light. Very interesting and a lot to gain there. But we should never forget that we have to look at all the conditions. In fact, what Professor Essing in the beginning started with the law of Liebig, it is all the different factors. And whether one is limiting or not, at least they all are important. It doesn't make sense, for instance, to invest a lot in lighting if your CO2 is not optimized. And if you change the light intensity and you want to optimize the growth, or you want to have optimal growth, you probably need to adapt the temperature as well. So we need to look at all the factors above ground, but also below ground. Water, the nutrients, oxygen. Few words about the root zone. Usually in indoor farming we are growing soilless. It can be aeroponics, it can be directly on water, so a deep flow culture. It can be on substrates like rock wool. We have to look then indeed nutrients, water, oxygen. Also here, I think initially we should choose it to maximize growth. And maybe the last days before harvest, so let's say again the 5 or 7 days before harvest, we might think also, well, how can we maximize the quality? Can we increase some nutrients? Can we do some biofortification, selenium, iodide? Or maybe we want to decrease some nutrients like the nitrate. What crops are grown in a vertical farm? What do we see? In most cases, it's leafy vegetables, and it's leafy vegetables, and it's leafy vegetables, and it are microgreens, and it are herbs. What about the other crops? Well, strawberry is an interesting crop, and as you see now also the interest increasing a lot. Tomato, also on the wish list of many companies. So I expect that in the coming years many more crops will be grown. Feeding the world, also what I heard at the beginning of this symposium, is then vertical farming the solution? Well, I think feeding the world is also about this crop, about rice, about wheat. And just name a crop, and I'm sure I can grow it technically very well in an indoor farm. Any crop I can grow, no doubt about that. Grow them at a high yield with a good quality. Is it economic feasible? Well, I don't think that these type of crops will be or are economic feasible, and well, I don't know what it will be in 50 years, but for the coming 10 years, I don't think that these crops will be economic feasible. But it's then very interesting what we could do with these crops. And I know there was also a very interesting article last year of Professor Essing showing on how high this yield of wheat can be. So it's very interesting to see what is the potential. But as I said, I think if it comes about economy, vertical farming at this moment is in particular for fresh products of a high and products which have a relatively high quality. And that is, I think, needed to make it economic feasible. Besides that, I think there's also a lot of opportunities for companies propagating young plants. And for crops with a very high value, crops which you see here, medicinal cannabis, artemisia, so crops for medicinal purposes, pharmaceutical purposes, cosmetics. Because what is so important in these crops, then it is important that the content of the metabolites is at a specified level, that we are sure what those metabolites are. To do so, we need to control the conditions very well. And that's in fact what we can do in a vertical farm. So yeah, an example of medicinal cannabis, different light levels, you can create completely different plants. So it affects the growth, it affects the floral yield, and it may affect also the content of the metabolites. We should also think about what cultivars are we growing in a vertical farm. At this moment, there are no cultivars which have been specifically bred for vertical farming. I'm sure that many companies are working on breeding for vertical farming, but usually it takes a number of years before they are there on the market. Yeah, what type of cultivar would be ideal? Well, probably a phenotype which is very dwarf, high rate of photosynthesis, a high harvest index. The roots, well, maybe if we are sure that we supply the water and the nutrients in a very good way, probably we don't need so many roots, so we don't need to breed so much for that. Do we need to breed for disease resilience, which is most important in general in breeding? Well, if you keep the diseases out, then that's not so important. So we can focus on other aspects, and for sure focus on quality. And we also have to realize there's a lot of genetic variation. This is an example of tomato. We grew many genotypes and the different spectra, different colors. This is just an example of 6 genotypes grown at 4 different light spectra. It's, for this example, not so important what the exact effects are, but what you can see, the different bars representing the different colors. Not all the genotypes respond the same. And this is important to realize. In these systems, well, we get more and more sensors are becoming available, not only the sensors of the environment, but in particular sensors of the plants. We can collect a lot of data. Well, then there is now the opportunity to combine all that data using algorithms, using mechanistic modeling, using artificial intelligence. In my viewpoint, we need to combine all those different things, and then we can nicely control the crop. And then we are going towards an autonomous control, where I think there can be a control room, where we collect all the data, can control the conditions in the vertical farm. So where I said of local production, I can imagine a local production, but maybe one control room which controls a number of farms all over the world. Last part, I will just briefly mention a few things about sustainability, but I think later on this day there will be more dedicated talks about that. So if we apply the ultimate level of hygiene and some disinfection, still also very good monitoring that the first time something happens that we can take immediate action, we can do it without pesticides. The nutrient use is very limited. We only need to use the nutrients that are taken up by the plants. Very little water use. But we also need to be fair about the data, because often I hear 90% improvement. Yeah, compared to what? Here are a few examples. For tomato, if you would grow them in a very good way, then probably you can go as low as 60 liters of water per kilogram of tomato. If you grow tomato in the Netherlands in a modern greenhouse, so on average growers use about 17 liters of water per kilogram. In a vertical farm, I think we can go down to 2 to 4 liters of water. But then what we need to do is, of course, what we need to do is to regain the transpired water of the plant. Regaining a water vapor is technically possible, and then you can go down to 2 to 4 liters. And in many places on the world, this is very important. Extremely limited land use. And I also get often questions, what about biodiversity? How is that in a vertical farm? No, there is no biodiversity in a vertical farm. Sorry for that. And still, I dare to claim that the best, if we want to improve biodiversity, the vertical farming is the best solution. And why is that? If we don't need much land area for agriculture, we can give it back to nature. In the end, it is then a political choice, but if you would give that land back to nature, then it really contributes to biodiversity. If we grow the crops near the consumers, less transport, and if there is less transport needed, and we produce crops with a longer shelf life, we can have also less food waste. But if it comes about sustainability, so many criteria are doing very well. Energy use, that's where we have to work on. A few other examples, advantages is that we can create products with a higher added value. A better quality, but in particular guarantees on a defined quality, defined quantity. Food safety, healthy, all very important. And then having a marketing concept that fits with what we are growing. So then I come to the end of my talk. So a few conclusions. Vertical farm, if I look at it, it's an excellent system for a full control of the production and the quality. Growing at high rates, high quality, defined quality, we can do it anywhere. It is possible now, but we also have to realize it's still very new and a lot of improvements can be made, but also have to be made to make it really economic work. Sustainability, on most aspects, very good. Energy use, that's what we need to work on. And we also need to reduce the cost price. And then I would like to thank you for your attention.
Thank you very much. That was also understandable for a non-expert like me. Thank you very much. We have also received some questions. Thank you for that. The first question is, effect of blue light maybe depends on species. Has the role of microorganisms been studied yet? After all, microorganisms are strongly associated with plants.
Yeah, I also get often the question of that a vertical farm is sterile. No, of course it is not sterile. I did not do work myself on a microorganism or biostimulants. I think there could be a role. On the other hand, I think biostimulants, microorganisms in particular, they can have beneficial effects if plants are grown under stress. And here we try not to stress, in most cases, the plants. So I think the benefits are smaller than under stressful conditions, but also here there could be advantages, but as I indicated, I did not study that myself.
Thank you. The next question is, what is the current ratio of food energy output per kilowatt hour of energy invested in VF? How do the input-output values compare to other forms of agriculture?
So I had some different, because I hear a lot of...
I know it's really hard here, right? I showed the question to you. What is the current ratio of food energy output per kilowatt?
Oh, okay, so wow. Yeah, that is... So first of all, we have to look at what is the efficacy of the lamps? Well, then I guess probably the most modern LEDs, they can probably go up, according to some companies, to about 3.5 micromole per joule electricity. But then for producing the food, we have to make the ratio of kilogram food per joule. I can calculate, but not by heart, sorry.
Okay. What do you think about the trend that many luminaire manufacturers offer more and more luminaires with just white LED?
I think that's a lot of research is ongoing at this moment. What is the best spectrum? So I can't give a simple answer. What we have to look, and we should think about the plant. I don't dare to say what is exactly the best. When working in it for the human, it's often easier if there is a little bit of white light. Let's say if you have only red or only blue or red-blue light, it's less nice working than when there is a little bit of white. And I would say, I guess you will see in the coming years a lot of publications on comparing the different spectra, also with white, but also if you're talking about white, there are many different forms of white light. Maybe our eyes see it as white, but it can be different type of mixes of colors. That also makes it so difficult to answer the question.
Okay. Where do you measure the light output? Because the plant is growing to the light.
What we usually do in our experiments, then we define the light intensity at the top of the canopy. So let's say at apex height. That is what we typically do in our experiments. But this is a very good question, because in in most systems where we grow them, the light intensity drops with the height of the plant, and that's indeed a complicating factor. Even measuring the light at the top of the crop, you could debate if that is the best. I think it is the best what we can do at this moment. So I would take that always as a comparison.
So we also have further questions, but you're also participating in the panel discussion later, right?
Yes.
So then we will discuss the other questions. Thank you very much. So that already worked out pretty well. Thank you for having so many questions and also for using the Slido tool to ask your questions. As we've said, we will have another panel discussion at the end where we will discuss also the other questions. But now we will switch to insects because we've heard it already that plants need light to grow, and we want to dive deeper and learn how to optimize plant performances. And we will hand over to Professor Dr. Tracy Lawson from the University of Essex and the speech, The Effect of Light Quality and Intensity on Photosynthesis Synthetic Processes. Please go ahead. Thank you.
Hi, everyone. I hope you can hear me okay. Presume so. So good morning, and I'd first of all like to thank the organisers for inviting me to talk today about some of the research that we've been doing here in Essex. It's really sad that I can't join you in person. I would have liked to, but hopefully sometime in the future. Okay, so just a bit of background to what I want to talk about today. First of all, I'll give a bit of background to photosynthesis and light use, and some of the previous work that we've done sort of more at a mechanistic understanding of how light influences photosynthetic processes, but then how we might take some of this knowledge and transfer that into use in vertical farming. And in particular, I want to look at the impact of light regimes on plant growth and a little bit on energy use, and that's something that we're sort of starting to get more and more involved with. I want to then move on and look at some of the work that we've been doing with far red light, which Leo has just really nicely introduced, so I can skip some of my introduction on that. And then some really exciting work that we've been doing where we're using one of our monitoring tools for plant processes and using this as a possible feedback system so that the plant sort of tells the system what light is required at what times of day, and that might help us to optimize various different light protocols. So just a brief introduction to photosynthesis, and not in any detail, but that photosynthesis is broken down into two key components, what we call the light reactions and the carbon reactions. And ultimately, the light reactions that take place in the thylakoid membranes, this is all inside the chloroplast, it's inside the mesophyll cells in plants. So we have the chloroplasts that are made up of the two photosystems, photosystem 2 and photosystem 1. And those are responsible for capturing that light energy using the light harvesting antenna. That energy is then funneled down through the electron transport chain, ultimately producing NADPH and ATP that then feed into the Calvin cycle reactions that take place in the stroma where CO2 is fixed. And both sides of this or both components of this photosynthetic system, and use those as monitoring tools that I'm going to talk about a little bit later in my talk. So we use infrared gas analysis to look at CO2 uptake or carbon assimilation, measured in micromoles per meter squared per second. And we also use chlorophyll fluorescence as a tool that measures how well light energy is transferred in the electron transport chain. So this is either electron transport or in photosynthetic efficiency. And these are two terms that I'll talk about in terms of measuring photosynthesis. And just a quick overview of the electron transport chain, because I'm going to come back to some of this a little bit later on when we look at some of the properties of the spectra of light. The electron transport chain, as I'm sure most people are aware, is made up of these two photosystems, photosystem 2, photosystem 1, that are sort of integral membrane proteins in this thylakoid along with the B6F complex. And light energy is captured by these two photosystems. That light energy is funneled in to a special chlorophyll molecule here in the reaction center of photosystem 2, and this is where the electron transport takes place and the charge separation takes place. It leaves the special chlorophyll molecule here known as P680, and that's because it preferentially absorbs light. It's 680 nanometers. The electron leaves and heads down through the electron transport chain, where it's given us another boost of energy here at photosystem 1, and eventually makes the reductant NADPH. And the special chlorophyll molecule here is P700, it's named P700, and that's because it preferentially absorbs light at a slightly longer wavelength. And I'm just mentioning this because I'm going to bring this back when we come to look at the far red light and how that influences some of the photosynthetic processes. At the same time that electron transport has taken place here, this linear electron flow here, also protons are dragged across this thylakoid membrane, and these protons in the lumen space eventually build up and they make the ATP and the energy for the carbon cycle. I'm mentioning again this buildup of protons because it's also linked to a process called non-photochemical quenching, where there's conformational changes in the antenna system, specifically at photosystem 2, where that light energy is dissipated as heat rather than used in electron transport. And again, I'm going to come back to this in a little while, so just a bit of an introduction to that. So if we look at the light properties, and Leo's already mentioned this about when we look at the electromagnetic spectrum and we look at what plants really want or require, we sort of talk about this visible spectra, the same sort of light that we see, between 400 and 700 nanometers. And that's traditionally been known as the photosynthetic active radiation, the PAR. And actually our light sensors that we measure tend to measure between those wavelengths, 400 and 700 nanometers. But there's potential opportunities outside these regions that Leo's already mentioned. And so we maybe need to think about extending our PAR spectra, not only in relation to how that might manipulate processes of the plant that Leo mentioned, but also how it might influence photosynthesis. And I'm going to come back to that a little bit later as well. When we talk about absorption and looking at the pigments that's involved in photosynthesis and the absorption of those light for photosynthetic processes, we tend to think about the chlorophylls, A and B here, in the green here, and they have an absorption spectra that's specifically in the blue and the red regions, and then carotenoids can extend this or help to dissipate energy that's not needed. So this is why there's often this thought that only blue and red light are useful for photosynthesis, and that's true in a way of the absorption of those chlorophyll molecules. However, it's worth remembering that when we look at an intact leaf, so these are the pigments extracted from the leaf, when we look at an intact leaf, it's a much more homogeneous pattern that we see. So this is an example of absorption at the leaf, at a full leaf level, and you can see that although we do have some dips here in this sort of green yellow region of absorption, there still is absorption of that light. And this comes to looking at spectral qualities of light, and that we're doing some work at the moment looking at green light, and it's thought that green light can contribute significantly to photosynthesis as well as other processes such as stomatal behavior, etc., which I don't have time to go into today. But it's just worth remembering that at a leaf level, those absorption properties are much more different to what you traditionally see for pigments. And that's of course because the leaf has a structure. As that light comes down through the leaf, there's scattering of those light properties. The path length of the light is lengthened, and therefore there's more potential for absorbing that light energy. And that depends on the species, the growth conditions, and the structure of that leaf. And so if we look at how plants use light energy, we've got a typical light response curve, and I'm going to use several of these throughout my talk. So we've got light intensity on the x-axis, and this is usually measured in that PAR region, that 400 to 700 nanometer region, against photosynthetic rate, so CO2 assimilation rate from here. And this dotted line represents absorbed light. So as the light intensity increases, the absorbed light for the plant will increase. However, plants can't always use all of that light energy. And so there'll be a proportion of it at the lower light levels where there's a linear relationship between absorbed light and photosynthesis. But there will become a point where the light is saturated, and the light saturated rate of photosynthesis is reached. And any increase in light will not lead to an increase in photosynthesis beyond this point. And this is when we get these processes kicking in, like non-photochemical quenching. So there's a conformational change in the antenna that changes how the light is funneled into photosystem 2, and basically it's dissipated as heat rather than send that light energy into photosystem 2. If you do send too much light into photosystem 2, you can end up causing what we call photoinhibition, and you start to decrease photosynthesis. And that can be a long-term stress or damage, which can last hours or even days to recover. So that could greatly reduce productivity at these times. And it's worth just remembering that the light saturated rate of photosynthesis, or how much plants of light can take, depends on the species, of course, but also its previous growing conditions and previous history of that plant. So it's worth knowing some of these facts to try and optimize light use in such systems. So I just want to show you some of the variation. We're doing a project at the moment looking at densely grown salads, and sorry, I'm unable to move to the next slide. Sorry. So the this is a EU project where we're looking at optimal properties for growing salads densely in hydroponic systems. And this is just some examples when we looked at photosynthetic capacity in various different of our salad types. So we've got rocket, wild rocket, and spinach here. And sorry, my systems here is the figure legend for that. What we can see immediately is that the rocket has a much lower photosynthetic capacity at 15 micromoles per meter squared per second, compared with the wild rocket, and spinach is somewhere in between. So that's quite a large range of differences in that photosynthetic capacity. And also you can see that the spinach saturates at a much lower light level than, for example, the wild rocket, which is still able to cope with light levels right up to 1,000 micromoles of light. So of course there's an efficiency gain here of how much light you want to put on relative to that photosynthetic rate that you will achieve as well. So this is measuring it as the CO2 uptake. We can also measure that as the efficiency of light going through photosystem 2. And that gives us a really good and easy measurement of measuring photosynthetic rates. And I'm going to use some of these later. And again, we can see that the wild rocket sits on the top here, rocket down here, and spinach in between. Interestingly, they're very much together at the lower light levels, and then this separates out. So as the light increases, the efficiency of light use goes down because we start kicking in these processes of non-photochemical quenching and wasting light energy. Okay, so trying to optimize the right amount of light on the plant relative to what it's using could be really informative for optimizing inputs and, of course, the output of yield. And just to mention that this variation within this photosynthetic capacity, not only between species, but also between cultivars. So again, this is just some examples from our spinach varieties that we're using within this program, and you can see that there's significant variation with different types. So all plants are different, but also cultivars within that can see significant variation within those systems. And then I just briefly wanted to introduce some work that we've done in the lab where we've been looking at dynamic light regimes. And Leo mentioned about the fact that lighting regimes could be an important component in the future. And I totally agree that this is a system that could really be optimized to try and deliver not only in terms of yield and in terms of energy inputs, but maybe also in terms of secondary products, metabolites, and compounds of interest. So this was part of our sort of fundamental research that we were doing in the lab several years ago, where we wanted to see how photosynthetic processes changed under different light conditions and dynamic lighting conditions. And what we found is that, so we grew plants in either a square wave regime, here shown by the blue purple, and in a dynamic light regime, which was just recorded from the roof in Essex. We programmed that into our LED systems. And we also grew plants in a sinusoidal regime, typical of a sunrise, sunset, and that varied every single day. So it had a bit of variation within that. And this is the typical sort of value of what that looks like. There's two lines for each one because we also did a high and a low light environment. I don't want to get bogged down with that, but we know that photosynthesis acclimates to high and low light. But what was really interesting is that when we looked at photosynthesis in these plants, and these are our different light regimes here, again, it doesn't matter about the different light regimes, and the high and the low. So here the high, these are the low ones. Whether we measured them as a square wave light environment or as a fluctuating light environment here on the right, what we see is that photosynthesis is high during the first part of the day, and then there's this decrease in CO2 assimilation rate. So we see that in a square wave light environment, and we also see that in a dynamic light environment. Again, the black line here is the measured values. The red line, the dotted red line, is what was predicted. And we can see that at the beginning of the day, the plant performs as we would expect with that photosynthetic capacity that we know. As we head towards the end of the day, there starts to be a big discrepancy between what we would expect and what we measured. And there's this decrease in activity and productivity towards the end of the day. And so this led us to some of the work, and I'm just going to give you a very brief overview of some of the work that we've been doing, trying to look at optimizing some of these light regimes and light recipes based on what we've found from these photosynthetic processes. So we've looked at photoperiod and intensities, etc., and we haven't got time to tell you about some of the photoperiod work that we've been doing. But if we look at some of the light regimes, we thought, well, what do we get when we, and this is work on a Basel project where we've been under ERA funding, where we've been looking at productivity delivered by either a square wave light regime, shown by blue here, or by a sinusoidal light regime. And basically this was a very basic light regime that we set grown, and we didn't really see any changes in biomass from these plants, whether it was grown under these two light regimes. I should just point out that these light regimes deliver exactly the same photon dose, so they receive the exact same photons, just in different packages of light. We thought, should we be a bit cleverer? And if we think that we've got this increase in photosynthesis at the beginning of the day, should we skew our light regimes to have more light at the beginning of the day? So we added in a positive skewed sinusoidal light regime shown in the grey here. And again, I'm just comparing the sinusoidal with the positive skew. We saw no significant differences in the plant growth from this. However, we were using light that was in these regimes. We were using light, when we look at our light response curve and look at the saturated rate of light for these plants, we could see that PAR saturated at a much lower light level, around about 500 micromoles of light. So really, when we're given these regimes, both of them, we're losing a lot of our light energy that's way beyond what that plant could cope with based on its photosynthetic capacity. So this led us to think, well, could we cap the regimes? So this is what we started to do. So basically we chopped the top off that regime. So we give our square wave in blue that you can just see here on the left-hand side of the graph. I hope you can see my pointer. This skewed sinusoidal that then decreases as we head further through the day, and our typical sinusoidal. We didn't see any differences in our plant growth. And this is ongoing research. We're now seeing some differences in plant growth, but I can't share those data with you at the moment. But what we did see from this is that although we didn't see any increase or significant increase in plant productivity, we did find that we needed 17% less electricity to deliver the same amount of biomass. So again, we're thinking about this in terms of inputs and outputs. So we maybe didn't achieve it on the output. We've got new data that I hope can share with you in the future. But we did save some energy. So this is some ongoing work, and I'm going to come back to a little bit of this later on when I talk about the chlorophyll fluorescence feedback. Okay, another part of our work, we're doing lots of different work on light and recipes in terms of not just timing, but also what is the spectral quality that you can add. And as Leo mentioned, there's lots and lots of different opportunities for this. And we've been playing around a lot with red-blue ratios that Leo also mentioned. We're particularly interested in stomatal behavior and how that leads to evaporative cooling, but also uptake of various nutrients, etc., in stomatal behavior. And as Leo mentioned, you get lots of different changes with this red-blue light ratio. With more red, you can increase biomass, have larger plants. Blue tends to make the plants compact, but you can change anthocyanins, colorants, secondary metabolites, etc. Lots of different opportunities that we're looking at. But what I'd like to just mention today is some of the work we've been doing on the far red ratios. And I don't think I need to introduce this because Leo did a great job of doing that, it's the balance of far red in your spectra relative really to the red, because it's a phytochrome response, which is known as the shade avoidance response, where the amount of far red swaps what that response will be. So it's far red relative to the red response. And that can elicit a whole host of different changes, as Leo mentioned, and we've been looking at that in terms of leaf shape, anthocyanin production, etc. So this is a typical blue red grown rocket, and then here we've got a bit of far red added to that. We can see that changes the anthocyanins, changes the colour. We've put far red in at night time and shown different shapes of those leaves. We've put far red and blue in as nighttime breaks. These all change this phytochrome response, and there's lots and lots of different ways that you can manipulate the plant. But what we're really interested in is how that might change photosynthesis. And I mentioned that we have our PAR range that sticks between 400 and 700 nanometers, and that's typically what we think about for photosynthesis. But there's lots of work that's been done by others and ourselves as well, where if you add far red light, this is again our typical light response curve, if we add far red light to it here, we can see that we can increase that photosynthetic rates around about 4 or 5 micromoles, which is quite considerable. And it actually is like extending the white range of light by putting that far red light on. Of course, far red light LEDs are less efficient, and can have different costs involved in them. So we're trying to look at some of these ratios of how much light that should be. And just to say, the reason why this happens and why that far red light could be really important is if we go back to our photosynthetic electron transport chain here, I mentioned that the photosystem 1 preferentially absorbs light at the longer wavelengths, 700 nanometers. So what far red light does, it preferentially stimulates photosystem 1. And that does two things. It drags the electrons down through the electron transport chain to make NADPH and ultimately ATP with the protons. So it drags the electrons down through the electron transport chain, which means that it makes photosystem 2 more efficient. It takes the electrons away from that, and it also prevents light being too much on photosystem 2 and damaging it. Photosystem 2 is very delicate and easily damaged with too much light, so it sort of protects that. And it can also drive cyclic electron flow, where the sort of electrons go around here and make ATP. And all of that can be beneficial for photosynthesis and photosynthetic processes. So what we're really interested in doing, and this is going to show you some really sort of data from that we're just started to do, is trying to find out what's that sweet spot for that far red. How much far red relative to red, blue, and your other spectra is needed? Because as you increase the far red and the timing of that far red, you start to drive these morphological changes that you may or may not want. You may not want that etulated plant. Might be beneficial for harvesting, etc. But what we do want to do is also increase photosynthesis. So where is that sweet spot for growing a plant with far red in terms of photosynthetic efficiency relative to development? And so we've employed a dual class system that allows us to measure photosynthesis in both of those two photosystems. So the efficiency of both of those two photosystems. And this is just some really preliminary data here where we've got electron transfer through photosystem 1 and through photosystem 2. Photosystem 2 shown by the black dots here, photosystem 1 by the open symbols. And basically, when we put the far red light on, we can see an increase in that efficiency, which then decreases when we turn the light off. And we've also got gas exchange measurements that show a 2 or 3 micromole increase in CO2 assimilation rate with this small amount of far red light on, which I think is around about 60 micromoles in this case of far red light on that system. So we're doing this work to try and develop this a little bit further in the future. And just to say that there's lots and lots of different recipes that you can do for various different components of that Leo mentioned, and we've got a review coming out hopefully soon that is covering some of these different changes. Yeah, I just wanted to talk to you a little bit about some work that we've done looking at chlorophyll fluorescence as a real-time feedback signal. And this came about really based on some of our earlier work that I mentioned, where we were looking at these various different demands of light energy through the day and photosynthesis in our sort of typical research that we were doing in the lab. And we typically see, as I mentioned before, this is for the basil, we see again, as we go through the day, there's a decrease in assimilation rate. So this is under a square wave light regime, and basically assimilation rate peaks for the first several hours, sort of 4 or 5 hours, 6 hours of the light regime, and then this starts to decrease as you go through the day. And this is also reflected in this efficiency of PS2, that other parameter that I mentioned here, the fluorescence parameter. Sorry, it's this red one here. It's not very obvious on this graph because all of these different photosynthetic fluorescence parameters are on, but we see this decrease in efficiency through the day. Measuring chlorophyll fluorescence is much, much easier than measuring gas exchange. And so we wondered if we could use this measurement of photosynthetic efficiency as a real-time signal for the plant to actually feed back and tell the light what it wants. So we built a very simple system where we measured chlorophyll fluorescence on the leaf. We put that into a controlled feedback system that then fed back to our lighting systems. So basically we've got chlorophyll fluorescence, and that sends a signal back to our lighting regimes, influences the plant, and then that changes that. And we hope by doing this, we could somehow optimize the plant growth and performance. We spent a lot of time trying to work out what the signal should be and how they integrate through that, through this signal. And so the first few studies we can see here, we've got, again, this is basil grown under a square wave light regime, and then we have our feedback system shown here in red. And so we did simple feedback that sort of worked at the end of the day on these systems, and basically we saw very little change in the efficiency of productivity. But we did see potential in reducing the energy demands for those plants. So a 3 to 7% decline in energy by using that feedback system. So just some conclusions from some of this work. This has sort of been a whistle-stop tour of some of the activities that we've been doing here at Essex. Is that I think that we've seen that both the spectral quality and pattern could influence plant growth and performance. And I think that patterning at different times of the day could be really important and a powerful way to manipulate not only the morphology and development of the plant, but key metabolites, secondary metabolites, and various different compounds of interest, flavor profiles, flavonoids, etc. So I think this spectral quality, intensity, and time has the potential to really alter some of these flavor profiles and could be really beneficial in the future. And I really think that our chlorophyll fluorescence feedback loop has shown some real promise for improving productivity and reducing output. So less input, less energy requirements, and a greater yield at the end, which could be really promising. So I just want to acknowledge the people that have actually done this work. So the ERA-funded project on the basil was done by Jim Stevens and Ben Clemlin. And a lot of the work that I've shown from the Hyphadense project is done by John Stamford and Phil Davey. And some of the previous work in the lab was done by Jack Matthews and Sylvain Ballet-Chambron, who Sylvain is now in Wageningen and Jack's just left the lab. So thank you very much.
Thank you. Perfectly in time. So again, we received some questions. The first question is, thanks for this amazing presentation. Do you think it could be possible to talk about light recipes for different varieties along with different species?
Yes, I think absolutely. I think that by understanding some of the capacities of plants, I think we can optimize those light recipes for different types of plants. So, for example, when we're doing our spinach relative to our baby leaf, we're looking at different light recipes to optimize those, because not all plants are the same and not all of those photosynthetic processes are the same. So I think it's understanding really how the plant responds to that light and then how we can use that information to optimize those processes within a vertical farm environment or an indoor growth environment.
Thank you. Just asking if there are some questions here, because we also have a microphone. So if one of you wants to address it directly, there's also the possibility to just stand up, then I can see you. Otherwise, I continue with the questions that we have received. One more question is, you showed a probe connecting to an, now I'm struggling, Arduino for fluorescence measurement. I hope you get it. Is that correct? Could you share what kind of sensor you used?
So yes, so it was connected to an Arduino for the feedback system, and it was connected to one of OptiScience's monitoring systems. Sorry, the name is eluding me at the moment, but I can easily provide details for that. So it was an off-the-shelf monitoring fluorimeter that we took the voltage outputs from that, put that into our Arduino for a feedback loop, and then sent that to a lighting system. And actually we built our own lighting system originally, but now that will also feed into some commercial lighting systems. And we're busy writing this up, so I hope that we can publish some of this as well soon with all the information in it.
Thank you. Professor Asseng also has a question.
Hello. Thanks, Tracy, for the very interesting presentation. You showed that the photosynthetic rate declines over 16 hours light. What happens if you continue to 24 hours and keep doing this with the rate, with the photosynthetic rate?
Yeah, that's a really good question. That's some of the work that we're doing at the moment that I didn't really have time to share with you. We've been looking at those photoperiods and how we can see how that works. It tends to just keep declining, not always at the same rate, but we tend to get a decline. And so what we're trying to do now is do shorter periods of light. And of course, you know, the beauty of the vertical environment and the indoor environment is that we're actually not constrained by a 24-hour day. The day can be whatever we want. So we're busy trying to do some of that work now. It's ongoing in the lab, and hopefully in the future we can share some of that with you as well.
Perfect. Thank you very much. Maybe one more question. Is pulsed light an option?
Sorry, is
Sure. I will say it again. Is pulsed light an option?
In terms of sort of making a dynamic regime, in terms of pulse and light in dynamics?
Maybe.
I think
Sorry, I just received the short question, so I'm not sure. But we also have another question. Is it okay? Yeah, we have a microphone here. There will be a clarification in a few seconds. Microphone is on the way. Thank you very much.
The question is pulsation of the light source at a very high frequency an option to save energy?
Yes, I think that could be an option, and I think some systems are already sort of looking at that. We've monitored some of that because, but not for that reason. We monitored some of it for our chlorofluorescence feedback because that system works on a modulated system, and so you have to be careful of where them two systems overlap each other with lights. So there is some sort of pulsing within the lights anyhow from it. So I think it could be an option, but probably somebody else could maybe answer that better than me for knowing what that could deliver from it.
Okay, thank you. Then we will continue with the next speaker. Thank you very much for sharing all your research results with us. Thank you. And we will continue with the effects of light, as our next speaker will also share his research findings about how light properties regulate yield and quality of plants grown in vertical farms. He's already here on stage. Please welcome Professor Dr. Eric Runkel from the Michigan State University. Thank you.
Okay, thank you. Good morning, everyone here at least. Thank you for the opportunity to be here, and I want to thank the conveners for organizing this and including me in this impressive lineup of speakers. So my focus in the next half hour is to talk about light and how the different properties influence yield, which is often of most interest, but also some of the quality attributes. And if there's going to be a take-home message, I think it's that there are often trade-offs, that it's difficult to find a light spectrum that will both optimize yield, but also optimize some of the quality attributes such as leaf coloration or perhaps nutrient density. First, I want to acknowledge a current and former graduate student of mine who have done the research that I'm going to talk about, Nathan Kelly and Ching-Woo Meng, as well as companies that have supported our research, or in the case of USDA NIFA, that's an entity in the United States that is supporting some of our indoor farming research. Okay, I also want to mention this project I am involved in. I'm directing a group of colleagues at Ohio State, Arizona, and Purdue, and our focus is on optimizing the aerial environment, so things such as lighting, temperature, humidity, etc., for indoor production of leafy greens. I've done work on other crops, including ornamentals, but I'm going to focus on leafy greens today. And so if you're interested in learning more information about this project, the website there is at the top, scri-optimia.org. Very briefly, this is an overview of my presentation. Briefly give an introduction, the general objectives of our research. I'll talk a little bit about light quantity and how it's delivered and how it affects quality and growth of lettuce. I'll show an experiment that looked at the effects of blue, green, and far red light on these different parameters, and then a follow-up experiment with that to better understand how blue and green light are regulating growth and how that might have influenced consumer preferences. And if time permits, I'll give a short introduction to some recent research we're doing where we're delivering different light colors at the end of production in an attempt to increase the quality. I can go through this fairly quickly because Leo gave a nice introduction about light already. When we talk about light quantity, we're referring to the intensity, or scientifically we think of it as the photosynthetic photon flux, or flux density. This is the primary attribute of lighting that influences the biomass, or the accumulation of leaves and stems. We think about the spectrum, or the photon spectrum, as the color of the light, the spectral distribution of the light, and that influences the morphology of a plant. For example, how big are the leaves? And then the duration of light, how many hours of light are delivered during the day and the night. And at least for ornamental crops, many of them at least, it can regulate the flowering process. Of course, it's not this simple, and I think what's exciting is we learn about how these different parameters interact with each other. And I think you'll see that's the subject of a lot of our research today, is to dissect how these different interactions influence yield and quality. So indoor farming is actually not that new. It's been done for several decades in Japan in particular. This is a slide showing Japan probably 15 years ago, where they were growing lettuce indoors, in this case under high-pressure sodium lighting. Probably more common to that was growing different plants under fluorescent lights. But of course, what's been changing and what has driven the development of this sector is the advances of LED lighting. And so this is a photograph taken about 10 years ago at a small indoor farm in Michigan, and they were in the midst of changing over from a conventional fixture, in this case, to a red and blue LED. The sector is advancing very quickly. The efficiencies continue to increase and the costs come down. And we've been doing most of our light quality research at Michigan State in indoor farms in the Controlled Environment Lighting Lab at Michigan State. And this is a lab that I developed in partnership with Osram. They developed the lighting fixtures, and we have the ability to deliver 24 different light environments at any one time at potentially at two different temperatures. So our general experimental objectives have been to consistently and rapidly produce leafy greens, considering a variety of attributes and, of course, yield. So what's the fresh mass that usually drives grower decisions, trying to optimize yield, but then looking at how light affects some of the quality attributes, such as the size or the shape or the color of the leaf. You can imagine it can influence the texture of the leaf and therefore the crunchiness or the softness of the leaf. Certainly it can influence nutrition, and these things can influence consumer preferences. And so we've done a little bit of work in that, and it's an area that we want to pursue, as well as Leo showed some nice slides looking at the effects of the light environment on the post-harvest longevity. So the first experiment I want to talk about is to understand a little bit more how we deliver light and try to deliver a target daily light integral. For those of you who are not familiar with the daily light integral, or DLI, that refers to basically the accumulated amount of photosynthetic light that is received per day in a square meter. And so the unit is moles per square meter per day. And the objective of this study was to look at whether growth of lettuce is similar when the DLI is the same, but the photo period or the intensity to achieve that was different. So we wanted to quantify growth at a range of DLIs and then have different photo periods and light intensities to achieve those DLIs. In this case, in all of our studies, it's important that we control the environment so that when we have treatment responses, we can have confidence that the effects are specifically to the treatments. So we had photo periods of 16, 20, or 24 hours, photosynthetic photon flux densities ranging from 120 to 270 micromoles, and those yielded DLIs ranging from about 7 to about 16 moles per day. For reference, I think many people consider the maximum DLI for a variety of lettuces to be around 17 or 18 moles per day, and beyond that, at least some cultivars become very susceptible to tip burn, and so commercially you rarely see light integrals beyond about 18 moles per day for most lettuce cultivars. Constant temperature of 22 degrees C. We have ambient CO2, so we are measuring it but not controlling it. And in this study, the light integrals were delivered by 50% warm white LEDs and 50% from red LEDs with a peak wavelength of 660 nanometers. We harvested plants 27 or 28 days after seed sow, depending on the cultivar. And most of our studies, that's a typical time we harvest them. They're typically small heads, but it's a common harvest time for commercial indoor production in the United States. Here's a graph with a fair amount of data showing the results. So we grew a green leaf lettuce Rex and a red leaf lettuce Rouchy. I'm not French, but I understand it's a French cultivar. We quantified leaf width and leaf number upon the y-axis and daily light integral on the x-axis. And you can see similar trends for both cultivars. Relatively good response where you saw for leaf width, it generally increased slightly as you increased daily light integral for both cultivars. And also we saw leaf number actually increased. And at least part of that could possibly be influenced by a slightly warmer plant temperature as we delivered progressively more light. So we saw wider leaves and more leaves as we increased the daily light integral. We also saw here that generally the way that we delivered the DLI had not too much effect on these two different parameters, although there are places where you might be able to pick apart a few treatment differences. But probably what is of more interest is how did this affect the fresh mass and the dry mass? So again, we saw very consistent effects, linear effects, where we increase DLI, we increase fresh mass of both cultivars, as well as dry mass. But what I want to point out is that at some point the light becomes relatively high for lettuce. Light intensity of 300 micromoles might be considered a high light intensity for lettuce. And so when we're looking at delivering close to that, 270 micromoles, which is in the red diamonds on the right, and compare that response. So we have 270 micromoles for 16 hours per day versus the lower light intensity of 180 micromoles, but delivered constantly. We begin to see some of these differences where the effects of the light or lighting becomes more efficient, or you have more biomass when you're delivering a lower intensity light for a longer period of time. So the 270 micromoles for 16 hours per day produced less fresh and dry biomass than the longer photo period at a lower light intensity. So we can conclude, and not surprisingly, there have been a lot of studies that show that increasing the daily light integral increases the fresh weight and the dry weight, the leaf width and the leaf number. We also measured other things where we saw an increase in chlorophyll concentration, which led to a darker green leaf or more of a purple leaf in the case of red leaf lettuce. But at the highest daily light integral tested, the lower photosynthetic photon flux density delivered for a longer time had more mass than higher PPFD delivered for a short time. So now I want to go into light quality, or the photon spectrum, and this has been probably the major focus of our research over the years, is to try to dissect how these different colors influence some of the growth and quality attributes of lettuce, and sometimes we include other leafy greens such as kale. In this experiment, we wanted to understand how substituting partially or completely blue light with green and or far red light would influence growth of these different greens grown in an indoor farming setting. And so you can see some photos here of the different types of spectral environments in the different treatments. So to walk you through the different treatments in this study, we started with 120 micromoles of red and 60 micromoles of blue. And so we have a total photon flux density of 180 micromoles. And I use that term because when we're including far red light, of course, far red light is not free. There are many cases where we want to include those photons, and so to keep treatments similar, at 180, we are quantifying TPFD and not PPFD. So we had 3 treatments where we partly or completely replace the blue light with green light. So we have all the way up to no blue light with 60 micromoles of green, and the rest is of red. So you can see we kept red constant. Another 3 treatments where we replace some of the blue or all of the blue with far red light. And then we had 3 other treatments where we had partial substitution of blue with green and far red. We had a control of a warm white LED, as well as a treatment that delivered more of a greenish light. It's delivered from a mint white or equalized white LED, which, as you can see, is rich. About 60% of the emission is in the green region. So we're keeping the other environmental factors the same. And what I'd like to do is show you a video of plant growth that was started when the treatments began, of these 4 different treatments in the square, which kind of represent the extreme differences among the treatments. So the plants are starting out the same, and within a couple days you can already see the effects of the light spectrum on the morphology of the leaves. So in the upper left you have the red and blue spectrum, which, as you would probably expect, you get very compact growth. Contrast that with the red and green spectrum, or the red and far red spectrum, where you have no blue light and you're getting much more dramatic leaf expansion. And then the warm white, which has a relatively low intensity of blue, you still see more elongation than the higher blue intensity, but not as much as the red and green or the red and far red. Then we can look at these plants. When we take photographs, we try to pick the most representative plant. It's not foolproof, but we pick a plant for representation. And we can see the final effects on the coloration and the morphology of the plants. The red and blue, we get a relatively compact but relatively dark red leaf. As we replace some of the blue with green, we have increase in elongation, but also a decrease in coloration, and similar effects when we replace some of the blue with the far red. And then the 3 treatments there on the bottom, we have somewhat of an intermediate response, probably because we have at least a low to moderate intensity of blue light in all of those treatments. Let's look at some of the data. In this case, we plotted it based upon the blue photon flux density, or the intensity of the blue light, because this proved to be a good way to describe the different treatment responses. So here is data for Rex cultivar of lettuce, the greenleaf variety, as well as a kale variety that we included in the study. So in this case, the solid lines refer to the light that was partly replaced by green, and the dashed line refer to light in which blue was partly replaced by far red. So here we see that leaf length and leaf area showed similar trends for both varieties, where as we increased blue, we got smaller leaves or shorter leaves, and that including far red in a spectrum increased leaf expansion more than including green light in a spectrum. Let's now look at shoot fresh mass. So again, somewhat similar responses, where as we increase the blue photon flux density, we actually saw a small decrease in the shoot fresh mass for both cultivars. And probably not surprisingly, when we replaced some of the blue with green, that had more fresh mass than when we replaced some of the blue with far red. So far red still contributed to photosynthesis, but including green, we tended to see a little bit more harvestable yield. Okay, so what was happening in that last set of experiments, in the last set of treatments, is that when we were changing blue, we were also changing green. And so it was really hard to know, were the effects because we were decreasing blue, or were the effects because we were increasing green? And I think green has been probably the least understood waveband for indoor lighting. I think we actually know more about far red than we do about green light. And so we designed an experiment to look at the effects of whether the increased growth that we saw in the previous study was caused by increasing green, or if it was caused by the decreasing blue radiation. So we had a set of treatments here where we had only red, and we replaced part of that with blue light to the point where we had very high levels of blue of 100 micromoles of blue with 80 micromoles of red. And then we had similar types of treatments where we had blue the same as the treatments above them, but we were substituting some of the red with green. So we could compare at a certain blue intensity different combinations of red and green. We had an additional treatment of warm white lighting. It's kind of our control treatment. And in this study, we also grew plants in a greenhouse. So obviously, a greenhouse environment is going to be very different, but we delivered a relatively similar daily light integral, and plants were grown at a similar temperature. Of course, light was from the sun, and you're going to have a lot more fluctuation in the light environment than you will in a controlled environment like an indoor farm. But all study is done here with a 20-hour photo period at 22 degrees centigrade. Okay, again, looking at the data, we thought it best to plot it based upon the blue photon flux density. So we have shoot fresh mass and dry mass on the top, and leaf length and leaf width on the bottom. So on the top, we have the dashed lines that represent treatments without green. So it was basically combinations of red and blue. And then the open circles with the, did I say that wrong? I meant the dashed line is with green. The solid line is without green. And so similar to the previous study, we saw that as you increase the blue photon flux density, you're beginning to decrease the shoot fresh mass. And that's at least partly because we're seeing that the leaves are a little bit smaller. Regardless of whether we are substituting some of the red with green or not, we see that the leaf lengths decrease slightly as you increase the blue photon flux density. And similarly, the leaves become narrower as you increase the blue photon flux density. And then there are some comparison treatments with a warm white LED, which again has relatively low blue light intensity, and then in a greenhouse where you have a high blue environment, roughly 25% of the total photon flux density would be from the blue light. These are representative plants from the different treatments. I think it's a lot easier to digest data when you see pictures as opposed to staring at graphs. At least that's the case for me. So red light, we have so this is for the cultivar Hushi. You don't have the pigmentation. In fact, the red leaf variety is no longer a red leaf variety when you don't have blue or UV. And then as you increase the blue, you get progressively more compact plants that are also a darker red. The warm white LED we see has a little coloration because it has a little bit of blue. And then on the bottom, you can see very similar effects as we did with the blue light. And so it turns out that we can really attribute the responses to not the changes to the green light, but the changes to the blue light. And so within a particular column where we have blue intensity the same, we can see very similar plants, whether it's from the red or from the green. In this study, we did a consumer preference study. So we took 7 of these treatments and worked with a colleague in food science, and we asked them to do some taste testing and to evaluate their preferences of these different lettuces. They were not told anything about the way they were grown. They weren't asked for too many specific things. They were basically asked, how do you like this sample? And were given, I think it was a total of 5 different samples and asked them questions. So the treatments were randomly delivered to people who only had, the only criteria to participate was that they ate salad at least once a month. So then it was, kind of whoever signed up. We had, obviously on campus, we had a large number of students, but we also had people who were older. So they were given basically these small samples of lettuce, asked to consume them, and then given a series of questions on a computer. And these are the ratings. And so most of these treatments are from plants grown indoors. The treatment on the far right of each set of columns is plants grown in a greenhouse. And as you can see, when there were differences, they were usually just different response for plants grown in the greenhouse compared to the plants grown indoors. So looking at the sensory rating on a 1 to 9 scale, the plants grown indoors had a slightly higher acceptability rating than the plants grown in the greenhouse. And similarly, the flavor and the aftertaste were deemed to be a little bit better than the plants grown in the greenhouse. Looking at a sensory rating, the plants grown in the greenhouse were probably not liked as much because they were more bitter and a little bit less sweet. So again, that's probably a correlation. But because of that, when they consume these different varieties, you can see that the greenhouse-grown lettuce had slightly lower ratings for meeting expectations and willingness to buy. So that was surprising. We didn't expect it. I was expecting also that we would see some differences in preferences among the different indoor lighting treatments because the colors of the leaves were quite different and presumably the taste, but apparently they did not perceive those at least consistently. I think I have time to go into, well, actually I'm just about finishing. So what I'd like to do is I'll very quickly go over some of our most recent research. It's still preliminary, but we're looking at delivering either UVA or blue light at the end of production to increase some of the quality attributes, specifically the coloration and the nutrient densities. Here are the different treatments, but basically we're giving 6 days, we're growing all the lettuce the same, and then we're changing the light environment for the last 6 days of production, delivering either UVA alone, blue light alone, or in one case, the two of them together. This just shows you some of the results that we have seen, and some similar results have been published by others. And I think Dean Kopsel in our later presentation might touch upon this. But we see elevated coloration when we're giving additional blue light at the end of production, and similar responses whether it was delivered from UVA or with blue light. We're also measuring some of the concentrations of some of the secondary metabolites, things like anthocyanins and total phenolics, and typically we see them increase as we increase the intensity of blue with or without UVA. So with that, some general conclusions of this study that we saw went over very quickly. The yield and the quality attributes generally increased with light and DLI. But I didn't mention this, that tip burn can also occur under, actually I did at the beginning, under higher light intensities, and that some of the effects that we have noted will likely depend on CO2 concentration and temperature. That's not something that I have studied, but I have a colleague who's part of our Leafy Greens project who is looking at interactions of light with these different environmental parameters. So we conclude here with the general effects of these different wave bands on influencing the morphology and quality attributes of many of these plants. We know that blue light and UV act relatively similarly. We get more coloration, more nutrition, thicker leaves. We get more compact growth. Some varieties we see less intumescence, but oftentimes this comes at an expense of decrease in yield. Green light is generally well used by plants. It can penetrate leaves and sometimes canopies a little bit better, can promote extension growth, but also you may have less coloration and thinner leaves. And finally, a lot of work being done now with far red. We know it promotes elongation. It can potentially then capture more light. It can influence flowering of some ornamentals, but it can have some undesirable effects like diluting the color of the leaf so it's less green or less red, and make the leaf a little bit thinner. So with that, thank you for your attention, and hopefully we have a few minutes for any questions that there might be.
Thank you. We're really a little bit short on time, but of course we have some questions. One question was, in the panel test, how did you standardize production? Were plants of the same age?
I'm sorry, can you repeat
It's
that?
really hard to understand each other here on stage, I know.
There's a bit of echo that.
Yes.
Yeah.
So, in the panel test, how did you standardize production? Were plants of the same age?
Yes. Well, so when we do all of these harvestings, we make sure we capture all the data on the same day. So in that last study that I went over very quickly, we just basically they were grown under the same light environment. We then gave 6 days of different treatments, and as a control treatment, we also, instead of adding more blue or more UV, we added more red and more green, so that the photon flux density was the same, but we weren't delivering more of those high energy wave bands.
Okay, thank you. So there's one more question, but maybe we can ask it in the panel discussion afterwards, okay?
Very good.
Perfect, thank you so much.
Thanks so much.
And we will continue with a short coffee break, so every one of you can have a warm coffee because it's really cold inside here. And also for our digital participants, grab a coffee, grab tea, or any snack. And then we will be back in 30 minutes. And for everyone being live here, we also have different information stands where you can inform yourself by grabbing a coffee. See you in 30 minutes. So, back in time, back here in the auditorium, I think it's called, or something like that. We will continue now with our next speaker. Please take your seat, and I hope everyone digitally is also back with us. So, our next speaker is from the University of Copenhagen, and there she is part of the crop stress physiology group in the section of crop science in the Department of Plant and Environmental Sciences. And she will tell us more about the interaction between leaf temperature, light, and CO2 concentration for photosynthesis, and how this can be used for the optimization. Please welcome Professor Dr. Eva Rosenquist. The stage is yours.
Thank you very much for inviting me to the first symposium in 2 years' time. I was asked to talk about temperature in relation to plant production. And what this will actually be about is how to use the climate response of photosynthesis in the way you actually control the climate in a greenhouse or a vertical farm. So, the climate parameters we have available in climate control is the light, CO2, temperature, and air humidity. And of those two, the light and CO2 concentrations, they are the crucial ones for the biomass production. And the temperature fine-tunes all biochemical processes. Of course, it's involved, but it's also very important for the developmental rate of a plant from vegetative to flowering stage. The air humidity is immensely important if you have problems with it, because you can get stomata responses where the stomata don't respond to changes in the environment if the humidity is too high, and you can get fungal disease. But when I talk about temperature, I will focus on the temperature from a photosynthesis point of view. But one should be aware of that temperature is also important for the plant morphology. And you will see a lot of examples that is taken for ornamental plant production and not vegetables, because that is what we have been doing the research on. But to the right you can see two chrysanthemum plants that has been grown under different temperatures. And the biomass is actually not much different between them, but the stretching. So many plants respond to a little bit too high temperature, but just growing in the height. And this I sometimes also see when I buy thyme, and sometimes even lettuce in the shop. I can see these long wobbly ones. And they are often actually grown at a little bit too high temperature. So which temperature do we choose for the different crops? Well, it depends where they come from. And when it comes to vertical farming, up to now it has mostly been like herbs and lettuce, microgreens. And there we get the plants in two groups, the warm-loving ones and the cool-loving ones. And basil and coriander, they don't mix very well with all the other species, because the other species, they are temperate. Most of those I can actually grow in my own garden, while basil and coriander definitely needs to go into the greenhouse. So in these huge systems, and people are trying to have a wide range of produce, there are some mixing that is not so good. So the photosynthesis climate responses, they respond with a saturation curve for light and CO2, while the temperature response is an optimum curve. And these curves are related to where we actually have the optimum temperature for growth. So we have the temperature response of photosynthesis, and strictly speaking, this should actually drop in the end. It's taken from a book, so I just copied it. And then we have respiration. Because we produce carbohydrates in photosynthesis, but we also use these carbohydrates in respiration, which is necessary for growth, but there we lose CO2. So if we take the red minus the blue curve, we get like the growth potential. And each species will have a base temperature. And if we take a plant from Greenland, or if we take a plant from a tropical rainforest, of course the base temperature will be very, very different. But all of them will have an optimum range, which vary depending on the species. And since the growth is a combination of CO2 input and CO2 losses, the higher temperature we have, the higher respiration we have, and you will lose more CO2. And it's a necessary loss, but we can balance what we do. So in many cases, it's actually better to have a lower night temperature where we only have the losses than day temperature in comparison to a constant temperature. For many of the ornamental plants, that is the case. So the light response and the CO2 response of photosynthesis, that determines the potential for carbon gain at the different values of the parameters. So the question is, how much light do we give in a vertical farm? And what CO2 concentration do we have? And when it comes to light, this is only a wild guess. I have no idea how much light that has been installed in vertical farms. But if we compare to conventional climate chambers, it's often up to 300 micromoles. When it comes to CO2 concentration, the CO2 response curve is plotted against the intracellular CO2 concentration inside the leaf, which is slightly lower than outside. And that arrow indicates the range which we are when we have 400 ppm CO2. And a good stomata conductance, so stomata are open to let the CO2 into the leaf. But if you look at where the arrows are, we still have some potential of photosynthesis that is not always utilized if we do it at low light and ambient CO2. So what light do we have? This is just the light data from a quantum sensor on top of the roof of the greenhouse. And it's just 3 different days. And if we have clear sunshine, which is the red, we have this very nice bell-shaped curve. If we have scattered clouds, we actually get a little bit more absolute light because we have the white scattering from the clouds. And if we have overcast and rainy weather, it just crap. So the numbers up to the right, that is the daily light integral, as has been spoken about before. So if we look at how much light we get, this is 4 years of data from Denmark, the top of the roof of the greenhouse. So in Denmark, we can get up to 50 moles per meter square a day. And in the winter, it's less than 10% of that. So in Denmark, we really need to have supplementary light in greenhouse production. Otherwise, nothing happens with the production in the winter. But if we then look at how much light do we get from the lamps, the graph to the right, you have the par light in micromoles per meter square second on the x-axis, which is in the upper part of the canopy. And then you have the burning time. So if we have 200 micromoles for 20 hours, then we have a DLI that corresponds to March and September, October in Denmark. So to me, it has a lot of potential more for carbon gain if we can increase the light level. But then Eric showed some very interesting results where he peaked at a DLI of 18. But I would not be surprised if you get different results if you have traces approach in constructing this DLI and actually do it with different light levels during the day than if you make a completely square light. So we show all these curves that are measured at steady state when the plant is in balance. So the question is, what happens in real life? Here we have 3 graphs that was measured in a greenhouse on a chrysanthemum plant. So 9:30 in the morning, I put the leaf in a cuvette, and this is natural daylight. And the temperature in the leaf increased gradually because I turned off the temperature control. And this is the photosynthesis that I got. And the pattern in the light level corresponds to what you see in photosynthesis, except for one place. Around just below 240 minutes, there is like 20-30 minutes with low light. And then suddenly an increase in light level. And their photosynthesis cannot follow up with an immediate increase in light level. So if we take the light and plot it against photosynthesis, we get the light response curve. And this light response curve also follows the temperature response, because the blue data, which is cooler leaf, have higher photosynthesis than the red data, which is a warmer leaf. So it's down the slope of the temperature response curve. And the circled data points, they are the ones that was taken just after this increase in light level. And there we have a limitation in stomatal conductance and the activation state of the enzyme in the Calvin-Benson cycle. Because all these things, they are co-regulated. And biochemistry and stomatal regulation, it takes a little bit of time. So if we move on to the temperature response, this is the basis for the dynamic climate control that we do in greenhouses. And it was a project from 1996 to 2001 called IntelliGrow. And on the X we have leaf temperature, on the Y we have photosynthesis. If we take a light level which is in the bending part of the curve, 990 micromoles per meter square in second, we actually get a peak, kind of. It's wide, but there is a point where you have the best photosynthesis. But if you lower the light level to the steep slope of the curve, this temperature response gets very, very flat, because the light limits much more than the temperature. So I am not the one who is going to try and say where we have the optimum here. So if we, from this hypothetical optimum, decrease the temperature by 5 degrees, we will have a loss at high light that is much bigger than at low light. So the ideology behind doing this dynamic climate was that, okay, when we have real crap weather, we don't heat the greenhouse more than necessary. We let the temperature decrease to maybe 17 degrees, because we don't lose so much carbon gain anyway. And if the light is there, we make sure that we have a better temperature, but we get that for free. When the sun is shining, we will have a higher temperature in the greenhouse. So, having said that, in January and December, this does not work in dynamic, because we only have crap days. But from February to November, this actually works. So, to move on to CO2, the orange curve shows the temperature response over a wider range of temperatures at ambient CO2. And you can see on the CO2 concentration that this was measured a few years ago. And the upper curve shows at 1,000 ppm CO2. So if we, at the temperature optimum at ambient CO2, increase the CO2 concentration to 1,000 ppm, we gain 40% more CO2 fixation. And in conventional climate control, we often ventilate at around 25 degrees for chrysanthemum, and we often have a set point of 21. So we operate from 30 to 50% extra CO2 in this range. But when I saw this data, I thought, why don't we allow the temperature to increase to 30 degrees? Because we have the light. So if we do that, we gain 75%. And we started to make a ventilation set point of 30 degrees, but always making sure that we had elevated CO2. So the question is, what makes this temperature shift, the optimum shift to a higher level? And to find out about that, we need to look at the Rubisco enzyme that does the CO2 fixation. And the Rubisco enzyme in the Kelvin-Benson cycle, it needs ATP and NADPH from the light reaction. And the C and the O means carboxylase, which is CO2 fixation, which makes carbohydrates that are used for growth, while the oxygen means oxygenation. And that does not produce any carbohydrates, and it releases CO2, so it's only a loss. But on the other hand, if there for any reason is not available enough CO2, then by the oxygenation, the light reaction can still go on, so we avoid photoinhibition, which Tracy talked about. So what determines the CO2 fixation and oxygen fixation? It's the CO2 to oxygen ratio in the cell sap, which is in the water. So this is how much oxygen and CO2 that can be dissolved in water at different water temperatures. And it's more CO2, more oxygen available in the cell because we have more oxygen in the air that is in the equilibrium with the water phase. And if we take the ratio between the two, we get a decreasing CO2 to oxygen concentration. And this is the reason why when we increase the temperature and we have ambient CO2 concentration, we will have more and more photorespiration taking place, which is why this temperature optimum drops from 23 degrees and up to higher temperatures. But if we give elevated CO2, then the CO2 concentration will actually increase in the water phase. And that increased CO2 will also increase the molar ratio of oxygen to CO2 to oxygen. So by increasing the CO2 concentration, we suppress photorespiration. So the answer to the question is that we suppress photorespiration by elevating the CO2 concentration. So it's basic plant physiology put in practice. So we have a leaf, and we have the light reaction, and we have photosynthesis. But the CO2 supply is dependent on the CO2 outside, the leaf temperature, and the stomatal conductance. How much is allowed into the leaf? So how much CO2 do we have available? Well, I have no idea if there are any commercial vertical farms that work with elevated CO2. I have no idea. But for the future, sooner or later, it will have to come. So this data is from the greenhouse at work. And it was in the autumn, so the ventilation was not so high. We didn't use the vents very much. And it was fairly filled with plants. So the question is, which leaf area index, leaf area per ground area did we have? And I did a rough estimation. So the different tables have 3 to 5. So fair amount of plants. And it was in the autumn, so it was sunlight, but it was medium light. And the greenhouse was mostly closed. A little bit of ventilation, but not much. And we had, on average, 322 ppm. And that means that we actually moved down to this steep part of the CO2 response curve. So I don't know how the heating and air exchange is constructed in these vertical farms. But if you have a certain amount of leaf area, and you may have half leaf area index on each level, But if you then have a stack of 10, you still get up to 5. And I don't know how much light you have and how much photosynthesis you have, but if you don't have an exchange of air with the surrounding, you will probably have subambient CO2 concentrations in a farm. So what I've talked about is summarized in this little paper, and you can find it at the link to the right. So for practitioners who actually work in vertical farms, this is where you can get some information about it. So this is the final experiment that we did in 2001. And it's an autumn production. And these are chemically growth-regulated plants to get the correct height. And we used a mean temperature modification of the set points from IntelliGrow to make sure that we didn't have any delays. And these are the energy savings. So to the right, we have the standard climate, which was 2119. And by having a span from 15 to 30 degrees, and the ATF means that we run the photosynthesis models on 80%. We don't run it on 100% because the aim is to save energy. But we can still save 49% by allowing the temperature to fluctuate in the greenhouse and just make sure that we compensate with the CO2. And of course, the 30 degrees is very, very short and very occasional, and especially from September to November. It's not much 30 degrees there. But in the summer, it also works up to 30 degrees. So the result of this is the official statistics of the energy use in the greenhouse industry. And from 2002 to 2014, it decreased by 56%. And the growers themselves, they say that it's mostly because of the dynamic climate control and secondarily because of investments in better climate shield. The electricity has been the same, and this is before people started to work with LED light. So in total, it's a halving of the energy use in total. But of the energy for heating, 25 to 30% is for humidity control. And this is an issue that we still work on. And if you get high humidity constantly, many species lose the ability to control stomata. And that's why I sometimes find packages of herbs in plastic boxes in the shop, and there is some condensed water inside. And it's not because they were like sprayed with water when they were packed. It's because the plant is actually losing the water and it condensates on the package. So in Denmark, we work a lot with the greenhouse industry. But many of these, they also start using vertical systems. So this is Kalanko blossfieldiana, which is a can plant, so they close the stomata during the day. But even so, this nursery actually have dehumidification. And they extract the latent heat of the air, and they store it in accumulator tanks, and they use heat pumps and heat exchangers, so they really minimize the energy use. And they also work in layers. This is the propagation area in the basement, under one of the very long greenhouses. And the quality you get from propagating the plants at very constant temperature and conditions, you can really increase the quality of the propagation. And they even have a double layer up in the greenhouse where they have put on LED lights under the tables, so they have a double layer where they transport the plants under the tables, and there they have a buffer zone where the plants can be kept for some days with the LED light before they come up in the end of the greenhouse and start to move towards where I'm standing. So why are we talking about dynamic climate in relation to vertical farms? Well, it's because there will be more and more green energy. And in Denmark, 50% of the electricity comes from windmills. And they don't operate by on-off a button. So in the dynamic climate control, we can save 50%. We can also save energy by using a dynamic light control, which takes the weather and electricity prices into account. And there are prototypes being built as we speak, where we are applying for money to look into the energy consumption in vertical farming. And that leads us to the final slide, where we work on having vertical farms and nurseries as part of energy buffers in a smart grid solution with more sustainable energy, where if there is an overproduction, the nurseries can be asked actually to turn on or upgrade the energy use and to downgrade it when it's not so much energy produced. And we want to integrate vertical systems into the horticulture industry, the existing one, and look at propagation versus the final production, and which light requirements you have in the different stages, and the energy use of the different parts of the production. So how much of the growth potential that we have in the crops that we use in vertical farms is actually used? I don't know. But it would be interesting to find out. And can we produce more than microgreens and herbs? And can we use it? Can we do it sustainably? That is the question.
Thank you so much. Next time I will also analyze it in the supermarket, all the sizes of the plants. Thank you. For one question for you, what method did you use to measure the leaf temperature?
Well, for all these measurements, they have been measured with a thermocouple in contact with the leaf. So it has been measured on the bottom side of the leaf, so the thermocouple is not hit by any radiation. So it's a contact sensor on the bottom of the leaf.
Okay. One more question. Did you investigate the effects of the fluctuation of the temperature during the night?
We haven't investigated as a separate parameter, because the thing with the dynamic climate control is that we had actually to integrate everything. Normally you take one parameter at a time, but if we are going to do that, we will have to work for 20 years before we got any solution. So we did it by integrating everything. So we have data that looks at 15 degrees and 70 degrees minimum temperature, but it's always in combination with different things that happens during the day, because it's the irradiance during the day that actually controls what happens, and that we cannot control.
Mm-hmm. Okay, we will see you afterwards in the panel discussion as well, right?
Yeah.
Thank you very much. So now we've already learned a lot how to kind of manipulate the plants. Let's also see how we can maybe manipulate the nutrient with our next speaker. His name is Professor Dr. Hans Lambers, and he's from the University of Western Australia. And one of his key researches is about mineral nutrition of Australian native plants. So good evening to Australia, and good evening to Professor Dr. Hans Lambers. Mr. Lambers, can you already hear me? I know there's a huge distance to Australia, but I hope the internet is quicker than we are. So maybe we check if I get a sign. Do we have a connection to Mr. Lambers? Couple of minutes, okay, because I thought he needs to be on time very early. So yeah, Australia is far away. I hope every one of you learned a lot. Mrs. Rosenquist, I can ask you one more question if that is fine for you, because we received one more question. Is it fine for you?
Yeah, sure.
Okay, perfect. Thank you for being so spontaneous. Let me check where the question was. Were the energy costs for the CO2 added in the greenhouses considered?
We actually pay less for the CO2 use in the dynamic climate than in the traditional climate, because you apply the CO2 when the vents are closed. And if you wait until 30 degrees to open the vents, in comparison to 25 degrees, if you work with 25 degrees, you have much more open, close, open, close. And what really costs is actually to fill up that volume that you have in the greenhouse. So we thought before we did it that it will be more expensive in CO2, but it turned out to be the other way around. Because the CO2 supply, when you do it in a closed greenhouse, you just substitute the leakage and the CO2 fixation into the plants. So the leakage is like 100%, and you turn the CO2 supply off, and then you have to refill the greenhouse. So the CO2 cost was lower in the dynamic climate than in the conventional.
Perfect, thank you very much.
Welcome.
Now checking again, one more second. Is he already now online? Do we have a connection? I'm not sure if that was a yeah, but please go ahead with your question then.
Hello, thank you for the presentation. I'm one of those guys that are actually trying to build a vertical farm. And yeah, you were wondering if there was someone using, it's just a comment, and you were wondering if there is someone already using this technique for concentrate CO2. As much as I'm aware, there is Grow Underground in London. They build the vertical farm in a former tube line. So there is a natural accumulation of CO2 in their farms using this system. So the CO2 is just going inside the tunnel, and they're just accumulating naturally that.
So I didn't understand. So does the CO2 decrease or
increases? No, increases. They concentrate the CO2 naturally in the tube station.
Yeah. Yeah, that's a way to do it then.
Yeah.
Yes.
Thank you. I think there was another hand raised. Perfect.
Yes, thank you for the presentation. Actually, I would like to ask him about, because I think everything that you presented was for C3 plant. And I would like to ask if, I don't know, if you know anything, any type of this response for a C4 plant.
Well, the C4 plants, they don't respond to elevated CO2 like this because they have a CO2 pump themselves. But they often also have a higher temperature optimum automatically. So, for example, we don't have any natural C4 plants in Denmark because the C4 metabolism, it requires more energy to run. So it's only under conditions where the CO2 supply into the plant is limited or photorespiration is high because of high temperature. That's where it pays off really well to be a C4 plant. So I haven't done anything on C4 plants in this system, but I would suspect that they manage quite well up to 30 degrees just on their own. But you don't need to have elevated CO2 to go do that.
Okay. And about this relative humidity problem, I don't know, maybe it's a comment, but did you try, I don't know, maybe you tried, but about ventilation, artificial ventilation to remove this artificial?
Yeah, well, in the greenhouse, we always use ventilation to get rid of the humidity. So what you do is that you open the vents and you put heat on the pipes so that the warm air is actually pushed out of the greenhouse. And that's why it costs like 25 to 30% of the energy to regulate the humidity. But this is in greenhouses where we have a cold roof. And that means that we have a lot of condensation on the roof. So the roof in itself works as a dehumidification device. And then some nurseries on top of that have dehumidification. And you just tell me when Hans Lambers comes on.
Now, if it's possible, if I can interrupt now and you can continue afterwards. I'm very sorry, but we're really tight in the schedule. But thank you very much for your questions. And Mr. Lambers, I already introduced you, so you can just go ahead. Thank you very much. Do we have the sound also?
Yes, you should have the sound.
Now we hear you, yeah. Perfect, thank you.
Okay. Well, sorry about the delay. I had some troubles connecting, but there we are. So oh, there's an echo in it. I hear. So the question that I was asked to address was, will increasing plant productivity in this wonderful
Mr. Lambers, can you hear us? And is everything fine for you?
create a problem for the uptake of nutrients in particular? So that is what I would like to address. And let's first have a quick look at what plants actually need in addition to carbon dioxide and water. Of course, they need a range of nutrients, in particular the macronutrients. They also need a series of micronutrients. All of them are essential, but chloride and nickel only for some plants. And then there are a number of beneficial elements, and silicon is particularly important. And it is even an essential nutrient for horsetails, for example, but for most plants, it is just a beneficial nutrient, but one to really pay attention to also in vertical farming, I believe. Now, in most systems, nitrogen and phosphorus, they are frequently the limiting primary productivity, the nutrients that are limiting primary productivity, both in natural and in managed systems. But of course, Santold already alluded to that. As soon as you make nitrogen and phosphorus no longer the limiting factors, then another nutrient will become a limiting factor. And we often refer to that as the Liebig law of the minimum. But as Santold already pointed out, I think it is really very important to give credit to Sprengel, because he was actually the person who started all of this. And I would like to use the word plagiarism, but if he had done what he did these days, then I think we would use that term. Anyhow, I think it is credit where credit is due, and I think it would be much better to refer to this as the law of the minimum, as the Sprengel-Liebig law of the minimum. Well, photosynthesis clearly is a crucially important process for plant growth and productivity. But when I listen to people talking about photosynthesis, not today at this meeting, I should immediately add, but I hear a lot that photosynthesis is the limiting factor, and that determines growth and that determines yield. But the question is that really as simple? And the answer actually comes from a big story that is recorded by Titus Livius, or Livy as he is called in English. And he was a Roman historian, and he tells the story about a consul that had trouble with the strike in Rome. And he basically, because the people were on strike, felt that they were actually treated very badly. And the consul told a very nice story. He talked about the stomach, and he talked about the other parts of the body. And the other parts of the body were really thinking that the stomach was getting a very easy deal and basically getting a free ride. So they went on strike, and the stomach no longer got any food. And of course, very soon it turned out that the rest of the body also suffered. And that is an important story, because plants actually function in very much the same way. There is not one particular process that we would say is the most important. We need all the processes. And if we don't have them, well, then we are in trouble. So is photosynthesis really the most important process when it comes to growth rate? Well, I go back to some work that I did when I worked at Utrecht University in the Netherlands, where we compared a wide range of species, 24 to be accurate. And we measured the relative growth rate, plotted here on the x-axis, a great variation in growth rate, and we actually worked out whether it actually was correlated with what we call the net assimilation rate, or with the leaf area ratio. And the net assimilation rate is not equal to photosynthesis. Photosynthesis is an important component of it, but so is respiration. And what you can basically see is there's no correlation with the net assimilation rate. And in fact, if you go to photosynthesis here on the right, you can see there is no correlation with photosynthesis either. So that difference in growth rate between all these species over here was actually driven by the leaf area ratio, which is a product of the leaf weight ratio over here and the specific leaf area. So, as Leo already pointed out, allocation is crucially important when it comes to productivity. It's not just photosynthesis. Of course, photosynthesis is crucially important, but allocation is also very important. And when it comes to yields, so not growth rates of native plants, but of yields of crop plants, we can go to wheat at the left, or we can go to peanuts at the right, and we can see if we go to old cultivars over here, introduced in 1900s, or younger ones, newer ones, introduced in 1980, you can basically see the total shoot weight is not really different. That's the same for wheat as it is for peanut. But the grain yield has gone up substantially in wheat and also in peanut. And that is basically a matter of allocation. So the harvest index, the proportion of material that we harvest divided by the total weight of the plant, the harvest index has increased. True for wheat, true for peanuts, and in fact it is true for a wide range of species. So allocation is really what has driven the increases in yield. And then of course people will say, yeah, but with the increasing CO2 concentration, clearly we have seen an increase in growth and productivity. And if you compare a wide range of species, indeed there is an effect of the elevated CO2 concentration. It's greater for C3s than for C4s, but by and large there is clearly a positive effect. So clearly photosynthesis must have been the limiting factor. And even here, you've got to realize that it is actually a little bit more complicated. And that is illustrated in this slide over here. And what we see over here is the filled circles are plants grown at high CO2, elevated CO2, and the open circles are plants grown at ambient CO2. And you can really see an increased growth rate at elevated CO2. But you can also see that with time, that effect actually disappears. So there is a boost of growth, and thereafter, the growth rate actually slows down to the level of the control over here. And all of that is basically summarized in this beautiful scheme over here. Indeed, elevated CO2 leads to more carbon, leads to greater rubisco activity, suppression of the oxidation reaction, as we just heard. Photosynthesis increases, but what then happens is there is an accumulation of non-structural carbohydrates, and they have a negative effect on photosynthesis. Rubisco is downregulated, and the rate of photosynthesis, the photostatic capacity, I should say, actually declines. And below ground, we also have an increase in the sugar concentrations, and those sugar concentrations actually tell the roots to grow faster, not just because the roots have more carbon to play with. It is actually because genes are turned on that control the growth rate. Ion transporters are upregulated, and again, they're controlled by sugar levels. The genes involved in phosphorus starvation, they're also upregulated. So those sugars, they actually tell the plants that there is a shortage of phosphorus. So can we go a little bit further? And can we go back to our tomatoes, for example, and look at the pre-storage phase and at the storage phase? And there's a very nice bit of work that we would like to talk about, and that is the storage phase of tomatoes. And what these people actually did is they increased the CO2 concentration during a 10-day period when sugars were accumulating, when the tomatoes were actually growing. And you can really see if you do that, increase the CO2 concentration from 350 to 700, you can really see that the rate of photosynthesis increases. The export of sugars also increases, but if you look at the fruit weight, there is no effect whatsoever. And if you look at the sugar concentration in the tomatoes, there is no effect either. So if you increase your CO2 concentration during the accumulation of carbohydrates, during the growth of the tomatoes, you will not find any effect of the elevated CO2 concentration at all. So the increased rates of photosynthesis during the growth of those tomatoes, they do not increase the fruit growth or the sugar concentration. And it is actually, as these authors clearly demonstrated, it is actually the activity of the hexose carriers that determines the growth rate and the yield of the tomatoes. Clearly, genotypes with a higher yield, they have a greater capacity to transport sugars into the fruits. And that determines phenotypic differences in growth. Photosynthesis, of course, is important, but once the fruit has decided how fast it will grow, well, then you can increase the CO2 concentration and it will have no effect on the growth rate at all. So, okay, we've covered photosynthesis, an important process. And of course, in vertical farming, you will really, and we've heard beautiful talks about how we can optimize photosynthesis. You really want to have a maximum photosynthetic production. But the question then is, will then the root's capacity for acquisition of water and nutrients become the next bottleneck? And do we have to tackle that? So let's have a look at an old experiment done years ago with wheat. And John Pasura pruned all the roots except one, and then looked at the dry matter accumulation in roots. Well, if he pruned down the root system to a single root, then the total roots accumulated less dry matter than if all the roots were kept intact. But now let's look at the leaf area, the right over here. Well, whether you have single roots or all the roots makes no difference for the leaf area development. And if you look at the dry weight of leaves and stem, again, it makes no difference. And if you look at the ear, so the actually harvestable part of the plant, you can actually see that curtailing the root activity actually increases the ear weight. So clearly, plants have plenty capacity to take up water. We don't need to really worry about that. What about nutrients? Well, when I was a postdoc a few years ago at Melbourne University, we grew plants with a split root system, and we either gave both plants nitrates, or we gave only one nitrate and the other got no nitrate at all. And we had a whole range of different treatments, and I don't want to go into the details. Suffice it to say that the plants with nitrates, they grew better, as you can also see in the photo over here, than the plants that did not get nitrates. But importantly, if you look at the shoot weight, all these treatments basically gave the same shoot weights. So it didn't really make much difference whether you gave nitrate to only a single root or to the entire root system. The shoot weight grew, the shoot weight developed in much the same way. And that actually led to an interesting model shown you over here, where we show that the roots take up plenty of nitrates, the shoots incorporate the nitrogen to some extent, but a significant amount is also cycled back to the leaves. So even those roots that are grown with a limiting amount of nitrate, they still send most of the nitrates or nitrogen amino acids to the leaves, and the leaves then send some of that nitrogen back to the roots. So there's a continuous cycling going on. So what should we aim for then, when we can grow plants in an environment that is really very, very favorable for growth and yield? Well, I think I'll get to that in a minute, but I've got a few other points that I would like to make before. And that is a model that was developed in France to actually account for the uptake rate of nitrates. And this model basically tells you if leaves require a lot of nitrogen, or if the growing pots require a lot of nitrogen, then the roots basically get told to take up more nitrates. And the important message here is that the nitrate uptake is not determined by the uptake capacity of those roots. It is actually determined by the demand of the leaves. or of the growing pots. And that model is still valid these days. It has been made a bit more complicated here in this slide over here, but basically what it amounts to is leaves need nitrogen, and when they need more, they will basically tell the roots to take up more. And this summarizes that story in our textbook, and that basically tells you the story. If we have plenty of nitrate, plenty of nitrogen for the plants to take up, they will also produce, the roots will produce a lot of cytokinins. And these cytokinins are the signaling molecules that go to the leaves. They build up a high photosynthetic capacity. They lead to rapid leaf expansion. And those leaves will consume most of the sugars produced in photosynthesis. So very little of the sugars actually go back to the roots. On the other hand, if we have a low nitrogen supply, we have very little production of cytokinins, then the photosynthetic capacity declines, but growth is far more sensitive to nitrogen supply than photosynthesis. So we end up with an excess of sugars, and those sugars are returned, are sent to the roots, and there they are a signal for the roots to grow faster. So there is signaling going on from roots to leaves. There's also signaling going on from leaves to roots, and that signaling from leaves to roots is basically a matter of signaling by sugars. So sugars are substrate for sure, but sugars are also an important signaling molecules. What about other elements like phosphate, for example? Well, this is a wonderful experiment carried out in David Clarkson's lab, who passed away earlier this year, but he did some wonderful work that I want to talk about here. And he had a design where he starved the plants of phosphate for some time, and he measured the phosphate uptake in a different compartment over here. And the control was not starved of phosphate at all. And the phosphate uptake measured in this smaller compartment was 288, and 62% was actually translocated to the shoot. When you starve the plants, you can really see that the uptake capacity increases. And when you starve the plants for 7 days, the uptake capacity increases even further. But less of the phosphate is translocated to the shoots. What does that tell us? Under normal conditions, the roots do not work as hard as they can. They wait for a signal from the shoot to tell them how hard they have to work. So there is plenty of capacity to take up. So what this basically means is that plants anticipate. There's a very clear feed-forward response. Plants don't function in a manner that makes a particular process limiting. Photosynthesis isn't limiting. There's always an excess capacity. Nitrate uptake is not limiting. There's an excess capacity, as we've shown with our split root experiment. There's also an excess phosphate uptake capacity. And sugar sensing and signaling plays a very important role to make sure that the shoot growth and the root growth and all these processes that are all important for growth are balanced. Spare capacity is tremendously important in plants growing in their natural environment for native plants. It is also important for crop plants because the environment is not stable. We have a high light intensity day, we have a low light intensity day. It's not always optimal. So the spare capacity is incredibly important for a functioning plant. But if we can grow plants in a favorable environment, well, then we can get rid of that spare capacity and we can really push the plants to their limits. And we really have to interfere with that sugar signaling and push the plants further than what they would otherwise do. So plants anticipate. And in a review that is about to come out next year in annual review of plant biology, I actually summarize this like this. And this refers to phosphorus. It's the main element that I actually focus on, but the story is very similar to our plants that anticipate. You can actually push yourself to the limit a little bit more and not worry about disastrous. And you wait till the fire comes and then you rescue what you can. And that is typically what a lot of crop plants would do. And I call that the starvation response, the nitrogen or the phosphorus starvation responses. When I go to native plants that I do a lot of work on, they've got a different story. They carefully consider the risks, and they are super efficient when it comes to using the phosphorus, but it goes at the expense of the growth capacity. So they are actually controlled in such a way that they don't actually exploit their full capacity, whereas plants that are left, they exploit a greater proportion of their full capacity. But when you really deal with plants in a protected environments, like we're talking about today, then you can push them a lot further. So what should we aim for? When we can grow plants in an environment that is very favorable for growth and yield, where you have actually pushed photosynthesis to its absolute limit. The temperature is perfect, the CO2 concentration is absolutely perfect. So what do we do then? Well, we actually basically want to control and optimize all inputs, and other speakers have talked about that before me. And so we really want to maximize the production. And in fact, and that is a matter of breeding, we want to minimize those safety margins, because those safety margins are only important to cope with fluctuating environments. And the genetic improvements that we want to make, they should actually get rid of those safety margins. And that basically means that we've got to target the sugar sensing and make plants less sensitive to the sugar sensing. Plants likely have sufficient capacity to take up most of the nutrients. That's what the evidence shows for nitrogen, and that's what the evidence shows for phosphorus. It is probably true for most elements. Calcium in tomato, well, that there may be an exception, because in glasshouse conditions, plants may actually get calcium deficiency. So maybe that's what you've got to be careful of. But other than that, I think we are absolutely fine when it comes to the below ground activities that plants have to carry out. They've got plenty of spare capacity. Thank you.
Thank you so much for your presentation. We have one question here live in the audience. The microphone is coming. It's down here. Thank you.
Yeah, Hans, a very nice presentation, and your conclusion that there is a lot of spare capacity in the roots. Yeah, I tend fully to agree, but I would like to ask, could it also be that we have too much capacity? The plants take up a lot of nitrate in these, when we grow the plants in a water culture, we have a lot of nitrate. They take up, there is an influx of nitrate, but there's also an efflux of nitrate. Don't the plants take up too much nitrate, and isn't that in fact a loss? So is there a potential that we should take that to lower the influx of nitrate, and that there is in fact no efflux anymore?
Now, Leo, that's actually a very interesting question. I get to hear myself with a 1 minute delay, so this is actually rather complicated. So I will turn my mic off while I'm talking, and then turn it on later on.
Yes, you can talk.
Okay. That's a very interesting question, Leo, and that takes me back to work that we did while I was at Utrecht University. And indeed, what we found is that plants take up more nitrate than they actually need. But the interesting thing is that is typical for slow-growing species. They take up about 3 times more nitrate, and what do they do with it? Well, 2/3 actually leaves the plant again. So the influx-efflux ratio is such that plants are actually wasting a lot of nitrate, are actually wasting a lot of energy to take up that nitrate and then let it go again. Interestingly, you find that for slow-growing plants, If you try to measure the same, and we did, for fast-growing grasses, what we then find is that the efflux component is actually very small. And so that fits in with the story that we are talking about. Plants have a tremendous capacity to take up that nitrate. And indeed, if they take up too much, there will be an efflux. But if we push the plants and really make them use all that nitrate, then the efflux component, then that wasteful component, if you like, is actually going to be very small. Push the plants and they will become more efficient in terms of the influx and efflux of nitrate.
Thank you very much. I know there are more questions, but we need to continue with our next speaker. So thanks a lot to Australia and good night. And now we're switching continents again and saying good morning to Florida. I love this internationality here. And imagine we would have such a global event 18 months ago. I think it wouldn't work that well as it does now. So I welcome Professor Dr. Dean Kopsel, who is of the University of Florida. And please go ahead and tell us something about how we can manage the light quality to maximize the input. Thank you very much. The output, sorry.
Okay, well, good morning, everybody. As you look to the east to Australia, now you can look to the west for Florida, where the sun has not risen yet. So thank you very much for this invitation to speak, talk a little bit about the research that I used to do, looking at different ways to manipulate the growing environment to increase the nutritional value or the phytonutrient value of our crops. And I really wanted to kind of tailor this to the vertical farming theme of the symposium. The speakers so far have been great. I've been watching the live feed on YouTube, and that's actually been working out really well. So most of you might be aware of Florida. You know, we have a lot of beauty here and certainly a lot of attractions. We have, you know, our coastline. We have our springs area, you know, sport fishing off the coast, and of course, Disneyland and Disney World, excuse me. But Florida does actually have some hidden dangers. So we have alligators, we have poisonous spiders, and we have invasive species galore, whether they're organisms or reptiles or plants. So we have a lot of challenges here in Florida agriculture, certainly. Before I started with my presentation, I thought I'd give you a little bit of background if you're not familiar with Florida horticulture and some of the aspects of our industry here. So we have a little over 47,000 farms. They represent almost 4 million hectares across our state. You can see that within the U.S. agricultural system, we rank #1 for grapefruit and oranges, some vegetable species like snap beans and cucumbers. We rank #1 in the United States for sugarcane production. That happens a lot in Central Florida. And then we also rank very high among our horticultural crops, whether they're ornamentals or whether they're vegetable crops or whether they're herbal species. As you heard from most of the speakers already in the symposium, you know that organisms adapt to light, and plants are no different. And so when we think about how plants have actually evolved, we as horticulturalists have the ability to manipulate the environment to kind of push plants in different directions to get out of them what they want. And yes, if you look at the pictures on this screen, that the alligators do have places to go, and they will occasionally cross the streets on campus. Okay, so when we talk about light and plant physiology, we know that light is one of the most influencing factors on crop growth and development. You've also heard that, you know, from speakers this morning and speakers later on, that there are a lot of options when we come to lighting and especially vertical farm systems. And those light wavelengths carry different energetic values. The plants have evolved to have very specialized photoreceptors that will sense the light environment, and hence they can respond to that change in the light environment, and they can do that in ways that relate to primary and secondary metabolism. So when we actually talk about the nutritional value, we can look at both the primary metabolites and also the secondary metabolites. And in some of the work that we've done, and certainly some of the work that's been going on around the world in the past 15 years, light can have an impact on the nutritional values. And that's really exciting when we look at the industry and ways to manipulate plants in order to maximize those nutritional attributes. So first of all, I think I should probably define for everybody, what is a phytonutrient? And a phytonutrient is basically a plant secondary compound that provides some sort of protective mechanisms beyond normal mineral nutrition within the plants. And the more you learn and research about phytonutrients, you come to the conclusion that the roles that they play in plants are very similar to the roles that they play in our bodies, whether they're antioxidant or whether they're, say, photoprotective functions. And it's the role of these protective mechanisms that really allow horticulturalists or agricultural producers to maybe manipulate those environmental aspects by changing those conditions to maximize those things. And of course, you've heard of all the complexing interacting factors already this morning with the external environment, and phytonutrients are no exception to that. This particular graphic comes from a recent article, which was really nice as far as a summary of all of the different impacts that light will have on plant growth and development. So when we talk about the photoreceptors, they're going to impact everything from architecture to morphology to physiology to metabolism to mineral nutrition, etc. And what we had been doing in our program, where we're looking at some very interesting parts of the secondary metabolic pathways. So specifically, we were looking at plant pigments, so carotenoid phytonutrients, lutein, zeaxanthin, beta-carotene. We also had ability to measure chlorophylls. Now, chlorophylls don't really have an antioxidant value, but they are starting to play some roles in gut health and also overall nutrition for people who consume, dark green leafy vegetables. In other secondary metabolites, the nitrogen-containing secondary metabolites like glucosinolates. So we did a lot of work with brassicas, and brassica species have the accumulation of glucosinolates, which have been shown to have very important nutritional values. And then obviously the micro and macronutrients. So you heard speakers this morning talk about mineral nutrition from the plant sense. Really what we were looking at is mineral nutrition for what delivery mechanisms that plants could provide to human diets. Okay, so improving nutritional values, it's very interesting. You're starting to see a lot of research, certainly, on looking at LEDs and different wavelengths from those LEDs, and that's the advantage that it gives horticultural producers. And when we look at vertical growing systems, you can actually manipulate that environment, change light intensities, even at the different shelves on the stacks when you actually look about the production systems. And so the specialty crops are really amendable to these controlled environments. You can grow microgreens, baby greens, etc., and it really all depends on the market that those producers are actually going to. And the nutritional components that we were really concentrating on, again, like I said before, are the carotenoids, glucosinolates, the macro and micro elements, and then also some soluble carbohydrates, and this is flavor. And so that's one of the last things I'll talk about in my presentation, because when we look at increasing the value of some of these phytonutrients, most of them are associated with an acrid or a bitter flavor. And really, when we know consumer acceptance, taste is really one of those main factors. So are there ways that we can actually manipulate the light environment to be more conducive to a better flavor or a better quality of that particular commodity? So most of the work that I'll present today are on microgreens. So, these are really fast turnover, I can grow a wide variety in different climates, whether the greenhouse, grow chamber, or actually vertical systems. Very simplistic types of experimental designs, small shallow trays, with and without a grow pad. Normally, we don't have to provide any sort of mineral nutrition, so you just rely on what's actually inherent in maybe the media that you have or in the seed itself, and then you're going to harvest them at the optimum level, usually anywhere from maybe 12 to 20 days of production for a microgreen, and that depends on the species. Okay, so the first study I'd like to share with you is one of the first ones that we actually did as far as manipulating the light environment to see what we could do or what sort of impacts it would have on the nutritional values. So this particular study involves just a pre-harvest application of just blue light from an LED diode, and we use sprouting broccoli microgreens as the study system for the project. Okay, so as far as the materials and methods, what we did is we grew them in the grow chamber. So we set a day and night temperature of 24 and 20. We put them under a 24-hour photo period at a relatively low light intensity of 350 micromoles. Low light for a normal environment, but a good light intensity for a grow chamber environment. The light environment, we used LED diodes. In one experimental treatment, we used 20% blue and 80% red, and you can see the corresponding wavelengths on the slide. And then for the actual treatment within the study, we applied a 100% blue light environment for 5 days pre-harvest. And we thought that maybe this could be something that would be possible maybe in a commercial setting if the growers could actually have this available and then kind of dose it with light and then right before harvest see what sort of impacts. And I want you to notice that the normal LED light environment was 350 micromoles, but when we switched over to the blue light environment, since we didn't have enough on the array, that lowered the total light intensity for that treatment down to 41. So a really, really low light environment. And then we harvested them as microgreens and then did the lab analysis for the secondary metabolites. So first of all, I can tell you what impact it had on biomass. So when you look at the average plant biomass per gram seed weight, it didn't change between the treatments, or it wasn't statistically different. But obviously from the picture, you can tell that there was a difference or the impact in morphology. So due to that low light environment, the plants actually stretched, and we've heard a little bit about that already today. So no difference in the total biomass, but certainly a difference in the morphology that was present. Here's the data. So this is the carotenoid pigments that were present in the microgreens upon harvest or completion of the particular study. You can see that the shoot tissue pigments that we were able to analyze range from beta carotene, lutein, zeaxanthine, and down to the carotenoid pigments that function in the xanthophyll cycle. So for that red-blue treatment, so that was a continual 24 hours of red-blue, you can see the values that accumulated on a milligram per 100 gram fresh weight, which is typically what is actually measured by as far as nutritional content. The blue light treatment, again, which was 5 days pre-harvest at a much lower light intensity, but only the blue light environment. Interestingly, what was impacted the most was the beta carotene. So beta carotene actually accumulated to higher levels underneath that blue light treatment. No change at all under the other xanthophylls, lutein, zeaxanthine, some minor impacts as far as violaxanthin and some of the pigments that actually function in that xanthophyle cycle. Since it was a brassica, as a sprouting broccoli, we were able to characterize the glucosinolates that were actually present. So the red-blue treatment, you can see some of the values here, and these were actually expressed on a microgram per gram dry weight. I've highlighted two of the really important ones as far as our nutritional health. And you can see that that blue light treatment, which was, remember, 5 days pre-harvest at a very low light intensity of just strictly blue light, increased glucoraphanin in those broccoli microgreens by 37%. And glucoraphanin is really important because that hydrolyzes to form sulforaphane, which has been proven to be a very potent phase 2 detoxification enzyme. And it really has some good anti-cancer activity. Interestingly, gluconasturtium, which is an aromatic glucosinolate, normally found in watercress in very high concentrations, that increased by over 400%. Now, admittedly, it was under a very, very low level under the blue and red treatment, but it increased to that 400% under just that blue light treatment. And gluconasturtium is very important because that hydrolyzes to a very unique isothiocyanate called phenylethyl isothiocyanate. And that PEITC has been actually shown to have very potent chemopreventative agents too. So again, just changing that light environment showed a dramatic impact on some of these phytonutrients that were present in the microgreen tissues. What will be most interesting, especially probably to the previous speaker, is the impact it had on the mineral elements that accumulated in the tissues. I've highlighted the ones that are the most significant, I would say, in probably human health. So for the macronutrients, you can see the dramatic increases from that pre-harvest 5-day blue light treatment in concentrations of potassium, magnesium, and calcium. And then also, if you look at the micronutrients, iron and zinc, iron accumulated to really great extents under that short duration blue light treatment, and zinc accumulated to higher levels too. So when we look at manipulating that light environment, we're not only changing the secondary metabolites, but also the primary metabolites that have accumulated in those microgreen tissues. So that first study, certainly established a relationship between maybe that blue light exposure or that light environment with the secondary and primary metabolic pathways. And it was really interesting, just short duration, very low light applied pre-harvest had those impacts on not only beta carotene, glucoraphanin, gluconasturtium, and also some of those mineral elements. So looking through the literature, there is previous evidence to show that blue light or blue wavelengths will affect the gene expression in some of these metabolic pathways. As of yet, we don't really know which pathways were expressed or which pathways were actually impacted the most. but it kind of laid the groundwork to show us that the quantity and quality of some of these blue light wavelengths were certainly having an impact. Could that be the higher energetic value of the light, the impact it has on some of these uptake mechanisms or primary metabolic pathways? We really didn't know yet as far as that point in our research. The next experiment, we looked at kind of comparing incandescent fluorescent light, which is typical of what we saw in an older model grow chamber, to LED light under different quantities of red and blue light, or different ratios of red and blue light. We stuck with the sprouting microgreens again. And in the experiment, again, we used a grow pad, so very simplistic types of growing mechanisms. We germinated the seeds right on the grow pad. It was an ebb and flood kind of hydroponic system where we would put water into the tray, a certain quantity of water in the tray every day. We used a slight nutrient charge just because we wanted to see what we can get as far as a robust growth rate. And then we harvested them 21 days after sowing. So in a 3-week period, you can certainly get a lot of biomass in some of those grow pads. Here's a diagram of our lighting treatments. So we had a light intensity of 200 micromoles across all the treatments. We set it to a 16-hour photo period and a consistent air temperature of 24 degrees C. The incandescent and fluorescent light environment, using a spectroradiometer, we were able to determine the quantity or the percentage of each one of those wavelengths, the blue, the green, and the red, that were present in that light treatment. The regular light treatments, or the LED light treatments, changed as far as the percentage of blue, and then just due to the array that we had, we added green in there to some of those treatments. So treatment number 2 was 5% blue, 95% red. Treatment number 3 added in green at the expense of red, so it was 5, 10, and 85. Treatment number 4 was 20% blue, 80% red, and then treatment number 5 was at 20% blue, a little bit of green, and then 70% red. Here's a picture of the biomass when we harvested the plants 21 days after seeding. You can see that there were some relative advantages of having that LED environment relative to the regular incandescent fluorescent light environment in a grow chamber. They weren't dramatic differences, but you can see here that the 5% red, blue, green, or 5% blue under the red, blue, green environment actually had the highest biomass. Really wasn't an advantage of increasing the blue to a certain extent as far as the actual biomass production in those microgreen tissues. And these are just a depiction of the light, the plants that were actually growing under the light environments. When we look at the carotenoid pigments within those microgreens underneath those light treatments, you can see that when you compare the fluorescent incandescents to the LED light treatments, there were differences in the shoot tissue carotenoids that we measured in those microgreen tissues. And I'll highlight the ones that were probably the most significant in our minds. Increasing beta carotene under that LED environment relative to the fluorescent incandescent, and certainly increases in lutein. So those are the predominant carotenoids that you'll actually see in a profile if you were to do them on a leaf tissue. Really weren't significant differences among the light treatments. A little bit higher concentration in the 20% blue, 80% red relative to the others, but certainly a dramatic increase from just that incandescent fluorescent light environment. And most of you already know that the industry is moving more into the LED lights, and the incandescent fluorescents are starting, you're not seeing those anymore in the industry and certainly within the research environment. For the mineral elements, so we'll look at the macronutrients across those particular treatments, and really just focus in on the big macronutrients. They're really important in plant metabolism, and they're certainly important in dietary health for humans. So we can see the differences between that fluorescent incandescent environment and then the LED light environments, the differing concentrations or percentages of red and blue in the mix. So you can see the big increases in calcium and also potassium, and then magnesium as we move from that incandescent fluorescent environment into the LED light environments. Statistically different as far as the 20% blue, 80% red for most of our macronutrients, calcium especially. So that's really important when we actually talk about delivering calcium into the human body by consuming a vegetable or a micronutrient. Okay, so what I'd really like to close on, and I think this might be the most interesting data that you'll see from my presentation, is that in looking at the impact of a light environment on quality when we talk about flavor attributes. So we did an experiment where we looked at LED wavelengths and we measured the impact on biomass, secondary and primary metabolites, but also on water-soluble carbohydrates. So looking at one of the aspects of a flavor attribute. So in this particular study, it was a collaboration between a colleague I had back at the University of Tennessee, where most of this work was done, another colleague at West Virginia, Dr. Waterland, and then my graduate student, Ms. Metallo. Okay, so what we did here is that, in looking at all of the data that we had been accumulating on the impacts of blue light, I approached Fluent Bioengineering and I asked them if they could find me different LED arrays or different diodes of blue light. And so they were intrigued by my question, but certainly said that they could actually get those for me. So I had them spec and design and construct different blue LED arrays. So you can see here's a picture, and most of you have tried to take a picture of LED light. It's really hard to with a regular camera, but you can see that the arrays that they were able to produce for us, I got them to put 400 nanometer diodes on an array, 420, 450, and 470. And those represented 4 different areas on the blue light spectrum when we actually talk about that energetic value. And then they gave me some arrays of the red light at 625 and 660. And then for the experiment, we compared those to just a regular white LED array that came from Orbitech. For the experiment, we used a 14-hour photoperiod. 250 micromoles was our light intensity. And then the red-blue ratio was 40% red, 60% blue across all of those particular treatments. So what was actually differing there was the actual nanometer energetic value of that blue light. And we used kale microgreens, or Toscano kale, which is a spatulator or a dinosaur kale. A spectroradiometer readout, as you looked across all the different treatments, the black line there represents the white LED treatment, which you probably all know that's a mixture of red, blue, green diodes and the phosphor that covers that particular diode. And then each one of the peaks that you see there, starting at, from the left to the right around the 400, there's the 400 array, the 420, the 450, and the 470. And I put that red over there just to show you that of the 400 and the 420 array, they bled a little bit into the UV light range. So there was just a very slight percentage of the UV that was in those particular arrays, but that was due to the manufacturing. So we didn't know if that was a confounding factor of the experiment, but I can talk about that a little bit later. But those were the only 2 diodes, the 400 and the 420, that had any sort of UV light that was actually present in that diode. So as far as materials and methods, we grew them on a 10-20 tray in a soilless mix, so it was a shallow tray. We used 10 grams per flat across that soilless mix. We harvested them 16 days after seeding underneath those LED treatments. We freeze-dried them, measured them for nutritional components within the lab. We measured them for NDF. We were kind of interested in what it would actually do to the fiber content. And then just through a simple hot water extraction, we were able to pull out sucrose, glucose, and fructose. And then we ran them on an ELSD procedure with an HPLC, so evaporative light scattering detector, with a monosaccharide column here, which is a really fast way to actually measure those water-soluble carbohydrates. Okay, so when we look at the biomass of those microgreens, remember these were all LED light treatments, so it was just comparing that white environment to treatments that had 400, 420, 450, or 470 as far as the blue light that was actually present. And you can see from this data that when you look at the fresh mass and the dry mass, the fresh mass was certainly greater under that white LED environment, that broad white LED environment relative to just the blue light environments. Interestingly, when you look at the percent dry matter, that was actually different under the blue LED arrays relative to the white LED array. And then there was actually a higher concentration of NDF or percent NDF under the white array relative to the blue arrays. So again, when you look at NDF, what we were really looking for is maybe palatability when you actually talk about the textural analysis of it. So, if you have a very high amount of fiber in there, you could actually feel that as far as a mouthfeel when you would consume those. For the soluble carbohydrates, and this was really interesting when we measured this particular data. When you look at these differences in sucrose, glucose, and fructose, remember these are water-soluble, non-structural carbohydrates, the white LED arrays versus the blue LED treatments, so the 4, the 420, the 450, and the 470, you can see that there was a dramatic increase under the 400 and the 420 treatments relative to the white and also relative to the 450 and the 470. And I can tell you by personal observation, that was a perceivable taste difference among the LED light treatments. So they were perceivably sweeter. So there was 130 to 150% increase in soluble sucrose underneath those blue wavelengths of the 400 and the 420 relative to the other blue wavelengths, and then also the white LED environment. No significant change in the glucose and the fructose, but certainly a dramatic difference in the glucose and the fructose, but a dramatic difference in the sucrose. So as far as a wrap up of that particular experiment, the results showed that there was an impact on those kale microgreens from those different LED blue light environments. Those blue LED wavelengths that you can get from manufacturers influence biomass, the percent dry matter, and also the percent NDF, or the fiber content that was actually in there. There was actually that perceivable taste difference and a significantly higher concentration of soluble sucrose under 420 and 400 relative to the other blue wavelengths. Now, remember when I showed you that spectroradiometer output there, we really don't know yet if those lower blue wavelengths of 400 and 420, was it that little bit of UV that was actually impacting that soluble sucrose or that sucrose metabolism within those microgreens. I'm not really sure. If you look back at the data, you'll notice that the soluble sucrose was higher under the 420 than the 400, and there was actually more UV under the 400 than the 420. So we still don't really know yet. And one thing I could share with you is that the results that we saw for the microgreens, for the kale microgreens, are consistent with other species that we actually did in some preliminary experiments. So there were increases in soluble sucrose for some lettuce and basil microgreens that we grew under the same conditions. They just weren't as dramatic. So they were higher, but they weren't 150% higher that we saw in the brassicas. So that would lead to maybe a genetic impact or a genetic response from those blue light environments. So as far as a conclusion for my overall presentation, obviously most of you know by now that microgreens show a very high degree of plasticity. So with a light environment, you can certainly change morphology, you can change physiology, but you can also really impact some of those primary and secondary metabolites, which we know equate to phytonutrients in the human diet. What was really interesting to us is not only that we changed the nutritional value, but those light treatments were actually having an impact on those flavor profiles. So changing that flavor dynamic, which is usually number one when we talk about consumer acceptance, was really interesting for us. You guys know by now that there are many LED wavelengths that are, as far as options on the particular market, and it's one of the fastest moving parts of horticultural technology in my mind when you actually talk about the impact and the rapid impact that it's having on the industry. So again, in conclusion, it was really exciting to see those impacts on those flavor profiles. But, as most of you know, with research, there's still so much more to learn about these plant responses to light. And with the amount of options that are present now for researchers and also for the industry professionals, there are just so many interacting factors to look at. And that's it. Thank you for the invitation.
Thanks a lot to you, and also for showing us those beautiful pictures of Florida. I think everyone is envious here now. So our first question is, would you anticipate higher wavelengths, light, violet, and UV, further inducing some phytonutrient production, especially antioxidants/pigments?
In a word, yes. And I think that, using those UV wavelengths, they're higher energetic values. And really, when we look at phytonutrient or secondary metabolite production, most of us probably know or have a really good familiarity that, those are stress response compounds. And if you can stress the plant, you can get it to create some of those stress response compounds, whether they're antioxidants or other types of metabolites are actually in there. Now, the really key magical question is how much can you stress and how much is enough to actually get the response in that particular plant? And that's where all the research has to be done. But I would almost be confident that you could change those values with different types of UV applications or different UV wavelengths. But really, it's the concentration, again, that's going to be the important one to actually figure out.
Okay. We have another question here in the audience from Professor Marcelis. The microphone is coming.
Dean, very nice presentation. And I was wondering, you showed many metabolites, and sometimes you express it as a percentage dry weight, so let's say on a dry weight basis, and sometimes on a fresh weight basis. And dry matter percentage, you showed also in one of the last slides, can be dramatically different. That's also my experience. So what is more relevant, or why do you show sometimes per dry? Is there a reason to do it sometimes per dry weight and fresh weight? And which one is more relevant?
That is a great question, and I think that when I was in my research days, I think I kind of rode the fine line between a horticulturalist, plant physiologist, and a nutritionist. So when you look at the horticulture sort of plant physiology side, it's usually expressed as dry weight biomass because we know that equates to photosynthesis or photosynthetic efficiency. But when you want to talk to the nutritionist, they want to see it on a fresh weight basis. So they want to see it on a milligram per 100 gram fresh weight basis. And I learned very quickly once I started working in that world there that a milligram per 100 gram fresh weight roughly equates to about 3/4 of a cup. So when you talk to nutritionists, they like to see it on fresh weight because that's what people consume. And then they also like to equate it to what would be sort of like a serving size. So whenever you see phytonutrient or whatever expressed as a milligram per 100 gram fresh weight, that's usually a milligram per serving size or 3/4 of a cup. But that's a great question. And we always debated on whether we, present our information on the dry weight basis or the fresh weight basis. And it really depends on which sort of world you have your feet in at the time.
All right, then now we would have our lunch break, and also for our digital audience, maybe you also have the chance to grab some snacks or food, and then we will be back in 1 hour. Please be on time. You know, we're Germans, we like to be on time, and then we meet each other at 2:05, and see you in a bit. Enjoy. Okay, then let's start again. For everyone who has just joined, welcome. And we're very happy that you're spending your day with us digitally. I also want to mention that you always have the time, the possibility to participate in asking your questions. And later on, we will also have a poll. To use that tool, Slido, we have a link, which is in the description of the video. Right at the bottom, you can just click the link and then you are redirected to Slido, and there you can ask your questions and further on also participate in the poll later. But now let's continue with our next speaker, who is Professor for Agricultural and Biosystems Engineering at the Controlled Environment Agriculture Center at the University of Arizona. And he will tell us more about sensing and automation in vertical farms. Please welcome Professor Dr. Murat Kassida.
Thank you for the kind introduction. And hello, everyone. And I would like to thank the organizers for the invitation to join you in this exciting symposium and present. I missed the opportunity to be there in Munich together with you for usual reasons. I hope there will be another opportunity to be together in person in an upcoming event. I will be talking about sensing environmental controls and automation in vertical farming towards optimizing resource use efficiency. Modern agriculture should no longer rely on availability of natural resources and regional climatic conditions. Rather, it should effectively embrace the digital age driven by smart technologies, sustainable production capabilities, optimized and integrated systems that can enhance the crop productivity, profitability, and efficiency of resource use, enhance human nutrition and health, and decrease the environmental impact. Here, I don't mean that the controlled environment agriculture will be a silver bullet solution, but it will be a complementary farming option for profitability as the technology advancing very fast. Efficiency of the systems are improved, and with resource conserving, smart environmental controls and automation are being incorporated. The bottom line is the resources efficiency in any farming practices that is to maximize the plant growth with the minimum resource inputs that will help contributing to minimum emissions of environmental pollutants and minimum cost for the resources used. For a controlled environment agriculture system in greenhouses or in vertical farming systems, we have the inputs as energy, water, carbon dioxide, fertilizers, and labor. And we have some outputs from such system as edible biomass, that is the food outcome, but we also have non-edible biomass, which can be converted into a useful resource and reused within the production system. And similarly with nutrients and water that can be recycled and brought back into the system for reuse. We can also capture transpirational water and reuse within the system. These resource reuse and recycling capabilities makes the controlled environment agriculture systems highly resource use efficient. In a vertical farming setting, we have complex processes. In order to achieve the optimal growth and production quality and resource use efficiency, these processes should be orchestrated properly and complementary to each other rather than conflicting with each other to enable an environment for the plants to also achieve the optimal growing conditions. In order to achieve resource savings and improve the production quality, we need to consider combining controlled environment agriculture system physics with the crop physiological information. I would like to enhance the trends and the needs to include plants in the decision making and as part of the environmental control as plants used as sensors to understand the plant status and health and use that information as a feedback to control the processes. I believe this will be the most effective and sustainable way of controlling the environment in a resource towards a resource use efficient way. We have vertical farms in indoor agriculture systems, such as large warehouses with different types of hydroponic systems used, hydroponics or different types of hydroponic systems used with nutrient film technique or floating draft systems or other types of systems used, bringing unit capabilities to enhance the production or increase the yield outcome in a smaller footprint. Similarly, with container-based farming systems in a smaller scale, portability is another unique opportunities with these type of indoor vertical farming systems. However, we see also challenges in growing in indoor vertical farming operations, mainly with the amount of energy, electrical energy needed to run the LED lighting systems, but also the environmental non-uniformity in that environment because of the two-dimensional nature of these production systems. So recognizing those challenges, there are a lot of research questions ongoing. Here in the United States, we have a collaborative multi-state research project funded through the USDA, United States Department of Agriculture funding opportunity, as we're collaborating with our colleagues to co-optimize environmental conditions in indoor vertical farming settings towards resource savings, optimize resource use efficiencies, as well as improving the profitability for plant production in vertical farming systems. Our research here at the University of Arizona is conducted in our vertical farming facility that we have here at the Controlled Environment Agriculture Center campus. One of the challenges in vertical farming system is the non-uniform air distribution or airflow, leading to non-uniformity in the environmental conditions for air temperature, relative humidity, or vapor pressure deficit, or air current speed and carbon dioxide concentrations. The lack of dynamic airflow and lack of proper airflow also leads to negative effects in photosynthesis or transpiration rate or net photosynthesis and gas exchanges between crop and the surrounding microclimate. So we need to further engineer these vertical farming systems, especially the air conditioning systems, with proper environmental control applications. In a vertical farming system, because of the stagnant conditions with the airflow, this leads to a larger boundary layer around the plant canopy, leading towards an increased boundary layer thickness, preventing proper transpiration rates and also transfer of nutrients and water from the roots to the leaves and its diffusion to the atmosphere. So this prevents a proper level of photosynthetic rates as well as transpiration rates, in some cases leading to crop physiological disorders, for example, in the case of lettuce, with a common crop disorder as tip burn. So we need to maintain a proper airflow or air current speed within the boundary layer to enhance the transpiration, so we can allocate the nutrients into the young growing shoot tips to prevent these crop disorders, such as in the case of tip burn. There are various designs when it comes to vertical farming systems in terms of how the conditioned air is brought into the vertical farming space from the inlets of the air conditioning, as well as the positioning of the exit locations within that growing space. It is time-consuming and not cost-effective to analyze all of the possibilities in terms of designs of these airflow and air distribution systems in a cost and time-efficient way. So we have been using computational fluid dynamics and computer simulations to evaluate some of these design aspects when it comes to air distribution systems. With computational fluid dynamics, we are able to create a geometry representing the problem domain and also discretize that domain into finite volumes and incorporate the conservation of mass, conservation of energy, and momentum principles, along with the crop presence using porous media approach, incorporating plant transpiration as external user-defined functions to observe or to look into the details of the energy exchanges and airflow distributions and environmental parameters in a more detailed fashion. This enables us to quickly evaluate an existing system design, but also identify opportunities where we can have some improvements in terms of these system designs for the vertical farming operations. In a recent research, we evaluated various design alternatives for the air distribution systems. You may observe cases with inlets being on one wall and maybe exits being on another wall in a vertical farming system, or maybe inlets and outlets positioned in the ceiling of a vertical farm, or in this case, maybe the air is brought in from the ceiling and maybe exiting from the side walls. We also looked into cases where we are pretty much focusing on and encouraging the localized climate control, where we are able to bring in the conditioned air in each layer and creating a dynamic airflow over the canopy, and then releasing that or rejecting that air from the ceiling in a vertical farming space. I believe the localized climate control, the climate control closer to the plant canopy and boundary layer, will be the most efficient and effective way of environmental control applications in a resource use efficient way in vertical farming settings. These are some of the results of that study. As you can see, in the case of inlets on one wall and exit being on the other wall, in terms of the air velocities, we see some non-uniformity in terms of the air velocities as well as air temperature, and you can also expect these non-uniformities when it comes to the relative humidity, vapor pressure deficit, as well as carbon dioxide concentration and distribution. In this case, in a different case here with the ceiling-based inlet and outlets, you can also observe non-uniformity in its distribution in the production space. And this is another result from a different case with ceiling inlet as well as the exits being on the side walls. So as we bring the air conditioning or air distribution actually closer to the plant canopy, we are able to achieve desired air current speed range, which happens to be somewhere between 0.3 to 0.8 meters per second. That's a desired range of air current speed that we would like to achieve, leading towards an optimal condition for gas exchanges over the plant canopy in a vertical farming system or in a controlled environment agriculture system. So bringing that localized control capabilities closer to plant canopy also enables us to improve the uniformity of the temperature distribution, as you can see on this slide. Vertical farms can be comparable in terms of the electrical use or resource use efficiencies when compared to the greenhouse-based systems. Here in a study that we conducted recently, we compared the electrical use efficiencies in different climatic conditions representing hot and dry conditions, or humid and hot conditions, or cold and drier conditions in those climate zones, with different layers of multi-layers of vertical farming systems, as well as incorporating some of the capabilities with economizer modes, where you can use and leverage outdoor environments and use that to your advantage to reduce the amount of energy used for cooling and dehumidification purposes. Here, the graph represents the electrical use efficiency in terms and its distribution, the energy distribution used for lighting, cooling, and heating with a two-layer vertical farming system, as well as the lighting system efficacies. The effect of lighting system efficacies are demonstrated. When we increase the number of layers, as you can see here, from going from 2 tiers to 5 tiers to 11 tiers, we have a close to a linear increase of the electrical energy used in a vertical farming system. However, I would like to point out the effect of using economizer modes, as you can see here, in each case 11, 5, or 2 tiers, and how the use of economizer modes actually reduces the amount of electrical energy used in a vertical farming system. If you focus on this orange colored areas for the cooling applications. And that advantage is actually emphasized more or more advantageous in cooler climates, in this case, with these two scenarios in Seattle and Duluth cases. But when we compare the electrical use efficiency between vertical farms and greenhouses, we observe that the vertical farms are very comparable in terms of their electrical use efficiencies in cooler climates, in this case, in this study, compared to those cases evaluated for the greenhouses. The greenhouses still tends to be more electrical use efficient when we consider warmer climates or with high light intensity regions around the world. I would like to emphasize that we need to focus on resource use efficiency and electrical use efficiencies, for example, as our metrics to compare different types of environmental control strategies and applications. And here, in research that we conducted, we evaluated different light intensities and daily light integrals, as well as the production or the yield outcomes with the electrical use efficiency. As you can see, as we increase the amount of light intensity and daily light integrals, we are also able to increase the production outcome. However, the demand for electricity is greater. And we can consider lowering the light intensities and daily light integrals with an expense, of course, a penalty over the yield. However, when you consider the electrical use efficiency, the results are fairly compatible. So the next step should be the decision making in terms of the market expectations, production qualities, but also considering the electrical use efficiency and the profitability economics of running a vertical system, so we can achieve the expected quality and yield and quality attributes, but also achieve the economics as well as the profitability in a vertical farming system application. Our recent studies are indicating that with carbon dioxide concentration and enrichment based on different daily light integral levels, 850 ppm seems to be a threshold that is an optimal level with lettuce crop, in this case, that can be considered in a vertical farming system. As we increase the daily light integral or the light intensity, as well as with elevated carbon dioxide levels, we also observe the occurrences of tip burn issues because of the speed or because of the increased rate of growth with the plant and possibly leading towards tip burn occurrences. So those are the types of observations that we need to consider when it comes to environmental control applications and control strategies. Automated crop diagnostics and decision support systems are among the emerging automation technologies. These technologies have been in maturity in greenhouses, but we are also seeing integration of these applications in vertical farming systems. One possible enhancement could be to use speaking plant approach, basically using plants in the decision making as part of the feedback mechanism to optimize resource use efficiencies, optimize use of space and also energy and other resources that we consider in a vertical farming system. Computer vision and machine vision modeling, predictive modeling for crop yield estimation could be an integral part of such biofeedback-based or phytometric feedback-based control capabilities. If we can combine the direct measurements from a computer vision, machine vision system, along with predictive modeling capabilities, we can easily identify, or in a practical way, we can identify the deviations from a normal accepted growth leading towards an improved environmental control applications and detect anomalies and make decisions in terms of the actions that are needed in a vertical farming system. We considered that approach and developed a multispectral plant health and growth application in a greenhouse system. We are trying to incorporate also that application in a vertical farming setting where we are interested in monitoring plant temperature, texture, color, as well as other crop indices to identify plant stresses in a timely way and incorporate that into the resource use in indoor agriculture settings. Our efforts currently is focusing on phytometric feedback-based decision support tools, where we are able to consider the environmental variables as well as the plant characteristics, plant canopy architecture to determine and to predict the yield outcome, and based on that, make a decision in terms of the control of the environment. Such approach can be used, for example, as plants to control their own environment when it comes to light intensities or maybe later spectrum, but also other environmental control capabilities. Using these modeling approach, we are able to analyze the effects of air temperature, carbon dioxide injection or the concentrations, as well as data light integrals in a more time-effective way without doing so many of the cases or scenarios with experimentation. Of course, we need experimental data to validate and to develop a confidence in terms of the models we use. However, we are using this modeling approach to identify resource optimization, resource savings with various what-if scenarios. For example, in this case, in this graph here, the consideration to reduce the daily light integral of the light intensity as we alleviate the CO2 to achieve a desired and acceptable yield. That approach, as you can see here in this example, is leading towards about 14-15% electrical energy savings when you consider dimming of the lights to an acceptable level as we elevate the CO2 applications. We continue to evaluate those what-if scenarios also in a way that carbon dioxide maybe can be enriched, can be used to enrich the environment, not during throughout the production phase from transplanting stage to the final harvesting stage, but maybe at different levels of injection at different growth stages in a more dynamic fashion. We are focusing on integrated vertical farming system applications as we are considering to leverage a waste stream and make it a resource inputs, valuable resource inputs in another production system in a vertical farm. And in this example here, we're considering the CO2 emissions that are significant from a mushroom system and being able to harvest that CO2, excess CO2, and then use it in a vertical farming system to reduce the amount of CO2 coming from other types of sources. But also we can capture the water from the environment in a leafy green production facility and then provide that into a mushroom production system where we need elevated higher levels of humidity in that growing environment. We are going to see further integration of robotics and automation in vertical farming space, because labor is number one resource input, and it will demand for further automation on robotics application for various processes in a vertical farming space. With robotics for harvesting or crop scouting, health monitoring applications will be further incorporated into vertical farming applications. Information, IoTs, as well as cloud computing, augmented or virtual reality applications are moving into indoor vertical farming space to help the grower and also support their production capabilities in indoor agriculture systems. Artificial intelligence is being incorporated. It has been demonstrated that it is effective in a greenhouse-based controlled environment agriculture system. Here, I don't mean that the artificial intelligence will replace the green thumbs, but it will be a significant tool, an important tool to empower the plant as well as to empower the grower so we can achieve profitability as well as optimize resource use efficiency in vertical farming space. Plant phenotype is a function of its genetics, environment, as well as the management practices. We can enhance the environment and through environmental control, the management through growers' capabilities or the system capabilities, but we also need to focus on the genetics of the plant. There are great opportunities and the need to consider new varieties through breeding. So the plants are, the varieties are developed and suited for indoor growing conditions and that are able to use resources in an optimal way. So we'll see further growth and development and innovation in that space as well. In terms of the trends in environmental controls, we will need to focus on plant-centric environmental controls, objective measurements versus green fingers. We need to focus on detection capabilities, timely detection capabilities with sensing and monitoring. We need to focus on seeing the invisible, bring in remote control capabilities with IoT applications, and also further develop decision support systems and autonomous controls to enable the operator and the grower. The data, the significance of data will be further emphasized because we have various processes where the data could have a high impact in terms of crop quality, energy and climate control, labor planning, nutrient management, the workflow, and other processes. So we will see more data-centric sensing, monitoring, and environmental controls further incorporated into vertical farming space. Speaking plant approach will contribute to optimal resource use and smart environmental control system applications. Diverse set of sensors already exist commercially, but we need to focus on what crop-related information we want to know and interpreting the results and readings and signals. Being able to identify stress symptoms have been a bottleneck and a challenge when it comes to speaking plant approach. I believe that the machine learning and artificial intelligence will play a significant role there. We need to focus on, again, the sensor capabilities, spatial and temporal variability of the signals being processed, and also sensor fusion modeling and expert inputs as part of the environmental control applications. As we are focusing on growing indoors in vertical farming space for earth-based applications, there are also challenges exist as we consider colonizing space and also space horticulture in microgravity settings or on Moon and Mars surfaces. So the vertical farming type of systems, indoor agriculture, controlled environment agriculture, will play a significant role in those localities as well. And as we have also projects trying to develop efficient water and nutrient delivery systems for crop production in vertical farming systems. I would like to encourage our participants to consider joining
Mr.
us
Kasira?
for the 40 dialogues on vertical farming offered by the International
Mr.
for the
Kasira, I'm
Society
very
of
sorry to
Horticultural
interrupt.
Sciences.
Could you please
As
come
we
to an end because
collaborate
we're
with
already
our
over the
colleagues
time?
Leo Marcellis, Francesca Orsini, and we have exciting presentations coming up in the coming weeks. Thank you for the opportunity.
Thank you very much. Maybe we can do one quick question so that we can also address a question to you. Which boundary conditions are you using in pre-processing?
In pre-processing, can we repeat the question, please?
Which boundary conditions are you using in pre-processing?
I believe the question is referring to our modeling approach for the computational fluid dynamics, and our boundary conditions are the environmental variables in that production space, such as air temperature, humidity, or the physical conditions, the radiation or the heat transfers from, for example, lighting systems. So those are the kind of boundary conditions that we consider.
Thank you very much. And again, sorry for interrupting, but the next speaker is already in line and waiting. Thank you.
Thank you so much.
So let's continue with our next speaker, and he is telling us more about the big challenge of higher energy use and about the LED technology and maybe possible solutions. Please welcome, with a digital applause, Dr. Morgan Patterson, who also works at the Solid State Lighting Services. Please go ahead.
Thank you. Thank you for inviting me to speak. I wish I could be there in person, but unfortunately, that's not possible at this time. So I'm going to talk about the energy for vertical farming, but more particularly the energy that's used for lighting and LED technology. There are LEDs have gotten a lot better over the last few years, and they're enabling new applications like vertical farming, but the prospects for LED improvements are not infinite, and I'm going to go through some of that during this talk. Just a little bit about me. I come from a materials science background, LED technology in particular. I work through my consulting business, SSLS, for the U.S. Department of Energy, developing strategy to reduce lighting energy consumption. And then also I work as an advisor to Lux Lighting, which is a horticultural lighting company, which is doing developing some very nice products, but mostly for marijuana growth applications in the US. And then also help with a smaller initiative, the Clean Lighting Coalition, which is working to remove mercury-containing fluorescent lighting. So with that, I'll get going. So LED technology's really been important to the world. It's reduced energy consumption, lighting energy consumption by a significant amount. In 2020, it's calculated that LED technology has saved so much energy that equates to something like $15 billion in the United States. So this is really a huge shift in the energy budget for the world and in the US, but also shows the proliferation of a new lighting technology in a very large application. So this is a big story, and I think this is important context for vertical farming because this technology has been developed for general illumination, and now the benefits of the technology can be applied to farming, to controlled environment agriculture, and to vertical farming in particular. So this is, it's all, I guess, different parts to the same story. But what's interesting to me, what I really enjoy is is connecting the technology capabilities of LEDs to new applications like controlled environment agriculture. So first thing I want to get into is a little bit more about the underlying technology of LEDs. So there's kind of two categories of LEDs. There's a phosphor converted white LED, which is shown in the upper half of the slide, and that means that a blue LED is used to, what we say, pump a phosphor, which is an optical down converting material, to generate white light. So the blue light from the LED, a portion of the blue light from the LED gets converted into green, yellow, and red portions of the spectrum to generate white light. So in general illumination, just about every single LED light that's out there is a phosphor converted light. So that's kind of the core of the technology right there. And then on the lower half of the slide, you can see that there's also color mixed light. Different LEDs directly emitting at different colors can be combined to make white light or used individually in this application, vertical farming. That's often the case. But it's really important to understand that not all of the LEDs have the same efficiency at the different colors. So the panel on the lower right portion of the slide shows that blue LEDs have the highest efficiency, and then there's a low efficiency area around green, and then the efficiency comes back up around red. In the LED business, in the LED industry, or the research part of it, we call this the green gap, where efficiencies are much lower for direct LED emitters in the green range. That's not the worst thing, because we have very good phosphors to convert blue light to make white, but it is a limitation, and it's important to understand that these LEDs have different efficiency limits based on their materials properties, and that becomes a consideration that translates into horticulture and vertical farming. So this is some information, some data from the Department of Energy work that I do, and it just shows how LEDs have improved over time. The upper figure shows how white LEDs have improved, and there's a dashed gray line there which shows the potential for color mixed white LEDs. So this information is expressed in lumens per watt, which is the figure of merit, or the characterization you use for human general illumination lighting. But the point is that the LED materials themselves have gotten quite a bit better and more sophisticated. The crystal growth processes for creating the LEDs have gotten better. Phosphors in the packaging of LEDs have gotten better. And these are all advancements that can be leveraged by vertical farming. On the lower panel, we can see that the individual colors are fit as though they can all achieve 90% power conversion efficiency in the future. That's a assumption that we use with the Department of Energy, and we say, well, we don't know that that's not possible, but there are some very significant materials challenges to improving each of the colors to get to that level. And what you see in that panel is right now, as I showed earlier, the green and the amber efficiencies are much lower than the blue and the red. And in fact, blue LEDs have, depending on the operating conditions, can be over 95% efficient. But that's kind of that impractical operating conditions. So next, kind of jump into the horticultural side here. Of course, when we're talking about lighting, LEDs and lighting for humans, we express performance in terms of lumens, which is based on the eye response spectrum, the eye response. And we use lumens and lumens per watt, the luminance values of lumens per meter squared. And we have an evaluation metric called the luminous efficacy of radiation, which shows you for a given spectrum of light, how many lumens would we get out for 1 optical watt of power in the light. So all of those metrics can be translated into horticultural lighting metrics. Lumens are equivalent to the photosynthetic photon flux, which is micromoles of photosynthetic photons per second. You have the efficacy of that process or the generation of those photons, which is micromoles per second per watt, which equates to micromoles per joule. And flux density is micromoles per second per meter squared. And then you have the photosynthetic photon efficacy of radiation, which is for a given spectrum, how many micromoles per second could be achieved for 1 watt of optical radiation. And I'll go into that into the next slide. There's some other differences with horticultural lighting. In general illumination, like I was saying, it's almost entirely a phosphor converted architecture that's used, even to get your red light within the white spectrum. With horticultural lighting, what I'm seeing, specifically with my work with lux lighting, is dramatically increased use of direct red LEDs. And this has always been a part of horticultural lighting. In the early days, there were blue-red spectrum. Then I think there was kind of more of a white spectrum with some red supplement, and now I'm seeing much more use of red. And what this has done is it's reinvigorated research in red LEDs, and red LEDs have gotten much better in the last few years. Previously, red LEDs were mostly just used for street lights and automotive tail lights and some architectural lighting applications where efficiency is not critical. But now in horticulture, definitely efficiency is critical. And that's going to be an interesting dynamic to see how that comes back into general illumination, since those light sources have become much more efficient. Definitely, the color requirements are different for humans compared to plants. It's kind of obvious, but it's important to acknowledge that. There is a greater demand on reliability for horticultural light fixtures, since if you have light output depreciation or light output failures, that's a much bigger deal for a production environment than for a general illumination environment. And the last point is that in this industry, because it's a production environment, I'd say there's less constrained. It's less constrained by the form factors of the previous technologies and the limitations of the previous technologies. So that's a nice benefit. Where it does come down to dollars and cents and productivity. There's not an attachment or a reluctance to reconfigure your environment to accommodate new form factors. So this figure just shows the physical limit of LEDs in terms of photosynthetic photon efficacy of radiation. For a given wavelength of light, for 1 watt of radiation at a given wavelength, there is a maximum number of micromoles per second that can be achieved in a perfectly efficient situation. So this is just a fundamental physical limit. it reflects the fact that, as was mentioned in a previous talk, the shorter wavelength photons are more energetic. They contain more energy with them, so fewer of them are needed to per energy level or per watt. So you just get fewer higher energy photons generated for a watt or within a certain watt of a given watt. So extending this out, you see that the red wavelengths beyond 600, you get a higher potential photosynthetic photon efficacy than for the blue wavelengths. I think everyone pretty much gets this, but there's some pretty important implications here. One feature or aspect of the technology and the use of LEDs for farming, as a light source for plants, is that LEDs are most efficient around 450 nanometers and 660 nanometers. And that corresponds to the peaks in photosynthetic absorption within plants. So that's some sort of happy physical accident. So here we go back to that other previous figure, which shows the spectral power distribution and the relative power for the different wavelengths of light, different colors of light. In the left side, you see the relative power efficiency for different colors. And on the right, you see the relative photon efficacy for the different colors. And the main point here is that the reds are equivalently photosynthetically efficacious as the blue LEDs, and even at this point, more efficacious in terms of photosynthetic output. So, that's those limits of the upper limits of the the photosynthetic efficacy of the LEDs. That is a limitation that's not possible to get around with LED technology or any lighting technology for vertical farming. So there's not infinite room to improve LEDs. They cannot be improved beyond those limits. And then I've touched on this already. There are material science and semiconductor device constraints and limitations on the performance of the LEDs. And again, this is apparent just based on the fact that blue LEDs are the most efficient, and then there's red, and then green, and then amber, and that highlights the material science and device challenges for making these different emitters of LEDs. This whole lighting revolution, LED revolution that we're going through, which is now part of the controlled environment revolution, agricultural revolution we're going into, was enabled by the invention of the blue LED by Shuji Nakamura and a couple of other researchers in Japan. And for that, they won the Nobel Prize in 2014 for this invention, which pretty much came about in the 1990s. But there's been a lot of development since then to get these devices to the point where they're at now. So there's, like I said, there's material science challenges, there's fundamental issues when making LEDs. Blue LEDs are made from indium gallium nitride semiconductor materials, which prior to the use of that material in LEDs was not really a semiconductor that had been used for anything. Red LEDs have been around longer. They're made from aluminum, indium gallium phosphide alloys, and require a different production process, different crystal growth process than the InGaN blue LEDs. And then green are typically InGaN LEDs when they're used. And the problem with green is that you start to stretch out the crystal and have problems with crystal defects. So just was going to touch on that. If you see the point between you have the GAN point on this figure, and you have an indium nitride point, and you see the rainbows of colors. The line between the gallium nitride point and the indium nitride point show that this semiconductor alloy can be made to emit light at any of these colors. But as you go from blue towards red, you induce crystalline property defects or in the devices. You come to the limitations of the crystalline growth process, and your efficiency degrades dramatically. Now, it is possible to make indium gallium nitride red LEDs. There's a lot of work in improving the green LEDs. A lot of this is driven by display applications where they'd like to have full color displays that are made on a single wafer by the same growth process using the same material set. I was lucky enough to have a cup of coffee with Shuji Nakamura on last Saturday, and he thinks he might have invented a way to improve red in GaN LEDs that he was telling me about. So, this is still an area of research, but there really are some fundamental materials properties limitations in the LEDs themselves, which are going to limit the ultimate performance of the LEDs when they're used for vertical farming applications. Another material science limitation in LEDs, and blue LEDs in particular, is what we call droop, and that's just where you have uneven distribution of charge carriers within the nanostructures that have been engineered in the LEDs to confine charge carriers to make them more efficient. And so what this just shows is you have too many charge carriers bunching up into one quantum well, which is a nanostructure within the LED. And when there's too much concentration of those LEDs, they tend to recombine, or not LEDs, sorry, those charge carriers, they tend to recombine non-radiatively. And so, of course, as you pump higher current into these devices, they bunch up more, and then you have worse droop, or Auger recombination is the actual name for it, but everyone in the industry just calls it droop. So, there's, like I said, there's materials limitations, and there's physical limitations to how much better LEDs can become. That said, this is a talk from a researcher, or a figure from a talk from a researcher at Osram, and they've made quite impressive progress on improving red LEDs. And this is just a schematic of their red LED structure with various features to improve the performance and to balance the optical considerations of getting the light out of the LED with the electrical considerations of getting the charge carriers into the LED. And so a couple of notable features is the surface roughening to improve light extraction efficiency. Looks like he talked about some improvements to the mirrors and optical structures within the LED. And what this has enabled, practically speaking, from my work with Lux Lighting, is the availability of very, very efficient red LEDs now. So, taken together, I've translated that LED performance forecast and historical performance figure from earlier slides to horticultural metrics. And so, the physical limits show on the right side, which is the maximum photosynthetic photon efficacy these LEDs can achieve if they were 90% radiatively efficient. And so different colors, of course, have different physical limitations, and then the current performance of these different colors of LEDs is based on material science challenges and development effort for the different colors. One thing to note about this figure and this data is that it is constrained to a one operating current density, which is something we do at the Department of Energy so that we make sure we're tracking progress and efficiency performance and not just changes in how LED chips are put into packages and how hard they're driven. So this is just, it's constrained in that way to make sure that the data is more consistent. But on the flip side, in practice, LEDs are no longer run typically at 350 milliamps per millimeter squared. They're run at a lower current density, and then so as a result have much higher efficiencies than actually what's being shown here. Not much higher, but meaningfully higher efficiencies. For example, I can buy a deep red LED right now that's 4 micromoles per joule. That's very good. And the whites are better than, they're at about 2.9 right now. So, and that'll be something here on this slide. So in this slide, it's just a status for LEDs right now, practical LEDs used in vertical farming. The efficiency is quite good for most of these LEDs. And, the efficiency of the LEDs themselves can get better, but not so much better. We're starting to run out of headroom for improving the LEDs. Just some images there of some red LEDs on a board and a deployment of lights in a marijuana operation, which is relevant. I'm gonna tell you why here in a few minutes. So with the LEDs, once you have the LEDs themselves, the LED performance is degraded, the other efficiency losses are induced when the LEDs are put into a light fixture, and there's power conversion losses from the power supply, which maybe 6 or 10%. There's optical losses in the fixture, which can be 6 or 10 or more %. LEDs become less efficient at higher temperatures, even though they are pretty robust at different temperatures. There are efficiency losses. And then one thing that's unique about the horticultural application is that in almost every fixture I've seen, LEDs are underdriven. So there is not a problem with droop, and that does enable some better performance. So I just did a little mathematical example of using a cool white LED and a deep red LED combination. The cool white is 2.9 micromoles per joule, and the red LED is 4 micromoles per joule. And I've used 75% of the power going to the red and 25% going to the cool white, which enables a combined efficacy of 3.7 micromoles per joule. And then when you multiply that by fixture losses, you end up with 3 micromoles per joule. And that's a very good result. Definitely, I think we've all seen products with higher efficacy than that, but this has a little bit nicer color, doesn't use blue LEDs. It ends up that the fixture is 60% radiatively efficient, which is quite a good result. And that's what the fixture looks like in that image there. That's showing my colleague Matt taking some measurements. So it's, the math works out, but these are all unavoidable losses that stack up when considering lighting products. Also with vertical farming and agriculture in general, we have to think about the cost, the reliability of the fixtures, their lifetime, how the light's depreciating over time, how easy these products are to connect and install, what the products look like, how they resist water and moisture, and the optical distribution of light. Are we providing nice?
I'm sorry that I need to interrupt, but we need to come to an end, please.
Sure. my final point, oops, not that final, is just that with the Department of Energy, we've done an analysis of how much energy savings there could be if controlled environment agricultural applications are transitioned from conventional technologies to LED technologies. And in this analysis, we found that in the US, there's approximately, this is for 2019, approximately 20 acres of vertical farm area within the entire US. In the third column there on the right, high intensity sole source shows, that application is another name for marijuana, and that's quite a bit more area being used and quite a bit more energy, which is the gigawatt hours per year. So just wanted to say, with vertical farming as it grows, this is 20 acres compared to perhaps 280,000 acres in the US being used outdoor for growing lettuce and leafy greens. There are significant energy considerations, and there's not significant headroom to improve LED technology to become more efficient. So it's just something that really needs to be considered as this application moves forward. And I'll end it there. Thank you.
Thank you very much. Thanks, and have a wonderful day. So when we talk about vertical farming, we're always talking about sustainability, but it's also very important to mention that, of course, the energy use is higher. So our next speaker will tell us more about if vertical farms are really sustainable, and I'm really much looking forward to that presentation. His name is Dr. Francesco Orsini, and he is employed at the Department of Agriculture and Food Sciences at the at the University of Bologna as associate professor. There you go.
Yeah, you can hear me. Okay, so yeah, well, thanks to the organizer for having me here today. My presentation is going to be about the use of resources in vertical farm and specifically in the use of land, water, and energy. So, yeah, my presentation is actually going to follow this outline. So we start with land, talking about the efficiency, so the amount of food that is produced per unit of land devoted to the vertical farm, and then water use efficiency, energy use efficiency, and then lead to the conclusions. From a land use perspective, well, first we need to see where these type of systems take place. So if traditionally open field cultivation is found in rural areas, and then we have greenhouses that are normally in rural and peri-urban areas, but recently also inside cities, as for the cases of rooftop greenhouses that you see in the image, vertical farm can basically take place in any of these locations. Also, as we have seen in the former presentations, we have different typologies of vertical farm with different size, going from the small container type plant factories to the warehouse type plant factories that take place in large warehouse systems. This also provides for the opportunity to recover potentially abandoned buildings that are not anymore in use, which provides support to urban regeneration and also potentially reaching zero land use, zero new land being used. On the other hand, this type of systems is quite rare in reality because the presence of columns or the limited age of the old warehouse systems, as well as the cost for meeting current regulation, normally hinder. But I still think that we should consider recovering land that has already been paved and install this type of systems. In general, when we are comparing different type of growing solutions, we see that vertical farms allow to highly increase the productivity per unit of land devoted. So if to produce 1 kilo per day of lettuce, we need more than 90 square meters in a open field. This goes down to 9 square meters in a greenhouse system and can be 0.3 square meters in a 10-layer vertical farming system. Also, given that we are paying the cost of electricity to provide light to the plants, strategies are under implementation on the way to maximize the amount of light that is intercepted by the leaves. So, for instance, if we want to consider a leaf area index of 3 as a viable amount of leaves in a certain surface, in a vertical farm, if we want to have optimal interception, then we can also consider to apply different planting density throughout the crop cycle. And this will allow to keep this constant leaf area index and therefore an higher interception of the light. I'm including this chart here. It's quite a provocative chart that we have elaborated a couple of years ago in a review paper that was published in the European Journal for Horticultural Science, where we compare literature data on the land surface use efficiency of different crops, including also reference of the land that is used to produce animal products. So we see that in a vertical farm, we are going much better than any other growing systems for what refer land surface use, and we have way higher efficiency also as compared to animal products. Moving to water use efficiency, well, a step back. We have been performing research mainly using red and blue diodes, and we know this. We have been talking about this already today. This corresponds to the peaks, to absorption peaks of chlorophyll, and generally it is considered that red and blue radiation is more efficient in fostering photosynthesis. So we have been working also together with Leo on defining what is the optimal red and blue spectrum to allow for improved water use efficiency, if this can play a role on the plant transpiration and the photosynthesis. And we observed that by increasing the RB ratio, so the red component as compared to the blue one, we could improve water use efficiency by reaching an RB ratio of 2 to 3 in both lettuce and basil, while higher RB ratio would then result in lower water use efficiency. Then we used the same spectrum, this RB3, and tested different light intensities, and we observed that as we increased the light intensity from 100 micromoles per square meter per second to 200 micromoles, we would have increases in water use efficiency, which were then not anymore found as light intensity was further increased. So we know then that light properties affect also the plant capacity to make an efficient use of water. And then, of course, we have seen also this in the morning. There are also new evidences that other light components are important for the potential inclusion in vertical farms. We know that green and far-red radiation can also foster photosynthesis by penetrating deeper in the leaf tissue, but also in the canopy, thanks to the fact that they are reflected by the crop and might foster photosynthesis in the lower leaves. And we also have seen, I think it was the presentation from Leo, that also white light is finding interesting application because of the easier possibility to work inside the vertical farm. It's less tiring and allows for better detecting potential diseases, for instance. Although yield is generally the same as in a red-blue spectrum, but with higher energy consumption. So we did some experiments this year. This is not yet published, but I'm testing how lettuce plants, both red and green lettuce plants, would respond to different spectrum at a constant light intensity that had either a RB ratio of 3 or a RB ratio of 3 added with a green or a white component. And we did not encounter differences in yield But apparently there was, in one of the two lettuces, the green one, an higher value of water use efficiency as compared to the RB spectrum. Again, bringing this value here, here you see how water use efficiency might vary in indoor farming systems as compared to the greenhouse, the field, or against animal products. But beside the values that we see here that are mainly associated to playing with the light properties, then there is also another important opportunity when you are in a vertical farm that is basically recovering the water that is transpired by the plants, and basically out of the 100% of water that you provide to the plants, less than 20% goes generally in the plant biomass or in moisturizing the substrate. You have some losses for ventilation, but then you can dehumidify the atmosphere and place this water back into the system. This is certainly a big opportunity. We should take care of the material that we're using in our climate control system, because glues used for piping or the panels that are inside the vertical farm, they might emit volatile organic compounds that then can be sent back to the plants if you irrigate this water again. Energy use efficiency. This is a constraint, actually, still a constraint. We have seen also in other presentations, one of the main issues is how to reduce energy use in a vertical farm. Variable cost account for about 1/3 of the total cost, and 30% of the total cost is actually going on electricity. This could be 50 to 60% going into lighting, and then there is also another major part that is actually related to the climate control system, and the least lamps are efficient, of course, the higher will be also the cost for climate control, because this is actually related to the heat that is dissipated by the lamp. There are ways for improving energy use efficiency also in terms of climate control, for instance, by integrating a cogeneration system instead of an heat pump system. This data from an experimental plant factory in Japan show that 30% of the energy could be saved by moving to this technology. And then there are possibilities also in the diodes, as we have seen in the last presentation. I will skip this, but yeah, as you see here, when you have LEDs with longer wavelength, generally more photons are released. One issue we encountered in some experiments we did some time ago was actually that depending on the temperature of the diodes, then also the efficacy changes. And normally when you move from the 25 degrees Celsius at the junction temperature to a higher temperature, then blue diodes might even increase their efficacy, but you see a decrease in the efficacy of the red diodes. Last year, we released this publication that is an inventory of about 400 LED manufacturers, and comparing data, declared data by the companies of 1,500 lamps. And although it is true that we are going toward efficacies that are above 3 micromoles per joule, the large majority of the manufacturers are still selling lamps that are between 2 and 3 micromoles per joule as efficacy. These are the experiments I showed before. They also provided data in terms of energy use efficiency. For instance, when moving from a predominantly blue spectrum toward a predominantly red spectrum, it was possible to increase energy use efficiency in both lettuce and basil, with highest value when RB ratio was equal or above 3 in lettuce and equal or above 2 in basil. And again, using a RB ratio of 3, that was a good spectrum for both crops, we observed that light intensity had an effect, of course, on energy use efficiency, but this was actually highest when we have 200 or 250 micromoles, but not beyond that. because then the increase in energy consumption was not counterbalanced by increase in yield. These data were actually partially presented by Leo before. We tested the photoperiod response in different crops. And basically by increasing, starting from a long photoperiod of 16 hours, but then increasing it up to 20 or 24 hours of light per day. And the highest energy use efficiency in most crops, in lettuce and basil and chicory, was when we had a 16-hour photoperiod, but apparently in rocket we could also go for an higher photoperiod and preserve energy use efficiency. We are also working on pulsed light. This is a topic that has been coming out today also. And we had some preliminary experience last year using lamps with different switching frequency. And although no difference in yield would be observed between the different switching frequencies, it emerged that when you have a high switching frequency, so the light is being on, it seems more on also to our high, you get higher antioxidant values, while if you reduce the switching frequency, then you can have higher energy use efficiency. Energy is also a matter of the type of energy sources that you have. And there is this study from Michael Martin in Sweden, where they compared the sustainability of a vertical farm depending on the type of energy mix they could be using. And this is something that should be taken into account, as it should be taken into account how changes in the cost of energy will affect the sustainability of the system. Again, this chart showing literature data on energy use efficiency is quite interesting, because differently from the previous ones, we see now that vertical farms have performances that are not as good as the other growing systems, and might be actually similar to those of some highly impacting animal products. I wanted also to bring some data on emissions, but the time limit is actually constraining here. But as you see, and this is mainly related to the energetic cost, vertical farms have also issues in terms of the emission. But this is something that if we look at there are very few papers on environmental assessment of vertical farms. And if you look at those that were released some years ago, and then the more recent one, you see already a trend with a decrease in the impact associated to production. So I'm quite optimistic that this can also improve in the future. Going toward the conclusion, we recently implemented an inventory of vertical farms, finding about 230 facilities in the world, and this was actually mostly concentrated in the US or in Asia, but also in Europe we had a certain amount of vertical farm existing and operating, mainly concentrated in the north of Europe. What is happening in the Mediterranean is that this type of system is finding also some application. We have 3, 4 from last week facilities also running in Italy. They are already in the market. You can find them, if you can find their products in the supermarket. And what we are currently working on is to understand how the local factor actually affect the sustainability of the system. Local factors are several that will determine whether the technology is good or not in a certain location. And we have recently developed, in the framework of a European project, it's called Foodie, Food Systems in European Cities. We have developed a prototype vertical farm in our university, where we are testing, we can modulate different settings of the environments, including dimmable LED lighting, providing up to 25 concurrent lighting treatments. We have hydroponics and aeroponics systems with 6 different treatments for fertigation that can be applied. And we monitor all the inputs that get into the system, from the water to the electricity that goes into each of the components, so we can elaborate resource use studies by playing with the variables inside the vertical farm, understanding what changes in the resources use. And we are actually also going to implement environmental and economic studies of this facility. So to conclude, and I think I was on the time, yeah? We can improve water use efficiency, playing with the light, and also, that's very important, condensating the air humidity. We can reduce the land use. There are strategies that can be set in place to do so. And we can also reduce energy needs. This can come from the use of highly efficient environmental controls, climate control system, but also with appropriate use of light in terms of spectrum, photoperiod, and intensity. This was already in the presentation from Murat before. We have worked on this series of webinar on vertical farming. They come out every 2 weeks, so if you look for it online, you can get there. There are also several other episodes that are coming in the coming months. Yeah, that's all.
If you want to have some water, this you can use it, okay?
Okay, yeah.
Perfect, thank you very much, first of all. Are there any questions in the audience here? Then we start with the live questions, but I also have some further questions here on the tablet. But do we have questions from the live audience yet? Okay, then let's start with the ones that I have on my tablet. Do you have information or considered exploring the effects of light on nutrient use efficiency?
Yes, well, we included some figures also on nutrient use efficiency in our papers on basil and lettuce. We did not find a very big difference between different spectrum on the nutrient use efficiency. But what we want to do now is that we have this experimental facility where we can combine different plant nutrition treatments and lighting treatments. So this is something that we will work on in the coming months.
Perfect. The other question is, do environmental impact studies on vertical farms generally include also emissions for infrastructure, or do they mostly only consider energy fluxes?
Well, this depends on the boundaries of the system. For instance, some years ago, we released a study where we only targeted how different spectral management would affect the environmental emissions. So in that case, we only concentrated on, we had very tight boundaries, but normally what you want to do is to control all from cradle to gate, or even to reaching the consumer. So there is this study that was performed in Japan. I think it's also cited in the presentation, where cradle to gate approach was applied over a large number of vertical farms and greenhouse systems. So this is what should be done. Okay. So to consider the whole system also, not only the operation cost.
Thank you. Do we have some more live questions here? One more look to the audience. Okay, then thank you very much.
Thank you.
So now we come to the point that I've mentioned so often today. We come to our poll where we need your participation, not only your live participation, but also the digital one. It's also in Slido. So here you can now see the QR code that you can scan, and then you are redirected to the Slido poll. And we have 2 questions for you. So the first question that we want you to answer, and again also for our digital audience, if you have not the link and you're not able to scan it, then you can just go underneath the description of the video. There you will find at the very bottom, you will find the link to Slido, and then you can participate. There are 2 tabs. One is for poll, one is for Q&A. Okay, so the first question is, from your perspective, what are the biggest hurdles and challenges with vertical farming? And there we want you to just type in your biggest hurdles from your perspective and challenges. So it's not a multiple choice, it's just that something that you can type in. We're also waiting a little bit, and here we can already see that energy was named a few times because it's in the middle and the biggest one. Also, the energy costs were not only mentioned once, but further times. Perfect. So this already works perfectly. We will wait also a little bit for everyone to participate, and we will afterwards discuss the results in our panel discussion. We just want everyone to have the chance to participate. And then, once we are done with this question, we leave it a little bit open. We have a second question, and there we want to know from your perspective, which opportunities and use cases do you see for vertical farming? You will have time to answer to those questions because we will now have a short coffee break for half an hour, and then we will be back here in 30 minutes with another presentation, and afterwards we have our panel discussion and also discussing your answers here. So 2 questions, coffee break, and then we see each other. Okay? Thank you. we're not on time anymore, I suggest that we start again. One more hint, because with the second questions, that didn't work out so well, so we try it again, because we also want to know from your perspective which opportunities and use cases do you see for vertical farming. So of course we also want to see the positive sides and not only the negative or rather challenging sides. So please take this QR code and participate so that we have your answers here too. All right. But now I also want to introduce our next speaker to you, and now we will talk more about the future of vertical farming and which visions exist there. Please welcome Kati Steppe. She is a bioengineer, doctor in applied biological science and full professor at the Ghent University. The stage is yours.
Thank you very much for the nice introduction. Also, thanks for being here, because it's a pleasure and really a great honor to share and talk about visions for vertical farming. Okay, so vertical farming, and more in particular, the multi-layer crop production systems, they offer great opportunities, and they enable us basically to create new innovative applications. And you see here, these applications can be on water, they can be on land, but they even can go beyond the sky, being in spacecrafts. And it goes without saying that the applications that we need to apply tomorrow, that we need to develop them today. And therefore, in 2017, the University of Ghent, Inagro, and the Auction Reo, they decided to launch the Ghent Agrotopia Endowed Share. And by putting together compelling research and developing visionary concepts and novel technologies, the ambition of Agrotopia is basically to tackle and to resolve the critical grand challenge of our current generation, being producing more food on less land with less resources. So Agrotopia wants to increase sustainability in greenhouse horticulture, wants to integrate principles of circular economy and increase the rentability in urban horticulture, propel the implementation of new innovations, and pioneer in vertical professional high-tech urban horticulture. So Agrotopia wants to inspire, wants to propel, and wants basically lead the way in Belgium, but also abroad and on a global scale, let's say. So for that reason, Agrotopia and leading researchers from our faculty, the Faculty of Bioscience Engineering, came together, joined in the Agrotopia Endowed Chair, and basically Agrotopia became our platform for synergistic and complementary research. And several knowledge leaps were already gained, so we talked already about it, and also at our university we deal with it, application of light for improving production and quality. We also study the development and the behavior of harmful and beneficial insects and their interaction with LEDs. Also an assessment, an objective assessment of, for example, biostimulants or nutrients, fertilizers that are harvested from waste streams are evaluated, and also the interaction of LED with fungal pathogens, and then the effect of these pathogens on the water relations. So basically, how do we tackle indeed these novel, and how do we go further beyond the state of the art, let's say? Well, at the Faculty of Bioscience Engineering, we vision to enhance and further excel and even create new breakthroughs in vertical farming by combining novel technologies, plant sensors, advanced plant models, and virtual reality. I see here that the screen is a bit mixed up, probably because I developed it on a Mac and now present on another. It's a bit a pity because, yeah, it should look nicer, but anyway. Okay, good. We talk about novel technology. So novel technology at our faculty we use for increasing the production and the quality of crops. We use novel technology, and light is there very important. We heard that already before. And we have the opportunity to play and test a new LED technology that allows us to mix the right light. So the right interaction between the plant and the crop in a very dynamic way. We heard already also before that carbon and sugars are very important in the context of taste. Well, at our faculty, we investigate really the carbohydrates, the sugars that are produced due to photosynthesis, how they are transported in the plant in real time, in vivo. And for that, we use technology coming from the medical science, and we use for that plant positron emission tomography. And positron emission tomography allows us to track the sugars in the plant itself. Here you see, for example, an example where we labeled the leaf of a strawberry plant with 11 CO2 as a tracer, and that allows us to track the dynamic transport of the sugars to each individual fruit. And then in that sense, we can learn, we can increase our understanding of how the sugar transport evolves and use it then to increase the quality of our crops. Okay, this novel technology is one thing. What we can do to indeed study the interaction between the plant and the environment in all these concepts is we can use plant sensors and advanced plant models. So basically, by equipping plants with plant sensors, we make them a biological indicator. And as you heard before, we can literally let the plants speak to us by translating the signals that they give us. So we interpret the signals of these plant sensors, and that tells the story what the plant wants to tell us. We use two sensors here as an example, ZEP flow sensors. With ZEP flow sensors, we can track the water transport dynamics, and we can calculate whole plant water use. With point dendrometers and clips, we can measure variations in stem, in fruits, but also the leaf thickness. Now, by combining sensors, these two types of sensors, we create basically a plant monitor, and this plant monitor can be installed on a plant as you see here. And with the ZEP flow sensor, we measure continuously the ZEP flow. So heat is here used as a tracer, and we can follow the amount of water that is taken up by the plant. If we now compare the transpiration rate, so the water vapor that comes through the leaves and is transported to the environment, if we compare this transpiration rate with the ZEP flow that flows into the plant, which is also an integration of the water uptake by the roots, and if this transpiration is larger than the uptake, basically then water will come from internal reserves. So the plants have also internal water reserves, and if these are depleted, the stem diameter, which we measure then with the other sensor, will shrink. On the other hand, in the evening, transpiration goes down, so decreases, but the uptake of the ZEP is still going on. There is a time delay between both, and if that happens, then the living cells that were emptied before, they are refilled. So the cells get again turgor, and that creates a swelling of the cells, and in the end, according to Lockhart's equation, will result in irreversible growth. So we have diameter swelling and growth. Of course, if we are in a vertical farm and we want to use these plant monitoring systems so that we do not need to go and look, but that the sensors in farmers speak to us, we need systems that continuously speak to us, right? So therefore, we monitor these plants in real time. And for that, we use a cloud service, PhytoSense in our case. And here you see the application which we use to look to our plants, and every 5 minutes a new data point comes in so that you really can follow these plants. And I must say it is addictive, so if you start watching it, you will always want to know what comes in the next 5 minutes, so be careful with using this. But it gives us a very powerful tool. So if we look at the sap flow pattern and we, for example, calculate the area below it, then we can quantify the whole plant water use. So you can exactly know how much water your plant has used. So in this case, for example, it was 1.9 liters, which allows you to, for example, do plant-based irrigation. On the other hand, the diameter variation, you see this stem. If you put a sensor on the stem, it's not like a static growth or a linear growth. No, there is dynamics in it. So these plants, they shrink, they swell, and they also grow. And if you look very closely to this slide, they grow during the night. That are the gray boxes that you see, that is night. And plants do grow during the night, not during the day. Why? Because the growth is driven by turgor, and turgor is maximum during the night. Is also linked with carbon, by the way. So, but these shrinkages, we can also use that as a stress indicator, because we want to we want that the plants talk to us, so we need to understand them. So the larger the shrinkage, the top minus the bottom of 1 day, of such a signal, that is the maximum daily shrinkage, and the larger that is, the higher the stress can be. Then, besides indeed stress indicator, we have the daily growth, and that also, of course, gives information. So that is a bit the background, what we do and can do with these signals. And therefore, these, you see these plant monitors, they have a lot of applications, they are important, and so therefore we also invested in these applications. In previous talk on sensing and monitoring, you saw that there are cameras that has been used. Also here in plant phenotyping systems, camera are used, and that is good to use cameras, but they give a snapshot. So what we did is we miniaturized our plant monitor so that it can be applied on the plant in the pot, and it can move on the belt. So you can envision that also in a vertical farm where, for example, plants are transported from the top to the bottom. So this system can be used, and you get continuous information about functioning, being, water and carbon relations. You can also use these sensors for developing early warning and stress detection systems, so that indeed the plant speaks and say, okay, I have stress, you have to do something, or I'm not happy with the light that you gave me, or the amount of nutrients. And the good thing is, you can see the invisible, because it gives you already, for example, the red line is a drought stress plant, where the dashed line is drawn at the minimum, because you see this maximum daily shrinkage, it increases, so there is really stress. Even that plant is not growing anymore, but continuously shrinking. And at the moment where we gave, again, water, that was when visual signs were observed. We saw already with the sensors many days before that there was stress, so you can really develop early warning systems. And even if we go a step further, if we combine these sensors with mechanistic models, so models that describe the reality how a plant functions, then you can even develop, for example, a plant-based irrigation system. When do you need to irrigate and how much? So that is all possible with these sensors. Okay, then this novel technology, plant sensors, virtual advanced plant modeling, comes together in what we call the virtual reality. So this virtual reality, here an example, the Agrotopia greenhouse. It's a 12-meter high greenhouse that is built in Roeselare. Basically, we create a virtual greenhouse with all the structures on it, and we make virtual plants, 3D plants in it. So that means that we have a virtual world where we can play with. So here you see an example of a tomato plant where that you can grow the tomato, but now in a virtual world. And of course, that gives great opportunities. You can use that as an optimization tool. So for example, just to illustrate scenario 1, you use stoplights. You create this virtual reality. In this virtual reality, plants are growing. They grow as a real plant will do, with transpiration, with photosynthesis, with sap flow. They absorb, as such, also the light. And you see, indeed, this gradient. There was this question before, where do you need to measure the light? Well, the light is measured at the top, has been said, but here you see how the light absorption is throughout the plant. You can calculate that, and then you see that scenario 1 is a good scenario because you have now photosynthesis. The crop is doing photosynthesis in comparison with no light. But if you now decide to put the lights as interlights, you do the same reasoning, but you see that it has no use to invest in that light because the photosynthesis stays respiration and you have loss of CO2. So no use to invest in that one. So that's just an example of what can be done. So that is, I think, also as a vision, we should invest in developing these plant models. So here you see a plant growing, but the cool thing is this plant grows in a virtual world, but also all underlying processes that we know so far are included. So there is transpiration, there is photosynthesis, there is absorption of light, and that can really help us in realizing and doing compelling, groundbreaking research. And maybe to illustrate that, one of these groundbreaking research is our Space Bakery project, so where we cultivate wheat in a closed environment. We heard already before from Leo that maybe he put a cross on this crop, but in this case, we need to grow wheat in a closed container because the idea is that you see here the different partners in this project, so Puratos, Urban Crop Solutions, Magix, then we, SEKSN, UHasselt, and supported by Flanders Food. So Puratos is a big bakery company in Belgium, and last year they became 100 years, and they say we have to have a vision, we have to think wide, and they wanted to be the first ones that bake bread on Mars. On Mars, we have then to cultivate the plants in a closed environment. And there we kick in because we are responsible indeed for this virtual reality, for creating this virtual representation of the crop so that we can run the different scenarios, and with this virtual 3D crop of wheat, we can determine the optimal and sustainable way of how to grow then wheat in such a closed biosphere system. So there things come together, we use plant sensors, we use the models, and that is how we create a virtual reality in which we can play and help the vertical farming further. So that was basically a little bit my talk. I'm happy to take any questions. And tomorrow, unfortunately, I will not be here because I have to go back to Belgium, because tomorrow there is the official opening of our Agrotopia vertical farm of 12 meters high. And you see that they came and make some movies at our faculty. So therefore, unfortunately, I have to go back already tomorrow. Thank you.
Wow, cool plan for tomorrow. That looks good. Okay, so we received some questions. What platforms are you using for the virtual reality?
What platforms?
Yes,
we develop everything in GrowImp, and also the development of the mechanistic models we create in a modeling platform, Phytosim. And so we combine those two to create then this virtual reality.
Perfect. Next question is, how big is the advantage of real-time plant monitoring over conventional sensors, for example, for water stress irrigation?
Yeah, for the classical sensors, I assume people then talk about soil sensors. And yeah, you can also measure in the atmosphere, and that's all good because you get then information on the microclimate. But it is important to measure on the plant itself, because then you have the response of the plant on that environment. So it's I will give an example. Suppose that you measure in the soil and your soil sensor says, okay, there is enough water in the soil, then you think, my plant is taking up enough water. But what if the roots are not functioning well? Then it can be that your plant is drying out because there is no water uptake, and indeed your soil sensor then says, it is okay. But if you measure on the plant itself with the plant sensors, then you have the signal of the plant itself. It's a bit like the heartbeat. If we go running, you measure also on your own wrist and not on somewhere in the environment whether the sun is okay. So that is exactly the same. So we need to measure on the plant itself.
Okay, wow. So do we have other questions here in the audience? Yes, there's a question, please. I love the silent moment.
Thanks, Cassie. Very, very nice presentation. When you grow wheat inside an indoor farm and then you use it for bread making, did you come across any quality issues due to the way you grow the wheat in there with high levels of high yield levels and high CO2? Any problems with quality, baking quality?
Yeah, well, we are halfway of the project, and indeed always the quality is checked. And so far, we see that there are opportunities for CO2, also playing with light, and depending on the detail of the treatment that we have, we see that we even can increase the quality in comparison to the classical wheat that is now grown also in Belgium, still outside. So that's the reference, but the Puratos, they take into account, they analyze this, and they found some positive effects of growing it inside.
Thank you. Further questions in the audience here? Okay, then I would say we continue with our panel discussion, and I would like to welcome all our experts here on stage and to take a seat, please. Can I have this? Perfect. No, you have to be on stage too. Okay, you can go there. Thank you very much for also taking the time for our conversation. Yes, I take that one. Then I can see everyone here in the audience when there's some questions. And of course, you still have the opportunity to ask your questions. Feel free to use Slido, please. And then I have here the tablet, and all the questions are coming in, so I can also mention the question. And of course, we also have our digital experts today with us. Thank you very much for joining too. Can you hear us? Okay, they're nodding. Perfect. So, as I said, hybrid events are really like the highest level, but panel discussions in the hybrid versions are even more complicated. This is why I would like to ask you, when you want to say something, please raise your hand. Also here in the live audience, but also please for you two who are digitally with us, please just raise your hand, then I can see that you want to say something, okay? And of course, you can always interact. So if you would like to comment on something, please just go ahead so that we have a fruitful discussion, and everyone, of course, can participate too. All right, so my first question is addressed to every one of you, because I would really like to know about your thoughts and your opinion if vertical farming will be the major form, or do you rather think that food production in the future, that vertical farming will be the major form for food production in the future, or is it just one of several production forms for high-density metropolitan areas? Maybe we start with Kati.
Well, my vision on that is that it will depend on where you are, which place and which opportunities there are. So in places where that you cannot grow and that land is indeed scarce, I am convinced that vertical farming will play a big role. Also, for example, urban vertical farming has, to my opinion, an important role to play. So that is, I think, what will happen.
Francesca, would you like to continue?
Yeah, well, I couldn't say it better. I would add that possibly, there are regions of the world or cities like Singapore or Hong Kong where it might play a big role in on the food security of the city or the country. And I would say that also in places where the consequences of global warming will be higher, that's where possibly there will be a greater development of this technology.
Thank you. Eva?
Yeah, I agree, because it will be geographically determined if it's a good thing or not. But I also see potential for integrating vertical systems in existing greenhouse production. And take like the best from two worlds and combine it.
Thank you. Please go ahead.
Yeah, I wanted to echo, I think, something that Leo said at the beginning, that there's no question we can grow any crop indoors and have a very high quality and a very high yield. The question is, can we do it in a profitable manner? At least that's the challenge for mass production. And I think there have to be a lot of criteria that are met for profitability to be achieved, at least right now. And so in our generation and the next couple of generations, I think it's going to be fairly restricted to high-value crops with a high harvestable index, short production time, et cetera, but going back to some of the first comments, that it's going to be very situational. So areas where water is scarce or land is scarce or other things that can dramatically change the equation, there could be cases where the crops that meet those criteria expand substantially compared to other regions.
Leo, what is your opinion?
Well, first of all, I fully agree with Eric, so I could say something similar and what I heard before. So yes, it depends very much on the region, but also in the context for what type of market you are producing. I think that vertical farming will develop everywhere in the world, and some regions more than in others. But I consider it as one of the production systems, the same as that we are producing now in greenhouses and in field. So we will have some field production, we will have some greenhouse production, and we will also have the vertical farming. And the borders between greenhouse and vertical farming, I think those borders might be not so strict. So there will be a lot of connections, crosstalk. So I think there is a lot of opportunity for vertical farming, but it will not be the only route.
Okay. Tracy, what is your opinion?
I think I probably just agree with what everybody said there. I think they've virtually said it. I think it will be one type of production, depend on the crop, it'll depend on the location and the requirements for that. We're also looking at sort of carbon footprints and things like that. These will all come into play of where those systems are. So I don't think I've got anything really to add to what everybody else has already said.
Morgan, what about you?
Yeah, I'd have to agree with basically what Eric said. I think it's a complementary technology or application, and it can work very well with greenhouse and outdoor production. I don't think it's going to be a major urban production. I think it's going to be suburban at best, because transportation is pretty easy to deal with, but real estate costs are not. And I do think the energy implications really need to be considered in terms of climate change. Any benefits for going indoors are have to be balanced with the energy consumption.
Thank you. Murad?
Thank you. I joined the group. I echo the comments made. I think it will be the integration of technologies, because each technology brings different capabilities and opportunities as well. The challenges are different in different climatic regions, meeting the market expectations. But I also want to say that our future farmers are going to be different than our traditional farmers. So I am also excited with the fact that vertical farming is creating these attractive opportunities and making agriculture again exciting and interesting for our future young generation farmers.
Thank you. Dean, last but definitely not least.
Okay, thank you. Yeah, well, I can certainly echo what everybody else has said. And certainly what Eric brought the point to is that we're going to reach an equilibrium between efficiency and profitability. And I think that, the vertical farming system will be just one part of an integrated system, and where that is geographically is really going to depend on the market. But I think that we can use the technologies and use the advancements to really improve quality. And when we look at quality, that's going to give a really good niche in the marketplace. So when we talk about these new farmers coming into the system, you know, they're going to be technologically advanced. They're going to be aware of efficiency and sustainability, but they're also going to be aware of what markets can they actually capture with crop quality. And I think with all of the engineers and all the plant scientists here at the symposium, you know, we got all the pieces together to say, okay, well, what do we want to go after? Is it going to be a metabolite like CBD or THC or other sort of therapeutic compounds that could then become a very profitable environment in that indoor vertical ag system?
Thank you very much. I saw a lot of people nodding during all your answers. Thank you very much. So you're agreeing with each other. And this is definitely the hugest panel that I've ever had in the moderation. Let's also come to your results that you had in our poll. And remember that the first question was, from your perspective, what are the biggest hurdles and challenges with vertical farming? And what is really dominating is something that is related to energy. So we have energy, which was mentioned most, but we also have energy costs. We have energy use. We have costs separately. Automation, profitability, acceptance, those are the ones that were mentioned mostly, but also there are a few others. I hope you all can see them now so that you can also study what other people see as biggest hurdles and challenges. Let's talk about the second questions that we had in our poll about the biggest, I think it was possibilities and chances that we had. Maybe can we change to the other slide so that I can also present the results that were mentioned there. Is it currently possible? Otherwise, I will continue with the next question, which is also fine. Thank you very much. Oh, this is very clear here. So the opportunities that you most see is space efficiency, that we've also heard quite often today, local production, self-sufficiency, data for sure, and suitable for certain crops as some examples. Thank you very much at this point for taking part in this poll. I think it's also very important for everyone to see what your opinions are. Coming back to the questions, because we had really a lot of questions coming in during the whole day, and of course we had a short Q&A after each presentation, but we didn't have the time to address all of them. So Leo, I would like to ask you some more questions. And as I've said, you can always interact. If you want to add something, please just let me know. What kind of sensors are and will be necessary to collect information about the plants?
Well, I think there are a lot of opportunities for sensors. I think some sensors which are now reasonably available, fluorescence sensors. That was also, I think, in the talk of Tracy, who can give an indication of the photosynthesis. So I would say that's one of it. Kathy in her presentation with the sap flow sensors, I think they're also very useful. And what you also see in general, the camera technologies, yes, Kathy called it a snapshot. Well, I think nowadays it is a bit more than a snapshot because you can do it quite often. So we can monitor many different things. So with a camera, the size, with the fluorescence, the activity of the photosynthesis, or if there is any stress. And the sap flow, and then the term, I would also include the thermal temperature measurement by a thermal camera for a number of reasons, because in the end, it is about the temperature, very important for developmental processes in the plant, and then it is really about the plant temperature rather than the air temperature, and plant temperature is usually also not exactly the same everywhere, so it's about organ temperature. So that we can get from a thermal camera, but also the next is that gives an indication about stomatal opening and transpiration. And I think then combining all those different sensors, the sap flow, thermal camera, and then I forgot, you can weigh the plants. Nowadays, also, depending a bit of what type of crop, if you put everything on the scales, that's another measurement for transpiration. So I mentioned now a lot of things, so ideal, and it comes at a cost. So that might be, if you look at economically, okay, is it all worth? But for me, as a vision is where we are going, combining all that sensor information. All those sensors also have some inaccuracy, but by combining the different sensor information, we can do so much better. So I really believe in sensor fusion, sensor combination, combining it with also with the databases that we have. And that's the nice thing with growing in a controlled environment. And if we are able, or no, we are able, if we are willing to share the data, there's a huge opportunity. So big databases, online monitoring, and of course, then we need the software to combining all that information. I'm sure we are going towards that. I think now at this moment, already quite a bit is possible, but also a lot of development is, well, needed or possible.
Thank you. I take a quick look if anyone wants to mention, add something. Okay, yeah, please.
Maybe I would like to add, because I fully agree that the cameras, just to make clear, the cameras indeed, they give a snapshot, but you can, of course, during the day, take several of these images. But some cameras also, for example, thermal camera, they have some limits when can they be used. For example, if it is dark, it is more difficult also for the fluorescence measurements. So if you can combine the pictures with continuous measurements, but then on less plants, because the advantage of a camera is to image more plants, then indeed by combining those technologies you have I think the opportunity to visualize the plant of the future.
Thank you. Again, anything to add? I'm sorry for always asking, but otherwise I cannot manage to see everyone and to see if there are more comments on that. Okay, Tracy, I would like to ask you one more question. Do you see a relationship between the ambient air temperature of the leaf with the amount of time that the plant is able to absorb your light regime?
We've not specifically looked at that, but I think there's been conversations today as well of interactions between them. And of course, the more light and the less that light's used, etc., that's going to change some of the thermal properties of the leaf, etc. And I think that's where, you know, just going back to the previous questions about sensors, etc., is, you know, combining some of these technologies together as well can be and provide us more information than them individually. So a prime example is using chlorophyll fluorescence as an idea of photosynthesis in CO2 uptake. Thermography is an idea of water loss and transpiration, and therefore we get an intrinsic water use efficiency value. So the combination of these is not only to provide multiple different sensors to really know what's going on, we get greater than the sum of the parts from them. So I sort of deviated off my question that I didn't really answer, but I think yes, there will be a thermal change with the light intensity as well on the plants.
Thank you very much. Then, Murat, one question that we that was also addressed to you, but we haven't had the time. Which factors will be different in space-based vertical farming applications compared to those on Earth?
Well, great questions. There are a lot of similarities when it comes to growing crops in space also relative to the Earth-based applications. And the crop requirements are pretty much similar in terms of the significant variables: air temperature, humidity, light intensity as quality, and other factors. But there are some challenges that are being addressed in terms of the lack of oxygen, especially in the growing zone, hypoxia, and how can we address to that challenge. Also, the airflow, the ability of not being able to mix water with air is another challenge. Convective forces are not there to help the crop to grow optimally, so there is a need to address that challenge also in growing in space conditions. Also, the ability to deliver water and nutrients to the plant, to the growing media, and also preventing salt accumulation over a long period of time for crop production is another challenge that we need to address in the space settings. Of course, energy is another factor, a major factor, because we want to make sure that we are using energy or the demand on energy is minimal, even though there are capacities to provide energy in space settings, but the lesser amounts needed, I think the less volume and mass will be required, which is a significant measure and metric when it comes to space production.
Thank you. Do we have more opinion on that? Okay, then one more question that I find really interesting. I think most of you can answer to that. Are there any mechanisms that minimize the huge investment cost, and are there future plans to disseminate, I hope I spelled correctly, a vertical farming technology to Africa? Murat, you're nodding. Would you like to answer to that question?
Sure. Again, I think vertical farming could be applicable in the setting of Africa as well. I think it can be kept low cost with multi-tier systems, but low cost applications, hopefully growing crops practically in a practical way and offering that for the market. I just want to say that there are opportunities there, but I think education and workforce development there, training the communities in terms of these technologies and their capabilities and also limitations will be key. The sensor technology, low-cost mobile sensing technologies should be integrated to that production setting and also empower farmers in that region, in that location.
Thoughts? Yes, please go ahead.
Maybe I can say, I think there is also quite some interest indeed in vertical farming in Africa. At least I get quite often have got questions from companies interested in investing there. Personally, I think I would not expect first the vertical farms in Africa. I think let's first prove in a bit more rich countries that where cost prices are higher, to prove that it is really economic viable. So yes, in Africa, I think it could definitely, technically also very well, although you have to realize that we need electricity, so you need also to ensure that there is a good infrastructure for the electricity. So technically, yes, could fit very well. If it fits within the whole society, I think it's only at this moment on a relatively small scale. But maybe if you are 10 years further, where most likely in many areas of the world it has proven, where it has more becoming a standard technology, then I think it is, yeah, if I would be an entrepreneur, I would wait a few years in Africa.
Okay, thank you. Other opinions on that?
Yeah, if I can quickly add, I fully agree with what Leo said. I think possibly when technologies that can be like off-grid, also for generating the electricity will be available, this will allow for greater development of the sector. There are some of the advantages of vertical farming, like the limited use of water is certainly a priority locally, so it might be that and also there is a lot of sun that can be used to get like solar energy, but yeah, still I agree with Leo that it's early to discuss on this.
Thank you. Anything else? Okay. Morgan, there was another question for you because we didn't have the time earlier. What is a good fixture? Oh, sorry, I'm not sure how to spell that word.
Efficacy.
Yes, thank you. Efficacy. Thanks.
Sure. I think 2.5 micromoles per joule is good and very typical right now. And you can get that with a white plus red LED combination. I've seen higher, but there is a trade-off as you go higher. as you aim for higher efficacies, you just can't have the full spectrum anymore. So the light becomes magenta, or as some people call it, blurple, and it's harder on the growers. And you do get an efficiency boost or efficacy boost, but it's it creates a pretty unpleasant environment. So I've been working on these white plus red combinations, which maintain some color quality for growers so they can inspect plants visually and have not a terrible environment, and get to 3 micromoles per joule, which is quite a good fixture level efficacy.
I have one more open question that isn't addressed to anyone particular, but maybe it's also interesting to get the different areas into consideration here. Which of the staple crops, corn, wheat, potatoes, rice, etc., would you predict to be the first to be grown profitably, vertically, and why? What do you think? Who wants to start? Okay, I see question marks in the faces. Yeah. I will ask the question again. Which of the staple crops, corn, wheat, potato, rice, etc., would you predict to be the first to be grown profitably, vertically, and why?
I'll go first. This might be controversial. I don't think they can be grown profitably because there's $7.50, give or take, of lighting energy per kilogram dry weight. At least that was an analysis from a few years ago by Bruce Bugby in a paper we wrote together. So and that's with even if LEDs get moderately more efficient, that just doesn't add up. So that's why I think vertical farming plays a complementary role. Maybe it starts crops off, maybe it plays a role in the production cycle for the staple crops, but I don't see it being profitable in general. Also, those crops store well, so why would so some of the incentive to grow them vertically just isn't there.
Opinions? Yes, please.
Yeah, well, I bet on wheat because projects are running already on that. And I agree, it is, vertical farming is a complementary technology, but as we discussed, depending on the place where you are or the spot that you want to be in, let's say on Mars or in a spacecraft and you want to grow it, then even if it is then more expensive, if there is no other way of growing the wheat, then that is the best way to do it. So it depends a bit on how you turn the question, right? Mm-hmm. So, but I think if we think about using vertical farm ideas and novel concepts to improve the growth process on Earth and in the field, that we can learn a lot from what we are doing in a vertical farm. So then it's of course from a scientific point of view and gaining knowledge. But for example, if you can then create a model that performs better because you could control everything, you can also apply that knowledge in the field. And I think that is then the benefit again. So it all depends a bit on what you want and what your aim is.
Thank you. Further opinions? Okay, then I'm also asking the audience here. Of course, you always have the chance to just stand up if you want to say something. Also, if you have a question, just feel free to participate. We already have a very big panel discussion, so we might have one more person here too. Okay, please just then stand up. One more question that we got is which kind of biomass stimulants can be feasible approaches in vertical farming? Who wants and can answer that question? Which kind of biomass stimulants? You wanted to say something?
No, I was going to ask, you're saying biostimulants?
Yeah. Yes.
Okay, I haven't worked with them.
Please go.
No, I have no, well, the only experience I have in
but that's probably outdated. In 30 years of research, in those 30 years, it was quite often that there came companies with some very nice stuff, which gives 100% yield increase. And when we grew that, but then growing crops at a very high rate, like we usually do in the Netherlands in the greenhouse, yeah, I was quite often disappointed. So, and I see now the research is booming on this whole area, so I think there are opportunities. But yeah, I would first optimize all the growth conditions and then add on some biostimulants. Because in many cases where the biostimulants are very effective is if there is a relatively poor growth, plants are under stress, then you can get enormous yield increases. But here, what we are now doing, this is like really top sport, what we do in this vertical farm. Plants are growing really at high levels, and then we need to boost them further. But maybe there might be opportunities for new biostimulants, but I'm not an expert on biostimulants.
Okay, thank you very much for giving an answer.
Maybe if I can add just a wild idea, because indeed we talked about quality of increasing nutrients, certain components in plants. We know also, for example, in tomato, if you stress the plants, that then the tomatoes will have a better quality in those compounds. So just while thinking based on this question, what you could think of is that you indeed not go for the optimal, but that you do stress the plant, but in a certain stress the plant in such a way that you don't affect the production, but that you do stress it, and then potentially, because these biostimulants indeed they work if there is some stress, and the combination of those could work. But I don't know whether there is already research done on that, so maybe could be something to explore further with a certain group. Yeah,
and I think this control, that's not the exact on biostimulants, but stress, but then in a controlled stress, there are a number of options to increase the quality, and then well, tomato, if it is indeterminate growing, then you have continuously fruit, and it is difficult, but with some other crops, you can just do it shortly before harvest, some controlled stress.
I also think that there an opportunity for vertical farming is because you can control everything, and the question is, okay, how do you want to control it? Because you can indeed grow the plant for maximum production, but maybe indeed you need to stress them for having the best quality, and by measuring and modeling and all the things that we have heard playing with the light spectrum, we are now at a stage to do that, and that is so exciting about this vertical farming.
Also, I'm looking forward to always have tomatoes that really taste like tomatoes and not just like water.
Yeah, exactly. But then you need to stress them a bit.
Any further comments on that?
Well, I'll just add that you know, we talk about light, and light can be a form of stress, and I think Dean Kopsel said it something like that we're intentionally or we're managing the stress to elicit these secondary compounds. We have to keep in our back of our mind, though, that usually with a stress, we're also then suppressing growth in some way. So again, I think there are going to be some trade-offs. If you choose to deliver some sort of stress, you'll probably have to sacrifice at least a small decrease in yield. So then the question is, what's more valuable? Sacrificing a little bit of yield for a higher quality, and I think that's going to be very situational.
Maybe I can comment on that, because, for example, if you do measure with these plant sensors, you saw this graph where you have the several days before we see the visual sign. And, well, what we also investigated is the effect of that drought stress on photosynthesis. And photosynthesis only starts to decrease at like 1 day before the visual stress was kicking in. So that means that you have, like, let's say now 5 days where the grower, depending on the quality that you want, can play. And I think that is also still not explored, all these possibilities that you have. Because indeed production, and then then you need the water and you need the photosynthesis, but as long as that is not really hampered, that is really not a problem. But you give this power to have the better quality. So I think there are opportunities.
Possibly. So it's a little bit speculation, right? Because I guess then the question is that sufficient to induce a stress response? So, and I don't know the answer. Maybe it is, or maybe you really need a certain level of stress before you see these plant responses. I don't know.
Please get it.
If the quality aspect includes taste, I think it will be possible to actually sell it with the benefit, because how many times have I not bought in the middle of the winter imported herbs that taste hay and winter tomatoes that doesn't have any acidity or sweetness or anything, and you just stop buying it and you wait for the spring. So I mean, if it's possible to get those qualities into the crop by the way you grow them, at least in Europe, I think it will be possible to sell because of proper taste. I mean, a tomato should taste a tomato and not just water, which they often do in the winter.
And in the US, I think almost year-round.
Any further comments? Take a look. If anyone raises the hand.
I think stressing the plant can be used strategically. Every action that we make comes with a cost associated with that. But at the end, if we are able to justify the outcome, and if the outcome is bringing a premium price or value to it, we can achieve that strategically against stressing the plant, because we don't have to stress the plant during the growth cycle. Maybe just a little bit of adding UV or blue light towards the end of the production, or maybe a salt stress EC increase towards the end of the harvest, or at the time of harvest could enhance flavor or nutrition. I think it will all come down to what is the expected outcome and quality attributes and the price associated with that, or premium price. If the energy or resource input stressing the plant is justifiable, then we can integrate it to the production practices.
Thank you. One more question that we received is, what about technologies like plasma water treatment, oxygen enrichment of nutrient solution via nanobubbles for your feeding nutrients in indoor farming, etc.? Are there comments on that?
I think there were many things in the question. I heard something about nanobubbles and oxygen.
I repeat it, okay? What about technologies like plasma water treatment, oxygen enrichment of nutrient solution via nanobubbles for your feeding nutrients in indoor farming, etc.?
Yeah, so I think with plasma water, there are two types of applications. So plasma water, you can gain nitrogen or nitrate from the nitrogen out of the air. So in that way, you can locally produce your N fertilizer. So that's at least a new technology which could be applied. And there are also plasma waters which can be used as disinfection, or even kill pests and diseases. There's a lot of research ongoing on this topic. So yes, there might be applications on one side for nitrate fertilizer or nitrogen fertilizer, I think, and disinfection. We also do some work on plasma water, where some other universities develop the technology and then we apply it to the crop. Well, simply they produced nitrate, and well, so I would say that works perfectly fine. So it's then on how efficient can they create the nitrate out of the nitrogen in the air.
Thank you.
And I'm not sure if others would, because the question about oxygen is a completely different type of question, I would say. With indoor farming, we often we usually grow soilless, and in particular if deep water cultures are being applied, it is important that there is sufficient oxygen. So yes, that is definitely something to consider. How do we ensure that there is sufficient oxygen? That can be done in many different ways. Nanobubbles could be really a cool way of doing that.
And I think also here there's still a lot of questions. How much, let's say there is oxygen and it is nanobubbles. We have for instance, also some research, not with nanobubbles, but just with bubbles. And if you have a lot of bubbling, you have root movement. In fact, that root movement, we got smaller plants. So there's still, I think there is still a bit to research, but I think also it means that there are some gains possible. So I think it would be interesting that we should do a bit more on oxygen in the rooting zone, and in particular what nanobubbles could mean.
Mm-hmm. Further opinions, statements? Okay. What about pathogens? Will a vertical farm resemble a clean room with complicated filters and airlocks? Who wants to answer?
I've visited some vertical farms that have very strict sanitation controls, similar to maybe a very high-tech greenhouse, where they do their best to prevent introduction of pathogens. And then I've visited vertical farms that have very few safeguards in place. And so will they ever be a clean room? I don't know. But I think it is important to have several practices in place to prevent the introduction of pathogens and insects, because if they get in there, they can be devastating unless you have some sort of biocontrol or something where you can try to salvage your crop.
Yeah, I fully agree. I think we should also take into account the pathogen idea and investigate how that performs in a vertical farm, because not only the plant will change, but also the behavior of the pathogen, or for example insects will change, and then the behavior of the insect or the fungi with the changed plant. So I think that's definitely an area in which we need to invest, because a true sterile environment is hard to reach.
The question was from the live audience, so if you want to comment on that or ask a further question, please just let us know, so then you can comment. Yeah, sure, please go ahead. Sorry.
Okay. Just like you talk about a lot of like the effects of light, temperature, but in the end it's the plant that have the fruit or the seeds. And I don't see too much like about breeding programs specific for greenhouse or vertical farm, and how the breeding programs can affect the future of vertical farm and the GMOs as well, the transgenics.
I think you need to repeat the question, because I can see question marks here, sorry.
The echo is difficult.
There is a little bit of echo in here, so sometimes
it's really hard
difficult to hear.
So yes, here on stage it's really hard to understand everything which comes from the...
Oh yeah, no, I can tell with no microphone.
Can you please repeat it?
Yeah, definitely. With like a lot of breeding programs, like in soybean, corn, and for like fields, and I didn't see yet like a breeding program specific for vertical farm, like plants specific for growing vertical farm, and I didn't see like how the GMOs or transgenic plants can affect or can increase the production in the vertical farm. What's your opinion about the GMOs in vertical farm?
Yes.
Well, I think application of GMO in Europe at this moment is very straightforward. It's not allowed, and that's a political choice. But at this moment, GMO using in production is not allowed. So but that's different in other continents. So this is typically something for Europe. I think that in the end, there is the environment, so we can do an environmental control in the vertical farm, but that can always be, it is about the environment and it is about the genotype, so the cultivar. So I think there would be a lot of room for improvement for, well, in general for breeding, and with GMO it goes much more efficient. So yeah, also here I think there's a lot of improvement, but for Europe, at least at this moment, it is not allowed. But that's different in China or in the US or in Africa. It's different. So there I think there are opportunities to apply it. Because I think we really, other type of cultivars would be better, or we could benefit a lot from other type of cultivars.
Anything to add?
Yeah, if I can add, and I think also, well, maybe there is not yet breeding. There are not yet breeding programs specifically devoted to vertical farming, but seed companies are starting to look at the sector, and I had also some contacts with companies that were willing to start thinking about which type of morphology you want in a vertical farm where you are growing on multiple tires, and you need to increase the light interception. Specifically also on the type of lamps that you are using. So I think there will be a lot of research going on this in the coming years.
I'll comment just a little bit about that. As far as the US goes, I think when you look at the success of genetic technology, it's in the form of trying to get the plant to resist the herbicide or an insecticide application. Okay, so it's more protection of the crop. So in a vertical farm, you guys have laid it out very eloquently. You can reduce all of those stresses. And the other thing about it is the regulatory and the cost aspect of it. To get a genetic event that would go through the regulatory process would be millions, if not tens of millions of dollars. And that's really not advantageous. And I think for the breeding side of that question, I mean, plant breeding is a numbers game. So you're looking at a lot of different progeny that you'll have to screen over multiple environments in order to find that magical genetic combination of what you want. So when you talk about the small scale, the high input, and the cost that Morgan just described as far as producing in a vertical system, it'd be really hard to screen a lot of genetic material to find that right combination. So I think as of now, they're just trying to find the material that works out in the field and maybe in the greenhouse, and is it adaptable or not to vertical farms.
Am I allowed to say one more thing?
Sure.
I would like to make just one statement, because we also used it in a title for the journal Trends in Plant Science, that I think with vertical farming now we have the option effect to move away from genetic modification to environmental modification. So I like that also, that statement, well, from genetic to environmental modification. And I noticed that also with a broad audience, they really like these things that, well, if we don't use genetic modification, then we do it by the environment.
We received so many questions. I go ahead with one more question. Can vertical farming be used to speed up breeding via speed breeding? You are nodding.
Yes.
Just a clear yes, or anything else to add on that?
Yeah, well, basically you get an environment that is uniform. You can have a lot of plants and you can have short cycle, and so I would say that you speed up the process quite easily.
Yes, sorry.
Can I ask a question?
Sure.
In the vertical farms that runs now different places and has been running for a few years, so they have got the first experience in actually how to do it. To what extent, I mean, we have talked a lot about spectral composition of light. So if we forget that part, because there a lot of things is happening, but in the companies, to what extent do they control temperature, air humidity and CO2 in the commercial businesses like this time?
I can share my observations that are mostly in the US. They're very good at controlling temperature if system. So I think the newer companies, as in the last few years, have learned from the mistakes of others, where facilities have been built that have been under-engineered for the HVAC, and result is that they can't control humidity when they fill their entire production facility with plants. And so the result is they may only use 80% or 60% of their facility before they reach a saturating humidity. So assuming they have a properly engineered HVAC system, they can maintain a humidity so that they're not excessive. Usually then that means temperature control is good. I think they're all mindful of airflow, but there's still, I think, a lot of opportunity to learn how to deliver a relatively uniform airflow, and I know Murat Kassira is working on that a lot. So I've been impressed, especially with companies that have built facilities in the last year or two, how well that they have understood environmental parameters, and I think also many of the new ones are enriching with CO2.
Anything else to?
I'll add, and this also goes a little broader to the question, but there is a history of entirely indoor cultivation of marijuana around the world, and that's maybe not what this group wants to hear about directly, but it informs this discussion greatly because they've been breeding plants to be entirely indoor for decades, and they have very sophisticated indoor growth now with almost perfect control of the environment. And this is just a topic I've been pulled into lately. So they control humidity, they control temperature, they can control automated watering and feeding, with really sophisticated systems. So it's a good application to learn from, I think, and not try to over separate from the best practices that are going on there. And they're going vertical now too. And these are huge facilities, in urban areas. And it's, like I said, it's probably not something that actually helps the world, but it is an example of technologies that can be used to help the world. So just a little bit of a broader statement, but in those facilities, I see very, very fine-grained control of all these factors, including CO2. And to the point where if you change the lighting technology, you have to reset up the whole facility because you've adjusted the temperature. And so just wanted to throw that out there.
Can I add to that conversation? I agree with Eric in terms of the major challenges. It's the humidity, vapor pressure deficit, especially, but also the airflow. And we touched on that earlier in terms of maintaining a microclimate around the boundary, closer to the boundary. I think that's where the focus should be, and the conditioning, environmental control should be in terms of monitoring, sensing, and control actions. But I think the vertical farmers are also incorporating more data into their decision making. A lot of data have been collected. I think now they are considering what the data is telling them in terms of the yield or quality attributes, as well as the resources used. They are looking more into the resource use efficiency as a metric rather than I have this much of this yield when I use a photo period of a shorter or longer photo period, or maybe this spectrum or that spectrum. Now they are becoming more familiar and concerned about, okay, what is the cost of me doing or delivering that resource in a strategic way? I think we need to come up with standardization or metrics that are standardized across the vertical farm, so they are comparable and they are valuable informations for design purposes or control purposes. I think vertical farming will move in this direction and they will become more efficient.
Thank you. Maybe we do one more and last question. Would organic production in a vertical farm on soil be economic? For example, indoor permaculture in microbially active soils. Who can answer that? Can you?
I'm not sure. Maybe other. Well, again, in Europe, this is not organic production. In Europe, organic production, it needs an undisturbed soil. I don't think vertical farming is under how the definition of organic is in Europe. That cannot be organic. And I also think there is no need to call it organic. Let's use it for what the benefits are. let's call it what it is. Let's make clear what the benefits are. Let's also be fair about where some bottlenecks are. But it's not organic. Maybe in, so let I only limit it to what the European perspective is, because definitions of organic is not the same in all the different continents. So in the States, it is a little bit different. So I'm not sure. So my answer only is relevant for, let's say, for Europe.
Okay, then let's ask our American experts.
Well, there's been a lot of debate whether plants grown in a water solution can be considered organic, and it's gone back and forth. I honestly don't know where it is now, but I think last I heard it was not considered organic. But I think people are also realizing that, okay, there is certainly a subset of people who are looking for organic, but sometimes there's confusion, or not, maybe consumers don't understand what organic means or doesn't mean. And if you tell them that a product is pesticide-free or locally grown, they may resonate equally well, or maybe even in some cases better than the term organic. So I don't hear many growers. In fact, it's rare when I hear them talk about striving to produce organically. I hear them talk about local production, which seems to be the number one, they think, that resonates with consumers, and then pesticide-free.
Thank you. Yes, please.
In Denmark, we have organic production of potted vegetables for, use on the balcony or in the garden or whatever. And that is approved as organic with the substrate and also organic fertilizers all the way through. And the system the prototype that has been built in the nursery in Denmark, that is also for organic production. Because there is an organic department in that company that produces vegetables. But that's pot-based, not hydroponic. And then you can discuss if that is organic.
Yeah.
But in Denmark, for the time being, it is. Maybe it's not in 5 years' time. We don't know.
So maybe I should say a bit more about what my opinion also is, that I really think that we should not even try to call it organic, because it is different type of philosophy. And I think personally that this is probably even better than organic, but let's see what are the benefits. But calling it organic is a bit of also a sort of a paradigm on what should be organic. And yeah, I think this is so different. I think it would be the wrong route to try to call this organic. That's my personal opinion.
Okay, then I would say, I think we could continue for ages, and there is a lot more to discuss and to talk about. But I think we already received so much input today, so many interesting questions that we've received. And I have to say, I never had a panel discussion with so many questions from the audience. Thanks a lot for really participating today. And thanks a lot to all of our experts. I think it's also a great chance, and not happening so often these days, that we come together from so many different countries to exchange. So thank you very much. And also to thank you, I want to welcome you two on stage again. Do we have another microphone for them? You already have a microphone. Perfect. Perfect. Then Claudia, looks, the word is yours.
It's working. Yeah, so from our side, it was great to have you all here, and also to have you here online. It was great working with all of you, and I want to thank you for coming here, but I also want to thank all the people behind the scenes. I want to thank Dominic and his team from the Technique. I want to thank the TUM team for doing all the work behind the scenes. Without all of you, this wouldn't have worked. It was our first hybrid, and I'm very excited that it worked so well. Thank you. So, and at least for those who came here, we have a little present. I'll give it to you.
Just to add to Claudia's words, so also big thanks from me for everyone who contributed, for all the speakers which came all the way to Freising. Thank you to you, but also to the speakers which are still on the video link. I think we're missing Hans Lammers. He had to move on. So big thanks to all of you. A big thanks to Claudia. and the whole team which was actually behind, maybe they can all just stand up and show how many it actually takes to carry such a symposium. And a big thanks to obviously the audience here and the audience on the link. We continue tomorrow. Tomorrow is another half a day, which will be in German. So I think some of you might stay. For the rest, we close the session. Maybe one person I forgot to thank. It's actually the moderator, Eliza Stein. Thank you very much for taking us through here. Very important person here.
Thank
which
you.
is very well. Thank you.
Yeah. I also learned a lot today. Thank you very much for the chance and opportunity.
That's great. For those who are here, there's another chance to sort of interact. We're having a beer and some bretzels out there. I hope you can stay and continue the discussion. One thing we haven't really talked about, maybe we can bring it up over the beer, it's perennials. What do we do with perennials today? I don't want to open this up here now, but maybe over the beer we can talk about this, perennials in vertical farms.
And before we stop, I have one more hint, which is really important because we need your support again for feedback. So at Slido, again, we have another poll asking you for your feedback because everyone wants to improve, and this can be done with good feedback. So thank you already in advance for taking part in our poll.
Great. Thank you very much, and enjoy the rest of the evening. Okay, and bye to the ones at the video link. Bye-bye.
Bye-bye.
Bye-bye.